Let's go. Okay. Okay. Exit that out. What else going on there? Get out of here. Get out of here, man. Come on, chromosomal. Let's do DNA. We'll do 4. That's how order we're gonna do it in. 4. Let's see what happens. That means completing week 1 today. Maybe tomorrow is when I'll do the timing chart stuff. I think the rest of the stuff for week 2 is probably, I don't know. I think it's probably a lot easier than what we'll do today for week 1. Oh boy. Okay. Start the clock already. Start the clock already. A long sequence of nucleotides. What is it called? A primary sequence of nucleotides. Nucleotides. What am I saying? Yeah, nucleotides. Nucleotides. A chain of nucleotides. Linear sequence. Linear sequence. That's fine. Point mutation. Let's go. Cofactor. Transfers phosphate groups from ATP. Transfer phosphate group using ATP. Essentially. Silento. Or DNA polymerase. Good. Nonsense. Yes, sir. What nonsense are you spouting? It serves to keep the DNA strand pulled apart, so that way the the strands don't re-anneal to each other after helicase is done, breaking bonds, but also so that single strand doesn't fold back onto itself to stabilize the structure. Boom. Don't you know, pump it up. You got to pump it up. Uncle Jeans. Listens. Couple summaries. Indel. Frame shift. DNA poly. Helicase. Transcription. Nucleus. RNA. Yeah, RNA. Nucleosome. H2A, H2B, H3, H4. DNA double helix turns into histone core, histones wrapping around, DNA wrapping around histone cores. 8 of them turn into chromatin, turns into supercore, turns into a chromosome. XO nucleotide repair. Nucleotide excision repair. Exergonic. Negative delta G. During transcription, towards the later stage of transcription, post-transcriptional modification. Undergone. genes, exons. Okay. Okazaki fragments. Max goes down, KM goes down. Lineberger plot parallel. The Henderson up. Slope the same. Introns. Non-coding RNA. Introns. Two genes, tumor repressing protons. Tumor suppressed genes. They stabilize the mRNA. Makes it so that they're not stabilize the mRNA. Makes it so that they're not degraded when they go into the cytosol by endonucleases, and contains the 5' prime cap, which is I think it's and then the poly A tail, and they serve as kind of identifiers for the molecules to signal that it's a mature mRNA ready to be translated. Right. Telomeres. Catalyzes cleavages with water, such as phosphatase. What are we talking about here? The Lac-Operon segment? Maybe we're talking about the Lac-Operon segment? The part that contains three genes? Or not sure. Okay, I didn't know this one. Polycistronic gene. Again. Polycistronic. Binding protein. Translation occurs. Third base. DNA primase. Success of the elongation phase, thanks to tRNA coming in to the ribosomal complex and binding to anticodons. Let's move through the mRNA strand, reading it from the 3' to the 5' and adding from the 5' to the 3'. Okay. Yeah. tRNA. Polycistronic. point mutation. Nonsense. Translation occurs in a cytosol at the ribosome. A ligase joins two molecules together, ligase, such as in the case of DNA replication. Ligase will come in that the nucleotides are binded to the AG, UAA, UAG, UGA. Initiation, Initiation, elongation is the last step. I don't know what the middle step is. Termination. Oops, actually. Initiation, elongation, termination. Silent mutation. DNA ligase. Okay. Missense. Frame shift. Indel. Attaches to the mRNA, I guess, right? Shino sequence. Shino. I don't know if I true, buddy. Why are we learning about prokaryotic translation? I don't really care about bacteria. Oh man. Oh man. Which episode was this? It wasn't even the one that I watched. Is it?
Let's look at our eukaryotic animal. Okay, to start with, A created by enzymes. So happens in the nucleus, talk about in how to.
Prokaryote.
And formal is simply the amino acid methionine, but with a form of glutathione. In case you remember what a form of looks like that. In eukaryotic cells, the first amino acid that can hold polypeptide chains is simply methionine. And it's interesting to note that formal methionine actually acts as an alarm system in the human body. So if you had some bacterial cells in your body that were damaged anyway, there would be these formal methionines floating around, and that tells your body that there are bacteria around and it's going to trigger an immune response. And the poly A tail is actually pretty long, so it's typically anywhere between 100 and 250 nucleotides long, so that's pretty long. So I didn't exactly draw it to scale. And the purpose of both the 5' cap and the poly A tail is to prevent this enzyme to translate till it hits the cell.
Okay. Initiation, elongation, termination. Nucleosome. Transcription. Chromatin into supercoiled DNA. AUG goes for methionine. Enters the ribosome at the A site. And then it's transferred to the P site. Well, kind of like the P site shifts it over to the A site, and then the A site shifts over to the left. And then the A site becomes a P site, and we keep it going. We keep the rotation going. Rotation. A to P to E, A to P to E, A to P to E. But P is always there with methionine, where it binds. That's the start codon. Starts to binds to P, then A, P, E, ape, together, strong. Ape, together, strong. Shundargando sequence. Post-transcriptional modification. RNA. mRNA specifically. Buddy. Translation. Termination. Catalyzes. It catalyzes redox reactions that involve the transfer of electrons. Genes. Right. Genes. Codons. Exons. Codons. Exons. DNA primers. RNA primers. Post-translational modifications. Most phosphate groups. Exons. Introns. Translation occurs. Protein synthesis. Stabilize the mRNA and get degraded. Protects. Succession of pRNAs bind to the A site, which then moves to the P site, which then go exits. The polypeptide chain transfer from P to A goes to P, and move through the polypeptide chain. Ribosome. Polycystronic. Cytosol. Cytoplasm. In the cytoplasm. Initiation, elongation, termination. At the A site. Shindog-Arno sequence. Termination. Post-translational modifications. Exons, mate. Yeah, we're gonna pause it here for now. We're gonna make sure that we I think when we come back we need to read you. We need to read this. And we need to scan for we need to do oh, that's it. Actually, we need to read this one all the way down to 179, and then we'll finish up the Yankee. Okay. But I'll be right back. jacket. It's been on the jacket today for a little bit. Just for a little bit. Just for a little bit. Yes, sir. Just for a little bit. Jibop, jibop, boppity bop, bop. Jibop, jibop, bop. Let's go ahead and sharing and share the going. Share. Bunch of hearts. I don't know if you guys can see. I might just here we go. Zoom in on it. Let's start at I think I read that actually. Yeah, I already read it. DNA mutations. Why is it so bright though? One big one and a big one to know. Nucleotide excision repair or NER. Hey, it's a NER. That's a NER. Helicase. NER. Xeroderma pigmentosa. Oh, really? Xeroderma pigmentosa. Whoa, what the heck? Ouch. Ouch. What's life expectancy on this? Sheesh. I don't know. Life expectancy. What's the life expectancy? All I know is life expectancy. You give me all this stuff, huh? Ooh, you're not that good, huh, buddy? Ooh, shortened by about 30 years. Oh my goodness. 4 people. That sucks. Well, that sucks. Come on now. Chromosomal mutations. Large segments of DNA. Down syndrome, Klinefelter syndrome, Prader-Willi syndrome, Kudet-Chat syndrome. Medical school. Structural abnormalities, changes in smaller sections of chromosomes. This is structural. deletions, duplications, inversions. Think if I took the top and bottom parts and switch their places. Non-homologous chromosomes. Chromosome 1 and the bottom of chromosome 2 and switch them. Numerical. Aneuploidy. Aneuploidy. What the heck? Oh, okay. Polyploidy isn't seen in plants. Monosomy. Three chromosomes at the 21st position instead of two. Staphylococcus aureus. Staph aureus. Bind to the PBBs. Methicillin resistance. Structural mutations. Structural mutations. Okay. Nerd. Sounds like you're saying nerd, right? Which I'm a big nerd. Alright, come on, guys. We gotta at least try for like before 1. You must have been going through something. An karyotype. Oh. Okay. Oh man. For like 20 minutes, I think. So good. Man, why is oatmeal so good, huh? Literally the best. Okay. I am extra awesome. I am EOA. I am extra awesome. I am extra awesome. I am extra awesome. I am independent and multiply. I am independent and multiply. Dude, can we clean this real quick? It's a mess. The table's a mess. Come on, man. Clean it up. Clean up, clean up. Everybody clean up, clean up, clean up. Everybody clean up. It's clean up time, everybody. Clean up, clean up. Everybody clean up, clean up, clean up. Do, 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 do. Who said you can mess around? Oh my goodness. Get back to work. What are you doing? Over there messing around. Get back to work. Yes, sir. Play it. I am extra awesome, independent, and multiply. If two or more than three independent. Either or. And. Either or and. Either or and. Either or and. I need X or Y to happen. I'm guessing there's supposed to be a little R here. Big Y, little y. Big Y, little y. Can you stop it? Just install it then. Go away, man. Can you go away? You need to go away. I am extrosum. I just want to practice for like 10 minutes. Make sure I have it down. Independent and multiply. What was it? Either or add. Oh, really? Let's go. Let's go. Oh, I see. Oh, let's go. Oh, that's it. Okay. 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 What is this? What the heck? What the heck is that? What are you doing? No. Definitely not. Okay, so man, it's almost what's this? We come back. Let's go ahead and do. Okay, so we finished reading the 2 we did yesterday. Man, oh man, oh man. So when we come back, let's go ahead and watch. Well, after Auntie, we'll go ahead and watch these. Go ahead and move on to. Well, I guess we can do the questions, huh? And then we can go ahead and do this. But before that, we're gonna do this one and this one. I guess I might as well just duplicate this, huh? And then so do that, this, and then we'll start on this one. Yep. Giddy up. I need a new card for today. Chromosomal inheritance, DNA technology, evolution and population, and principles of biogenetics. I hope they're like tiny. I think they are, right? Energetics. Yeah, energetics. And we have watch the timing chart. AMC sample test passages. And all to okay. And read yield. I'm just gonna keep it simple for today. We'll grab another card for the other side stuff that I have to do. Yeah. Okay. That's the side stuff. And stuff for today. What is this? I think that's fine. Okay. Let's take our 10-minute. Anybody say anything? Really shouldn't be taking 10 minutes, but whatever. Oh, let's have it on Anki when I come back. I'm already ready. Come on, man. Sorry, guys, I'm late. I know I'm late. I'm back. Make it work. Probably won't take another break. Oh, come on. Turn on. I'm gonna wash my hands real quick. What a day, man. What a day. Today is gonna be a good day. Today is gonna be a great day. Today is gonna be a day where we do 4 sections. Where we do 4 sections. Get over here. Get over here. Yeah. Continue, man. Go, go, go. I don't have time for this. I don't have time for this. Alicia. mRNA. Oh, pre-mRNA. Let's do pre-mRNA. The coding strand, also known as the sense strand. Reach DNA. The heck? The heck? DNA polymerase? Oh. Oh, RNA polymerase. Oh, okay. I was trying to tell me transcription. Okay. Long-term storage of genetic material. Pre-mRNA. Application 4. Primer. DNA primase. Oh, DNA polymerase. Oops. Adenine guanine thymine. AGTC. Complementary to the non-coding strand and identical to the coding strand. Pre-mRNA is almost identical to coding strand. RNA polymerase. The coding strand has the same sequence as the mRNA. This getting churned out. It's getting churned out the RNA polymer RNA ribosomal complex during translation. He's getting churned out the. What is he getting churned out of? Churn out the ribosome? Yes, churn out the ribosome. Bind to the primer. DNA polymerase. Hydrogen. To the region on the DNA called the promoter. No. The promoter? Yeah. Facilitating processes like DNA replication, gene regulation, DNA repair. Nucleotides form hydrogen bonds complementary to one another. Is that what it says? Yeah, I think so. Oh man, this one. Determined by the summation of the ionizable groups, such as the carboxyl group, the amino group, and the R group, in response to the pH of their environment. That's right. DNA stands for deoxyribonucleic acid. Matching the mRNA. No, by matching. Oh yeah, by matching the mRNA. Huh? Matching the mRNA. mRNA codons? Is it mRNA codons? Am I right? No. Is that right? I'm not sure. Yeah. Yeah. I mean, it's a year or so. Antisense strand of template. DNA primase. Cytoplasm or cytosol. From the 5' to 3'. But that's actually true. What is that about? What are you talking about here, buddy? I don't know. Length of DNA. A strand? Oh, it's from both strands. Adding nucleotides from the 5' to 3'. Second carbon. break the bonds between the complementary bases and nitrogenous bases. Oh, we got to unwind it first. DNA poly. DNA nucleotides. More robust, stronger, tighter, harder to break. Stronger, more stronger. DNA nucleotides. Cytoplasm. Well, RER. Yeah. Two strands. Both strands. Complementary RNA. RNA nucleotides. Complementary RNA nucleotides. Unwind the helix. Unwind the helical structure. Strength, right? Strength, robustness. Strength, stability. Reading it from the 5 prime to 3 prime. It doesn't read it, it adds in. DNA has hydroxyl group. Anticodons. Okazaki fragment. Remains in the nucleus. DNA polymerase sliding down here on DNA. Sliding down DNA track from the 5 prime to 3 prime direction. No, the 3 prime to 5 prime direction. Where's that? Unwind helix. Once the polypeptide chain is created, it is shipped off to undergo post-translational modifications, including protein folding. 3 prime to 5 prime. Adenine, guanine, thymine, cytosine. Pre-mRNA. Coding strand. DNA polymerase. RNA polymerase. Motor region. Ribosome. Complementary base pairs. Hydrogen bonds. Codons. Antisense strand or template. metabolism. DNA nucleotides. Both strands. Straight stability of the DNA helix. DNA motor. Helices. RER. Nucleus. Complementary RNA nucleotides. Unwind and that colon here by tRNAs. Peptide. The coping strand, otherwise known as the sense strand. HIV. Helicase. Pre-mRNA. Post-transcriptional modification. Elongation. Polycystoronic genes. Alternative pricing. Really. Complementary RNA nucleotides. Post-transcriptional modification. DNA polymerase codes for a protein. RNA polymerase. Used for transcription is complementary to the mRNA. It is a non-coding strand or antisense strand or the template strand. An allele. The leading strand. Something splicing. Something splicing. Is that what I have to do today? Where's the evolutionary and population dynamic? Oh, right there. Maybe we can get it done. I don't know. Maybe. We're gonna try. We are gonna try. Splicing. Something splicing. Splicing. I don't know. Something splicing. Oh, dang it. Alternative splicing. Chromatin. Chromatin. To, oh man. Chromatin. To densely coiled DNA. Chromatin. Oh, wait, what? Oh, chromatin is unraveled and less condensed. I did not know that. Okay. Unipolary glycogen called the promoter region. Top box. It's got that top box. Okay. Unraveled and less condensed. That is chromatin. Okay. Beginning side of post-translational modification, including protein folding. That's a mouthful. Alternative splicing during transcription. Post-transcriptional modification. Recessive alleles phenotype can be masked by the presence of a dominant allele and requires the presence of two recessive alleles. The first step of replication is to break the hydrogen bond between this one. Helicase. That's not true. The first step is to unwind it. What the heck? What the heck? Give me false info. Chromosome. Nucleosome. Chromosome. Oh, okay. Where two alleles are both expressed in a phenotype. That's called chromosomal linkage, allele linkage. Oops, codominance. Codominance. On a second carbon, on that sugar group. Sugar. No, sugar. No, I think that's a song. And it's on code. Endonuclease. Oh, exonuclease. Exonuclease. I'm probably gonna get rid of this one out. Again. Incomplete. Incrose. DNA polymerase. Two of the same alleles as their genotype. Two of the same homozygous. Homozygous. I'm locking in homozygous. Codominance. Single-stranded. Two different alleles. Heterozygous. The lagging strand. Okazaki effect. So viruses called retroviruses. Excuse me. Enzyme code Reverse transcriptase. Okazaki. A 5' cap. 5' cap. 5' cap. Yeah, right. Something, I think. Let me see. Oh, okay. Oh, triphosphatase. Triphosphatase. So it's both a phosphate and adding a methyl group to the guanine. Gotcha. It's both. Helicase unwinds a little bit to form sections of so that RNA polymerase. Helicase unwinds. Transcription bubble. Recessive. Double shine it. Come on, we gotta go. This looks like plus forward slash plus. Oh, minus. Sorry, minus. Oh, what? Plus. I thought it was minus. No. Why? Ow, my leg. My leg. I thought plus was wild type, and minus was mean knockout. Knockout. Oh. Plus is homozygous. Homozygous dominant. This is functional unmutated gene, and minus is non-functional or mutated gene. Knockout, mutant, disease modeled. Gotcha. I gotcha. I understand. HIV. Oh, retroviruses. What's the same thing? Post-transcriptional modification. Pre-mRNA post-transcriptional modification. Rough. DNA from RNA. Nucleus. nucleus, nucleolus, and a poly-egg tail. Wait, what? And splicing out the introns? There you go. RNA polymerase. DNA primase. No mutations plus plus. Homozygous. Homozygous wild type. Codes for protein. An allele. Poly-A tail. From 3' to 5' direction. Alternative splicing. Chromatin. 2 recessive alleles. Incomplete dominance. Chromosome. Exonucleus. Homozygous. Heterozygous. Retroviruses. Oh, reverse transcriptase. Oh, yeah, that's fine. Co-dominance. Recessive. 5' cat adds a phosphate to the guanine and a methyl to a different part of it. 5' poly-A tail splicing plus /+ nucleolus. Nucleus. Site DNA replication, DNA transcription, post-transcriptional modification. Surrounded by nuclear envelope with 2 phospholipid bilayers called the inner and outer membrane. Nuclear envelope. To make DNA from RNA. Let's go. We're almost done. We got one question. Epigenetics is changes in gene expression not caused by changes in the DNA sequence. There we go. Histoacetylation, DNA methylation. All right, that's fine. That's fine. Okay, let's go. Finally. Pulse. We got 9 hours and 30 minutes. 9 hours and 30 minutes. Alright, let's close that honky. Oh my god. We got roughly 2 hours and 30 minutes between each lesson today. Is that enough? I don't know. We're gonna try. Let's get out of here. Close out the Yankee. Put your back in and Let's go. Let's go. Let's go. All right, let's see if we can answer these medallion genetic questions. Let's start with this one, I guess. 5 questions. Okay, not too bad. Not too bad. It was the article dominant on O is recessive. Okay. Blue feathers are incomplete dominant to the gene for white feathers. What the heck? What the heck? Blue tip feathers. A white bird. A white bird. What is the chance? What is the chance? So you're saying there's a chance. So you're saying there's a chance. Offspring will have blue feathers. We got a 50% chance. That's not correct. What the heck? B is in the dominance. B? How can that make any sense? Incomplete. Oh, yeah. Is it 1/4? 0%? 0%? What are we talking about here? That's exactly what I did. Yeah, blue tip. The baby ostrich BB will have blue tips, and BB will have white. And for the oh, really? What is that? Blue feathers incomplete dominance to the gene for white feather. Incomplete co-dominant. Oh, complete dominant. Incompletely dominant. So what does incompletely dominant mean? Just spend like, I don't know, 10 minutes on this real quick. Start the clock. How? How did you do that? Oh, there you go. Gotcha. I guess I didn't know that. Should have been on the high yield guide. This was a pretty, I guess, important. Maybe I just don't remember it. It could make sense though, but that's okay. Sure. I mean, this is codominant, so they all have. This is true. Pure red. Is this one? Yeah. Yeah. Okay. Agouti, black albino. Dominant the other alleles. Bosses. Okay, small c, b, c, big c. But this one's dominant to the other alleles. So here we have, this one we have technically 2 agouti. And CBCB is dominant to albino. So that means here we have technically 2 black. So we got 2 black and 2 agouti. Let's go. Let's go. Are you not entertained? Only have a little bit of red left. I really like red. That means it's incomplete dominance. And incomplete dominance. We need two dominant alleles for a what? Full trait. Jesus. We need two dominant alleles for a full trait. Yeah. Yeah. This right now. I think that B blood means BB. So we're gonna be B A B A B A B B B B B. We're gonna have just type B and type AB. What the heck? I guess they can be type B and type AB. Oh, what? What the hell? Where do we add O into it? Unless it's this one. I didn't know. I didn't know we needed to add a BO. How is it BO? I don't know. How did I know to do BO? Okay. Oh, okay. Codominance. Um, yeah, I guess that makes sense. Either or and. I should think about this. One of the two dominant alleles. One. No, let's keep going. One of the two dominant alleles. One of the two dominant alleles. Oh, one of the two dominant alleles, but not both. Oh, okay. And that's from one, and then the other one is from the other parent. Gotcha. Well, I gotta do all that. What the heck? Maybe D? D or C? Maybe this B makes sense. I don't think it matters. Maybe D? Let's see. Okay. 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 I don't know. I really should move on. 50%? Sure. I don't know. I gotta move on. Let's just see. That's fine. Okay, let's go. Oh, we are on this one. This one first? Yeah.
How important Gregor Mendel's work was in 1866. He published in 1866. It's important to realize it wasn't like immediately in 1866 or 1867, the whole world changed and everyone said, oh, Gregor Mendel figured it all out. Like a lot of times in science, the big discoveries, the ones that really change people's thinkings, aren't really taken that seriously at first. And actually, Mendel's work, a lot of people either didn't take it seriously or kind of ignored it in 1866. And it wasn't until the early 1900s that people rediscovered his work and realized, wait, there's something very, very powerful here, and we might be able to connect it to things that we're actually observing inside of cells. But let's just remind ourselves about Mendel's work. So for most of human history, we've recognized probably that, okay, it looks like animals, or not just animals, any type of living creature seems to pass on traits to their offspring. I could look at you and I'd say, oh, your hair is kind of like your dad's and your eyes are kind of like your mom's. Maybe your nose looks like something in between. You walk a little bit like your uncle. So we've always recognized that we pass on traits to our offspring, but we didn't really have a rigorous way of thinking about it. And we definitely didn't have any way to make predictions that were testable based on those traits. And that's what Mendel gave us. He said, well, look, I'm observing, and he did this with pea plants, I observe these heritable factors. And there might be heritable factors on, let's say, height. If we're talking about plants, it would be the height of a plant. There might be heritable factors on, let's say, flower color. So flower color. And he recognized that there were different versions of those factors. And so a given plant might have one of the tall versions. So they might have a tall version for the height factor, and they might have a short version. Or they might have two talls, or they might have two shorts, or they might have a red factor and they have a pink factor. Or they could have two reds, it would look like that, or two pinks would look like that. But the important realization was that there was these versions of the factor. Today, we call these factors, we say, hey, there's a gene for height, if there is one, or that there is a gene for flower colors, and those variations.
Let's take just a 5-minute break before we actually get into this one, because I'm actually a little bit. I should finish up, by the way. I should finish eating. The lock cake. eyes. My eyes to burn, my eyes to burn. I don't know what I'm gonna do. Yeah, let's do it.
We call these alleles. So we'd say, hey, you have the variation. You have one copy of the tall allele and one copy of the short. Let me just write this way. Let me say these are all alleles right here. So you have one tall allele, one short allele. And what Mendel did is he realized, well, look, these things are he didn't know how, but these things are the things that get
I kind of want the socks. I don't know, I just feel like I want them.
Passed on from a parent to their offspring. And he started to describe about how they got passed on. He observed that even if you have two of these, that they tend to segregate when you go to the next generation. And what do we mean by segregation? Or I guess we could say the law of segregation. Law of segregation. Well, that means if I'm a pea plant and these are the versions that I have, to my offspring, I might pass on a capital A, the tall one, or I could pass on the lowercase a. I might pass on the tall, or I might pass on the red version of the flower color factor, or I might pass on the pink one. And he also realized that whether or not I pass on the capital A or the lowercase a, it's independent of whether I pass on the capital B or the lowercase b. So they independently assort. How this one assorts is independent of how this one assorts. So independent assortment. Independent assortment. Assortment. Law of independent assortment. And he also observed that some of these versions dominate the other one. So if an offspring has a tall version and a short one, if the tall one is dominant, the observed trait will still look tall. And then the only way they look short is if they have two versions of the short one. And so that one he described as his law of dominance. Law of dominance. And if all of this is completely new to you, I encourage you to watch the videos on Mendelian genetics on Khan Academy. But this is just an appreciation, a little bit of a historical appreciation. But as big of a deal as Mendel's work was, it's also important to realize what he didn't know. He had no idea of how this was actually happening at a molecular level or at a cellular level. And it wasn't until the early 1900s that people started to have fairly robust theories of how this happens. And so in 1902 and 1903, so 1902, 1903, these two gentlemen independently start coming up with the chromosome theory of inheritance. And it's called the Boveri-Sutton chromosome theory of inheritance, because right around the same time, they both started to realize that maybe chromosomes were the actual molecular mechanism, the cellular mechanism, by which these factors segregate and assort. And so this is, let me write this down. This is the Boveri-Sutton, and sometimes called Sutton-Boveri, Boveri-Sutton chromosome theory. Chromosome theory. And even though they're starting to say maybe chromosomes have something to do with it, they still don't know exactly what is inside the chromosomes that are allowing somehow this information to be encoded. And we'll get to that, and we will get to that in a little bit. Let me just underline this. Boveri-Sutton chromosome theory. And so what was their key insight? Well, they started to look inside of cells. Meiosis was observed actually after Mendel published his laws of inheritance.
Right.
And then chromosomes, or how chromosomes behave in meiosis, were discovered after that. And then these guys, they independently studied different organisms. Walter Sutton, he studied grasshoppers. Theodore Boveri, he studied sea urchins. But they looked at meiosis, and they looked at the reproduction and the fertilization during these processes. And they saw that the chromosomes seemed to do things that were very similar to these laws of segregation, laws of independent assortment, laws of dominance. And actually, the law of dominance we'll talk more about in future videos. But he saw that, let's say that you had an organism here, and in this particular organism, I just did it for simplification, it has 2 pairs of homologous chromosomes. So what does homologous chromosomes mean? Well, these 2 are different chromosomes, but they seem to be very similar. It seems like they're kind of the same length, same size, same shape. So that's 1 pair of homologous chromosomes. That's another pair of homologous chromosomes. So notice homologous chromosomes, 2 things that are kind of looking the same, but maybe they're a little bit different. We're not sure. Well, maybe this is what fits what's going on right over here with these factors. Maybe, just maybe one of these chromosomes somehow has on it someplace what encodes for the capital A, and maybe the other chromosome in the similar part of the chromosome, has what encodes for a lowercase a. Now this is starting to make sense because they would be homologous chromosomes, similar. Those chromosomes look like they code for the same thing, for the same factors, for the same genes, but there might be some variation between these chromosomes. And these guys weren't, they weren't able to somehow sequence the chromosomes, so they didn't even know that the DNA was what was important to the chromosomes. But they said, well, it looks like these 2 things, if we look through the process of meiosis, it seems like they segregate from each other. For example, this capital A one, it'll replicate. So you have capital A, and then this is the lowercase a one right over here. You might have some crossover, and we'll talk about that when you can review meiosis if that was unfamiliar. But then they segregate. You can have your capital A ones right over here, and then these sister chromatids split apart. So capital A, and then it seems like they segregate from each other. Chromosomes look like they code for the same thing, for the same factors, for the same genes, but there might be some variation between these chromosomes. And these guys weren't, they weren't able to somehow sequence the chromosomes, so they didn't even know that the DNA was what was important in the chromosomes. But they said, well, it looks like these 2 things, if we look through the process of meiosis, it seems like they segregate from each other. For example, this capital A one, it'll replicate. So you have capital A, and then this is the lowercase a one right over here. You might have some crossover, and we'll talk about that when you can review meiosis if that looks unfamiliar. But then they segregate. You can have your capital A ones right over here, and then these sister chromatids split apart. So capital A, and then you have lowercase a. These lowercase a ones, they segregate. And they independently sort from the other chromosomes. So this one right over here might be the capital B. This might be the lowercase b. And whether or not this gets a capital B or a lowercase b is independent of whether it got a capital A or a lowercase a. So it seems like these chromosomes independently assort. And so they came up with this chromosomal theory that it looks like maybe chromosomes are what contain these heritable factors that Mendel was talking about, because it seems like chromosomes behave very similar to those heritable factors. Maybe chromosomes code for multiple of these heritable factors that segregate and independently assort. And as we know now, they were right. So this was a very, very big deal. But it's important to realize that they weren't sure. They established the theory. They were able to make some observations with the grasshoppers and the sea urchins, and they saw the patterns between what Mendel was describing and the way chromosomes behave during meiosis. And then they know that each of these products of meiosis, each of these gametes will then go and form with other gametes to form the next organism. So parents will contribute either capital A or lowercase a, either a capital B or a lowercase B. So this is very similar to what Mendel was describing. So they laid the groundwork for the theory, but they still weren't sure. They didn't definitively prove it. And it'll take another decade or so until it's definitively proven. And even then, no one was really sure exactly how these different traits were encoded. And for that, we would have to wait a little bit longer.
I guess so, huh? Let's probably do this one first.
What we see right over here, this is a picture of a sperm cell fusing with an egg cell. So that's a sperm cell.
Go fast,
this is an egg cell. We call this an ovum. And even this scene depicted right over here, this is the end of an epic competition, because this sperm cell is one of 2 to 300 million that is vying for this ovum. So there's 2 to 300 million of these characters, and they're all vying for this ovum. And the one that you see that's about to fuse for it, this is the winner of this remember, 200 million to 300 million sperm are trying to get here. So this is a major victory. And to some degree, we should all feel pretty good about ourselves, because we are all the byproduct of that 1 in 200 to 300 million sperm cells that won this race getting to our mother's ovum. So the sperm cell came from our father, and the egg cell, this is all happening inside of our mothers. The egg cell is from our mother. Now, once this happens, let's talk a little bit about the terminology. So once these two fuse, we call this, or the process of them fusing, we call that fertilization. Fertilization. and it produces a cell that then differentiates into all of the cells of our body. So you can imagine this is an important process. So let's make sure that we understand the different terminology, the different words for the different things that are acting in this process. So each of these sex cells, I guess we could say, the sperm cell and the ovum, these are each called gametes. So this right over here is a gamete, and the ovum is a gamete. The egg cell is also a gamete. And as we'll see, each gamete has half the number of chromosomes as your body cells, or most of your or the somatic cells in your body. So outside of your sex cells that might be in your ovaries or your testes, depending on whether you're male or female, these have half the number. So let's dig a little bit deeper into what I mean there. So let's just do a blow up of this sperm cell right over here. So blow up of the sperm cell. And I'm not going to draw it to scale. You see the sperm cell is much smaller than the egg cell, but just to give a sense. So let me draw the nucleus of this sperm cell. So just like that. If we're talking about a human being, and I'm assuming you're a human being, so that might be of interest to you, this will have 23 chromosomes from your father. So let's do them. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And for the 23rd one, that's going to be your sex-determining chromosome. So if your father contributes an X, you are going to be female. If your father contributes a Y, you are going to be male. So these are the chromosomes in the male gamete, or I guess I say the gamete that your father's contributing the sperm. So this is a gamete right over here. And that's going to fuse with the egg, the ovum that your mother is contributing. And once again, I'm not drawing that to scale. So this is the egg. We draw its nucleus. So that's its nucleus. Once again, none of this is drawn to scale. And your mother is also going to contribute 23 chromosomes. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And then she will contribute an X chromosome for the sex determining. So your sex determining chromosomes are going to be XY. You're going to be male. If this was XX, you are going to be female. So this is also a gamete here. So a gamete is a general term for either a sperm or an egg. Now, once these two things are fused, what do we have? Once they're fused, then we're going to have you could say a fertilized egg, but we're going to call that a zygote. So let me draw that. Put this in a new color. I'm running out of space. I don't want this all to feel the same.
Hello? Oh, I got the good one.
Oh, you do?
Yeah.
Oh, well, have it later.
What? Okay.
So you see, if you want to get hungry, what's hot? It's not hot. It's just warm.
Okay, here. Okay, this one. Just put this in that.
How do you know you have to put something? Let's just see for later.
All right, thank you. I'm gonna go to the bathroom real quick. Okay. Okay. Oh, you're right. Oh. I got X from the mother, Y from the father. We got haploids here, 23 haploids. And, 22, and then the last one, sex determining chromosome. Sure.
Piece all on the same screens. This was XX. You are going to be female. So this is also a gamete here. So a gamete is a general term for either a sperm or an egg. Now, once these two things are fused, so once they are fused, what do we have? Once they're fused, then we're going to have you could say a fertilized egg, but we're going to call that a zygote. So let me draw that. This is a new color. I'm running out of space, and I want this all to fit all on the same screen. So I'll draw it not quite at scale. And so let me draw the nucleus of the zygote. I'm going to make the nucleus fairly large so that we can focus on the chromosomes in it. Once again, none of this is drawn to scale. So you're going to have the 23 chromosomes from your father. So let me do that. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and 23. And then the 23 chromosomes from your mother. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, and 23. So you got that X chromosome from your mother. And as you might have noticed, I've drawn them in pairs. So you now have a total. Let me make it clear. You have have 23 chromosomes here. Chromosomes in the sperm. You have 23 chromosomes in the egg, and now you have 46 chromosomes in the fertilized egg. 46 chromosomes. And now that we have a full contingent of chromosomes, and then this cell can now keep replicating, keep splitting, and differentiating into all of what makes you. We call this right over here, we call this a zygote. Zygote. So one way to think about it, the gametes are the sex cells that have half the number of chromosomes, and the zygote is the cell that's now ready to differentiate into an actual organism that has double the number, or that has a full contingency of chromosomes, that has 46 chromosomes. And you see I made them in pairs. And these pairs, we call these homologous pairs. And in each of these pairs, this is a pair of homologous chromosomes. Pair of homologous chromosomes. So what does that mean? Well, that means that in general, these two chromosomes, you got one from your father, one from your mother, they code for the same things. They code for the same proteins, but there are different variants of how they code for those proteins, those traits that you have. So, gross oversimplification is, let's say that there was a gene, let's say that there is a gene on that one from your father that helps code for hair color. Well, there'd be another variant of that gene on the chromosome from your mother that helps code for hair color as well. So these are homologous chromosomes. These two chromosomes code in general for the same things. And so the zygote now you could say it has 46 chromosomes, or you could say it has 23 pairs of homologous chromosomes. And this is, once again, this is the case for human beings. If we're talking about some other species, instead of 23 pairs of homologous chromosomes, or 46 chromosomes in total, you might be talking about 10 pairs of homologous chromosomes, with 20 chromosomes in general. Now, to help biologists, to help clarify when they're talking about the number of chromosomes for a given species, they introduce two words, haploid and diploid. And haploid is referring to when you have half the full contingency of chromosomes. So for human beings, the haploid number is 23. So this is the haploid number. It is 23. For another species, it would be something else. And haploid is based on the prefix haplo, or that's the prefix for single. So you have kind of a single member, I guess you could think of it, of each of the pairs. And now you have both of each pair. You have both chromosomes, or in each pair, or you have the whole contingency. And this 46 chromosomes, this is called the diploid number. The diploid number for humans, they'll refer to the haploid number as N chromosomes, and they'll refer to the diploid number as just twice that, as 2 repeats. And also makes you feel a little bit better about yourself, that just to exist, you, at least, I guess, half of your chromosome.
Right, pretty cool.
In the last video, we saw an egg from our mothers fuse with a sperm from our father to form a zygote that would keep replicating and turn into us. If let's just say that this was the sperm from my father that fuses with the egg from my mother, and then this zygote will keep replicating and eventually turn into Sal and make a video about fertilization. So how does that actually happen? So now that the zygote has a diploid number of chromosomes, once again, they're referring to that as 2N, where N, the haploid number 2N would be the diploid number, and in case of human beings, N is 23. In case of human beings, N is 23, and 2 times N, of course, would be 46 chromosomes. So my full contingency of chromosomes here, I got a Y sex chromosome from my father, X, of course, from my mother, so I'm going to be a male. And so what then happens? Well, now through mitosis, this zygote is going to keep replicating. So it will, after one, we're going to go into the details of the mechanics of mitosis. But after one round of mitosis, it is now 2 cells. And I'm going to draw it, once again, I'm not going to draw it at scale. It's now 2 cells. I want to make sure I have enough space on my little chalkboard here. And it has 2 cells. Instead of drawing all of the chromosomes, let me just say that each of these in my nucleus, I still have 2N. I have the diploid number. So each of these 2 cells that it is differentiated to still have the full contingency. That's what mitosis does. It essentially replicates the entire cells. You have the same number of chromosomes. And then this process is just going to keep happening. These 2 characters are going to replicate. And so then you're going to have through mitosis. And now so this is another round of mitosis right over here. Mitosis. So they just keep duplicating themselves. And each of these cells have the full contingency, 2N, the diploid number of chromosomes for, well, in this case, it's going to be 46 for a human being. And then this process is just going to keep happening. So this process is going to keep happening. I'll do ... to show that a lot of this has been going on. So mitosis is just going to keep happening. And so eventually you're going to have thousands of these cells. And eventually, as we'll see, you're going to have millions and 10 millions of them. So let me draw them really, really, really small. So there's a bunch of them there. And each of them are going to have the diploid number of chromosomes. They're going to have 46 chromosomes, 23 pair of homologous chromosomes. So we now have a big ball of these here. And these cells, some of them they're going to differentiate into me. They're going to differentiate into the different parts of my body. So for example, these cells right over here might eventually, they'll keep replicating, but then it's them and their offspring might eventually differentiate into my brain cells. These cells here will keep replicating, and them and their offspring, I guess you could say, the things that they replicate into, might differentiate into my heart. These right over here might differentiate into my lungs, and of course, all of these eventually will differentiate into their offspring will differentiate into all of the things that make me me. And so you have a lot more of this mitosis. You're eventually going to have a human being. So let me just say this is more mitosis going on. Mitosis, and now let me make an attempt to draw a human being. So that doesn't really look like me, but on here. So S rendering of a human being. That's nice. All right. I'm focusing too much on drawing this human being, but anyway, you get the general. Cells differentiate into the cells into a human being. These cells here in pink eventually differentiate into the cells in the brain. These cells here eventually differentiate into the cells into the lungs. And obviously at this scale, the cells are way too small to even see. These cells differentiate into the cells of the heart. Now, I want to draw an important distinction here, because most of the cells that I've just depicted here that are just a product of mitosis, these are your body cells, or these are your somatic cells. So all of these cells that I'm pointing out in your heart, your lung, your brain, these are somatic cells or body cells, somatic cells. And so you're probably wondering, well, how do I eventually get these haploid number cells? How do I eventually get, if I'm talking about a male, how do I eventually get these haploid sex cells, these gametes, these sperm cells? I'm talking about a female. How do I eventually get these ova, these egg cells that have a haploid number? And the way that happens is some of these cells up here are going to differentiate into germ cells. So they're going to differentiate into germ cells. In the case of, and they're going to differentiate into germ cells, they're going to differentiate into your gonads. In the case of a female, the gonads are the ovaries. And in the case of a male, the gonads are the testes. The gonads are the testes. And the germ cells in the gonads or the cells that are differentiated into being part of the testes and ovaries, those germ cells. So we differentiate them from somatic cells. So they're germ cells there. Germ cells in your ovaries and testes. They, through the process of meiosis, can produce the gametes. So if you're female, they're going to produce eggs. If you're male, you're going to produce sperm. But this is through the process of meiosis. You're going to produce sperm in the case of a man, and you're going to produce ova in the case of a female. And this brings up a really interesting thing, because throughout biology we talk about mutations and natural selection and whatever else. And it's important to realize how mutations may affect you and your offspring. So if you have a mutation in one of the somatic cells here, let's say in a skin cell or in your brain or in the heart, that may affect your ability to, especially if, God forbid, it's a really dangerous thing like cancer, and it happens when you're young, before you've had a chance to reproduce, and you're not able to survive. That might affect your ability to reproduce. But if this is happening in a somatic cell, it's not going to affect the DNA makeup of what you pass on. The DNA makeup of what you pass on, that's determined by what's going on in the gametes. So mutation, if on the way of differentiating into gametes, a mutation happens. So if one of these mutate and then keep replicating, so let's say there's a mutation here, and they keep replicating and they differentiate into the germ cells, the mutation is right over there, and then through meiosis, that produces some mutated sperm, then that would pass on to that has a chance of passing on to your children, because once again, it might not be all of the sperm cells that have that mutation. It could be only a handful of the 200 to 300 million of the sperm cells. And so that mutation somehow makes it harder for some of the sperm cells to kind of function, either fuse with an egg or even potentially develop and kind of swim through fluid. Then it still might not be the thing that makes it. So mutations only affect your offspring in the situation where the mutations in the cells in which they happen are eventually differentiate into gametes that you will pass on to your children. Before I dive into the mechanics of how cells divide, I think it could be useful to talk a little bit about a lot of the vocabulary that surrounds DNA. There's a lot of words, and some of them kind of sound like each other, but they can be very confusing. So the first few I'd like to talk about is just about how DNA either generates more DNA, makes copies of itself, or how it essentially makes proteins. And we've talked about this in the DNA video. So let's say I'm going to draw a small section of DNA, A, G, T, I have two T's, and then I have two C's. Just some small section that keeps going. And of course, it's a double helix. It has its corresponding bases. Let me do that in this color. So A corresponds to T, G with C, it forms hydrogen bonds with C, T with A, C with G. C oversimplification, but the idea is these two strands, they separate from, and those form mitosis. This is that easy to duplicate itself. Guanine, now if we're talking about RNA instead of thymine, this is tRNA, making copies, which we'll talk about more in the future. You have translation occur and proteins get formed. So mRNA, chromosome, I'll write them down here because you can already appreciate how confusing they are. Chromosome, chromatin, and chromatid. So a chromosome is, we already talked about, you can have DNA, you can have a strand of DNA, It's a double helix. Maybe I'll, this strand, if I were to zoom in, it's actually two different helices. And of course, they have their base pairs joined up. I'll just draw some base pairs joined up like that. So I want to be clear, when I draw this little green line wrapped, when we talk about the DNA in combination with the proteins that kind of give it structure, and then these proteins are actually wrapped around more and more, and eventually, depending on what stage we are in the cell's life, you have different structures. When you talk about the nucleic acid, which is the DNA, and you combine that with the proteins, you're talking about the chromatin. So this is DNA plus, you could do the structural proteins that give the DNA its shape, structural proteins. And the idea, chromatin was first used because when people look at a cell, every time I've drawn these cell nucleuses so far, I've drawn these very well-defined, I'll use the words, let's say this is the cell's nucleus. I've been drawing very well-defined structures here. So that's one, then this could be another one, maybe it's shorter, and then it has its homologous chromosomes. So when I drew it like that, if you zoomed in, you'd see one strand, and it's really just wrapped around itself like this. And then it's homologous chromosome. Remember in the variation video, I talked about the homologous chromosome essentially codes for the same genes, but has a different version. And it came from, if the blue came from the dad, the red came from the mom, but it's coding for essentially the same genes. So when we talk about this one chain, let's say this one chain that I got from my dad of DNA in this structure, we refer to that as a chromosome. Now, if we refer generally, and I want to be clear here, DNA only takes the shape at certain stages of its life when it's actually replicating itself. When the cell is dividing, DNA, before the cell can divide, DNA takes this very well-defined shape. Most of the cell's life, when the DNA is actually doing its work, when it's actually creating proteins, or proteins are being essentially transcribed and translated from the DNA isn't all bundled up like this. Because if it was bundled up like this, it would be very hard for kind of the replication and the transcription kind of a machinery to get onto the DNA and make proteins and do whatever else. Normally, DNA, let me draw that same nucleus. Normally, you can't even see it with a normal light microscope. It's so thin that the DNA strand is just completely separated around the cell. I mean, I'm drawing it here so you can maybe the other one is like this, right? And then you have that shorter strand that's like this. And so it's all, you can't even see it. It's not in this well-defined structure. This is the way it normally is. And you have the other short strand that's like this. So you would just see this kind of big mess of a combination of DNA and proteins. And this is what people essentially refer to as chromatin. So the word, it can be very ambiguous and very confusing, but the general usage is when you're talking about the well-defined one chain of DNA in this kind of well-defined structure, that is a chromosome. Chromatin can either refer to kind of the structure of the chromosome, the combination of the DNA and the proteins that give the structure, or it can refer to this whole mess of multiple chromosomes and DNA, of which we have all this DNA for multiple chromosomes and all the proteins all jumbled together. So I just want to make that clear. Now, the next word is, well, what is this chromatid thing? Actually, just in case I don't remember if I labeled these proteins that give structure to the chromatin, or that make up the chromatin, or give structure to the chromosome. Let's say this was my DNA before. Right? When it's just this normal state, I have one version from my dad, one version from my mom. Now let's say it replicates. So my version from my dad, first it's like this. It's a big strand of DNA. It creates another version of itself that is identical if the machinery worked properly. And so that identical piece will look like this. And they actually are initially attached to each other. They're attached to each other at a point called a centromere. Now, even though I have two strands here, they're now attached. And when I have these two strands that contain the exact, I have this strand right here. And then I have, well, let me actually draw a different way. I can draw multiple different ways. I can say this is one strand here. And then I have another strand. Here. Now, I have two copies. They're coding for the exact same DNA. They're identical. I still call this a chromosome. Chromosome. This whole thing is still called a chromosome. But now each individual copy is called a chromatid. So that's one chromatid, and this is another chromatid. Sometimes we'll call them sister chromatids. Maybe they should call them twin chromatids because they have the same genetic information. Chromatid. So this chromosome has two chromatids. Now, before the replication occurred, or the DNA duplicated itself, you could say that this chromosome right here, this chromosome on your father, has one chromatid. You could call it chromatid, although that tends to not be the convention. People start talking about chromatids once you have two of them in a chromosome. And we'll learn in mitosis and meiosis, these two chromatids separate. And once they separate, that same strand of DNA that you once called a chromatid, you now call them individually chromosomes. So that's one of them. And then you have another one that maybe gets separated in this direction. Let me make that, circle that one with the green. So this one might move away like that. Hopefully that clears up a little bit of some of the chromosomes started to look. So that's why that's why they call it a chromosome. So chromatin also will take up, well, I won't go into all of that as well. But hopefully that clears a little bit this whole chromatid, chromosome, chromatin debate. And we're well equipped now to study mitosis and meiosis.
Is mitosis on here? Maybe cellular division, right? Yeah, it's on there, so that's fine. I'm not gonna do this one right now. I am gonna do this one. I am gonna do this one, and I'm gonna do this one. Sure, let's continue. Hmm. Why is this like stubborn?
is to some degree dependent on the idea of variation, that within any population of a species, you have some genetic variation. So for example, if they have a bunch of, well, this is the circle species, and one guy is that color, and then I got a bunch more, maybe some are that color, same color, that one, and that one, and that one. And for whatever reason, sometimes there are no environmental factors that will predispose one of these guys to be able to survive and reproduce over the other. But every now and then there might be some environmental factor, and it makes maybe all of a sudden this guy is more fit to reproduce. And so for whatever reason, this guy is able to reproduce more frequently, and these guys less frequently, and some of them get killed or whatever, eaten by birds or whatever, or they're just not able to reproduce for whatever reason, and then maybe these guys are something in between. And so over time, the frequency of different traits you see in this population will change, and if they are drastic enough, maybe these guys start becoming dominant and start not liking these guys because they're so different, or whatever else. We can see a lot of different reasons. These could eventually turn into a different species. Now, the obvious question is what leads to this variation? Right? In a population, what leads to this? In fact, even in our population, what leads to one person having dirty blonde hair, one person having brown hair, one person having black hair, and we have the spectrum of skin complexions and heights is pretty much infinite. What causes that? And one thing that I kind of point to, and we talked about this a little bit in the DNA video, is this notion of mutations. The DNA we learned is just a sequence of these bases, so adenine, guanine, so you got some thymine going, some more adenine, some cytosine. And that these code, if you have enough of these in a row, maybe a few hundred or a few thousand of these code for proteins, or they code for things that control other proteins, but maybe you have a change in one of them. Maybe this cytosine, for whatever reason, becomes a guanine randomly, or maybe these get deleted. And that would change the DNA. Now, you can imagine, if I went to someone's computer code and just randomly started changing letters and randomly started inserting letters without really knowing what I'm doing, most of the time I'm going to break the computer program. Most of the time, the great majority of the time, this is going to go nowhere. It'll either do nothing. For example, if I go to someone's computer program, if I set a couple spaces or something, that might not change the computer program. If I start getting rid of semicolons and start changing numbers and all that, it'll probably make the computer program break. So either do nothing or let you kill the organism. Most of the time, mutations. Sometimes they might make the actual cell kind of go run amok, and we'll do a whole maybe series of videos on cancer, and that itself obviously would hurt the organism as a whole. Although if it occurs after the organism is reproduced, it might not be something that selects against the organism. But anyway, it also wouldn't be passed on. But anyway, I won't go into detail of that. But the whole point is that mutations don't seem to be a satisfying source of variation. They could be a source or kind of contribute on the margin, but there must be something more profound than mutations that's creating the diversity even within, or maybe I should call it variation, even within a population. And the answer here is really, it's kind of right in front of us. It really addresses kind of one of the most fundamental things about biology, and it's so fundamental that a lot of people don't even question why it is the way it is. And that is sexual reproduction. Sexual reproduction. And when I mean sexual reproduction, it's this notion that you have, and pretty much if you look at all cells all organisms that have nucleuses, we call those eukaryotes. Maybe I'll do a whole video on eukaryotes versus prokaryotes. But it's a notion that if you look universally all the way from plants, not universally, but if you look at cells that have nucleuses, they almost universally have this phenomenon that you have males, and you have females. In some organisms, an organism can be both a male and a female. But the common idea here is that all organisms kind of produce versions of their genetic material that mix with other organisms' version of their genetic material. And if mutations were the only source of variation, then I could just butt off other cells. Maybe other cells would just butt off from me, and then randomly one cell might be a little bit different than whatever else. But that would, as we already talked about, most of the time we would have very little change, very little variation, and whatever variation does occur because of any kind of noise being introduced into this kind of a budding process where I just replicate myself identically, most of the times it'll be negative. Most of the times it'll break the organism. Now, when you have sexual reproduction, what happens? Well, you keep mixing and matching every possible combination of DNA in a kind of in a species pool of DNA. So let me make this a little bit more concrete for you. So let me erase this horrible drawing I just did. So we all have, let me skip the humans because that's what we are. We have 23 pairs of chromosomes, and each of those, in each pair, we have one chromosome from our mother and one chromosome from our father. So let me draw that. So I'll do my father's chromosomes in blue. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. I'm running out of space. Let me do more here. 16, 17, 18, 19, 20, 21, 22. And then I'll throw another one here that looks a little bit different. I'll throw one here that looks like a Y. We'll talk more about the X's and the Y chromosomes. And I have 23 chromosomes from my mother. And not to be stereotypical, but maybe I'll do that in a more feminine color. Let's see. So 23 chromosomes from my mother. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. So what's going on here? I have 23 from my mother. I have 23 from my father. 23 from my father. Now, each of these chromosomes, and I made them right next to each other. So first, let me zoom in on one pair of these. So let's say we look at chromosome number, I'll just call this chromosome number 3. Let me zoom in on chromosome number 3. I have one from my mother. Right here. Remember, actually, maybe I'll do it this way. Remember, chromosome is just a big. If you take the DNA just keeps wrapping around and actually wraps around all these proteins and creates the structure of this. It's just a big. You see it like that. You're like, oh, maybe the DNA. No, but this has millions of base pairs. So, maybe it looks something like that. It's a densely wrapped version of. It's a long string of DNA, and it's normally drawn like this, which is not always the way it is. And we'll talk more about like that. They draw it densely packed like that. So let's say that's from my mother, and that's from my father. Now, these are both. We call them. I'll call them. They're the same. Let's call this chromosome 3. They're both chromosome 3. And what the idea is here is that I'm getting different traits from my father and from my mother. For example, and I'm doing a gross oversimplification here, but this is really just to give you the idea of what's going on. This chromosome 3 maybe contains this trait for hair color. And maybe my father had, I'll use my actual example. My father had very straight hair. So, let's say he had someplace on this chromosome, there's a gene for hair straightness. So there's a little thing right there. And remember that gene could be thousands of base pairs. So let's say this is hair straightness. So my father's version of that gene, or he had the allele for straightness. And remember, an allele is just a version of a gene. Allele, so I call it the allele straight, for straight hair. Now, this other chromosome that my mother gave me, this essentially, and there are exceptions, but for the most part, it codes for the same gene. And that's why I put them next to each other. So this will also have the gene for hair straightness or curliness, but my mom does happen to actually have curly hair. So she has a gene right there for curly hair. So she has the version of the gene here is, let's see, allele curly. The gene just says, look, this is the gene for whether or not your hair is curly. Each version of the gene is called an allele curly. Now, when I got both of these in my body or my cells, and this is in every cell of my body, except for, and we'll talk a little bit in a few seconds about my germ cells, but every cells other than the ones that I use for reproduction have this complete set of chromosomes in it, which I find amazing. But the gene just is work this, but my mother gave me this essentially, and there are exceptions, but for the most part, it codes for the same genes, and that's why I put them next to each other. So this will also have the gene for hair straightness or curliness, but my mom does happen to actually have curly hair. So she has a gene right there for curly hair. So she has the version of the gene here is, let's see, allele curly. The gene just says, look, this is the gene for whether or not your hair is curly. Each version of the gene is called an allele curly. Now, when I got both of these in my body or my cells, and this is in every cell of my body, except for, and we'll talk a little bit in a few seconds about my germ cells, but every cells other than the ones that I use for reproduction have this complete set of chromosomes in it, which I find amazing. But only certain chromosomes, for example, these genes will be completely useless in my fingernails because all of a sudden the straight and the curly don't matter that much. And I'm simplifying. Maybe they will on some other dimension, but let's say for simplicity, they won't matter in certain places. So certain genes are expressed in certain parts of the body, but every one of your body cells, we call those somatic cells, and we'll separate those from the sex cells or the germ cells that we'll talk about later. So this is my body cells. So bodies. So this is the great majority of your cells. And this is opposed to your germ cells. And the germ cells, I'll just write it here just so you get it clear. And for male, that's the sperm cells, and for female, that's the egg cells or the ova. So, but most of my cells have a complete collection of these. What I want to give you the idea is that for every trait, I essentially have two versions, one from my mother and one from my father. Now these right here are called homologous chromosomes. Homologous. What that means is, every time you see the prefix, homologous, or if you see like a homo sapien or the word homosexual or homogeneous, it means same, right? You see that all the time. So what homologous means that they're almost the same. They're coding for the most part, the same set of genes, but they're not identical. They actually they might code for slightly different versions of the same gene. So depending on what versions I get, what is actually expressed for me. So my genotype. Let me introduce another word. I'm overwhelming you with words here. So my genotype is exactly what alleles I have, what versions of the gene. So I got the fifth version of the curly allele. There could be multiple versions of the curly allele in our gene pool, and maybe I got some version of the straight allele. That is my genotype. My phenotype is what my hair really looks like. So for example, so two people could have different genotypes, but they might code for hair that looks pretty much the same. So they might have a very similar phenotype. So one phenotype can be represented by multiple genotypes. So that's just one thing to think about. And we'll talk a lot about that in the future, but I just want to introduce you to that there. Now, I entered this whole discussion because I wanted to talk about variation. So how does variation happen? Well, what's going to happen so first of all, let me put it this way. What's going to happen when I reproduce? And I have a son. Well, my contribution to my son is going to be a random collection of half of these genes. I'm going to contribute either one for each homologous pair. I'm either going to contribute the one that I got from my mother or the one that I got from my father. Right. So let's say that the sperm cell that went on to fertilize my wife's egg, it just happened to have that one. I can just pick one from each of these 23 sets. And you say, well, how many combinations are there? Well, for every set, there's two. I can pick one of two different. I can pick one of the two homologous chromosomes. And I'm going to do that 23 times. 2 times 2 times 2. There's 2 to the 23rd. So there's 22 to the 23 different versions that I can contribute to any son or daughter that I might have. And we'll talk about how that happens when we talk about meiosis or mitosis. That when I generate my sperm cells are essentially takes one. You have instead of having 23 pairs of chromosomes in sperm, you only have 23 chromosomes. So for example, I'll take one from each of those through the process of meiosis, which we'll go into. I'll generate a bunch of sperm cells, and each sperm cell will have one from each of these pairs. One version from each of those pairs. So maybe for this chromosome I get from my dad. I get from the next chromosome I get from my mom. Then I get a couple. Then I donate a couple more from. I should draw next to each other. I donate a couple more from my mom. Then for the chromosome number 5, it comes from my dad, and so on and so forth. But there's 2 to the 23rd combinations here because there are 23 pairs that I'm collecting from. Now my wife's egg is going to have the same situation. There are 2 to the 23 different combinations of DNA that she can contribute just based on which of the homologous pairs she will contribute. So the possible combinations that just one couple can produce. And this is I'm using my life as an example, but you could use this. This applies to everything. This applies to every species that experiences sexual reproduction. Is that so? If I can give 2 to the 23rd combinations of DNA, and my wife can give 2 to the 23 combinations of DNA, then we can produce 2 to the 46th combinations. Now, just to give an idea of how large of a number this is 12,000, roughly 12,000 times the number of human beings on the planet today. So there's a huge amount of variation that even one couple can produce. And if you thought that even that isn't enough, it turns out that amongst these homologous pairs, and we'll talk about when this happens in meiosis, you can actually have DNA recombination. And all that means is that when these homologous pairs during meiosis line up near each other, you can have this thing called crossover, where all of this DNA here crosses over and touches over here, and all this DNA crosses over and touches over there. So all of this goes there, and all of this goes there. What you end up with after the crossover is that one DNA, the one that came from my mom, or that I thought came from my mom, now has a chunk that came from my dad. And the chunk that came from my dad now has a chunk that came from my mom. I'm using the right color. It came from my mom like that. And so that even increases the amount of variety even more. So you can almost now, instead of talking about the different chromosomes that you're contributing, where the chromosomes are each of these collections of DNA, you're now talking about, you can almost go to the different combinations at the gene level. And now you can think about an almost infinite form of variation. You can think about all of the variation that might emerge when you start mixing and matching different versions of the same gene in the population. And you don't just look at one gene. I mean, the reality is that genes by themselves very seldom code for a specific. You can very seldom look for one gene and say, oh, that is brown hair, or look for one gene and say, oh, that's intelligence, or that is how likable someone is. It's usually a whole set of genes interacting in an incredibly complicated way. Hair might be coded for by this whole set of genes on multiple chromosomes, and this might be coded for a whole set of genes on multiple chromosomes. And so then you can start thinking about all the different combinations, and all of a sudden maybe some combination that never existed before all of a sudden emerges, and that's very successful. But I'll leave you to think about because maybe that combination might be passed on or it may not be passed on because of this recombination. But we'll talk more about that in the future. But I wanted to introduce this idea of sexual reproduction to you because this really is the main source of variation within a population. And I mean, it's kind of a philosophical idea because we almost take the idea of having males and females for granted because it's this universal idea. But I did a little reading on it. It turns out that this actually only emerged about 1.4 billion years ago. That this is almost a useful trait because once you introduce this level of variation, the natural selection can start you can kind of say that when you have this more powerful form of variation than just pure mutations, and maybe you might have some primitive form of crossover before, well, now you have the sexual reproduction and you have this variation, natural selection can occur in a more efficient way, so that species that were able to reproduce and essentially recombine their DNA, mix and match it in this way, were able to produce more variety, and were able to essentially be selected for the environment in a more efficient way, so they started to essentially outnumber the ones that couldn't, so it became a kind of a very universal trait. But you could have imagined a world, and there are science fiction books written about this, where you have 3 genders, where you have, gender 1, 2, 3, you could have 10 genders. It just happens to be that on Earth, this notion of having 2 genders turned out to be a very efficient and stable way of introducing variation into a population. So hopefully you found that interesting. In the next video, I'll go more into the detail of how exactly meiosis and mitosis works.
Thumbs up, buddy. Same. This one looks different. And I don't know. I guess that one looks different. What are we talking about here? Looks like a normal human being, no? What the heck? What the heck? What are we talking about here? What are we talking about? What do you mean? Is that true? If that's not, that's false. Chromosome 16? What the? Oh, I think the person's fine. Aren't they going to be different? But number 1 looks different. 16. Oh, there you go. Let's see. What? I think it's gonna be viable. I'm pretty sure you're gonna be completely different. Yeah. Roughly half. Oh, there's A, B in here. Okay, never mind. Sure. There's a quiz on this? No thanks.
Let's give ourselves a reminder of how important
Whoa.
Gregor Mendel's work was in 1866. And he published in 1866. It's important to realize it wasn't like immediately in 1866 or 1867 the whole world changed and everyone said, "Oh, Gregor Mendel figured it all out." Like a lot of times in science, the big discoveries, the ones that really changed people's thinking, aren't really taken that seriously at first. And actually, Mendel's work, a lot of people either didn't take it seriously or kind of ignored it in 1866. And it wasn't until the early 1900s that people rediscovered his work and realized, wait, there's something very, very powerful here, and we might be able to connect it to things that we're actually observing inside of cells. But let's just remind ourselves about Mendel's work. So for most of human history, we've recognized probably that, okay, it looks like animals, and we're not just animals, any type of living creature seems to pass on traits to their offspring. I could look at you and I say, well, your hair is kind of like your dad's and your eyes are kind of like your mom's. Maybe your nose looks like something in between. You walk a little bit like your uncle. So we've always recognized that we pass on traits to our offspring. But we didn't really have a rigorous way of thinking about it. And we definitely didn't have any way to make predictions that were testable based on those traits. And that's what Mendel gave us. He said, well, look, I'm observing, and he did this with pea plants. I observe these heritable factors. And there might be heritable factors on, let's say, height. If we're talking about plants, it would be the height of a plant. There might be heritable factors on, let's say, flower color. So flower color. And he recognized that there are different versions of those, or that there is a gene. This was, if I'm a pea plant case, historical appreciation. But as big as fertilization in a similar part of the chromosome, segregate. You could have your capital A, those heritable behave during meiosis. Still weren't sure. They did until it's definitively proven. And even then, no one was really sure exactly how.
Great, buddy.
Where we left off in the last video, we were in 1902, 1903, and Mendelian genetics had been rediscovered at the turn of the century, and Boveri and Sutton independently had proposed the chromosome theory, that the chromosomes were the location for where these inheritable factors that Mendel first talked about, where they actually were located. But we talked about in that video that was just a theory. This was based on some observations of meiosis and seeing how chromosomes behaved, and they seemed to behave in analogous ways to some of these inheritable factors, but they really didn't have good cellular proof that chromosomes indeed were the location for these inheritable factors. And we don't really start to get that until we start looking at the work of Thomas Hunt Morgan. Now, in 1908, he decides to study fruit flies. So why does he want to study fruit flies? If you've ever seen a fruit fly, they're very, very, very small. So you can actually put a ton of fruit flies in one jar. So that's convenient. You oftentimes don't think about the practical logistics of science, but you could put a lot in one jar. They were actually cheap, and that's another practical concern of science, is you don't always have a lot of resources to do your work. And they had short lives, and they reproduced a lot. So you could very quickly get many, many offspring and many, many generations if you wanted to study how the different traits were passed on or not passed on. And so he spent some time, he started this in 1908, working with the fruit flies, and he kept breeding them in search for some type of a mutant trait. In general, when you look at traits in a species, the wild type, let me write this down, the wild type is the one that's typically seen, while the mutant trait is something that seems unusual. And after 2 years, he finally discovers a mutant trait in his fruit flies. He finds a white-eyed male. So this is the white-eyed male right over here. He says, oh, okay, now this is interesting. Let me take this white-eyed male and begin to cross it with the females. And you say, well, how does this actually occur? Well, what you do is you take a jar full of females and you put the white-eyed male in there, and then the crossing happens. And what was interesting was the inheritance pattern that he saw for this white-eyed trait, because you have the parent generation here, but then in the F1 generation, all of the females were red-eyed and all of the males were red-eyed. And so just off of that first generation, it wasn't clear that anything interesting was going on. But then when he crossed these to each other, and I know what some of y'all are thinking, wait, aren't they all brothers and sisters being crossed to each other? Well, yeah, they're probably half brothers and sisters if they came from different mothers, but some of them could have been brothers and sisters. But yes, that's what people are talking about when they're crossing the F1 generation. But when they crossed these with each other, he saw a pretty interesting pattern. He saw a 3-to-1 ratio of red eyes to white eyes. So for every 4 fruit flies, he would see, let me underline these, he would see 3 red-eyed, and he would see 1 white-eyed. So the white-eyed trait makes a reappearance, which in and of itself is interesting. It shows that this can be passed on genetically. And that's interesting because this was a mutant that just showed up after he did many generations of observations. But what was even more interesting about this 3-to-1 ratio, and that 3-to-1 is something that popped up a lot in Mendelian genetics. What was even more interesting was that he only observed the white eyes in the males in this F2 generation, in this the second generation of the crosses right over there. And so you're thinking, well, why is that a big deal? Well, he was a pretty astute guy. And he says, well, look, if I'm only seeing it in the males, and it's not like he only got 4 offspring. It was in the ratio. He might have had hundreds of them, but it was in the ratio of 2 red-eyed females for every 1 red-eyed male for every 1 white-eyed male. And so across these hundreds in this generation, he only observed the white eyes on the males. And he said, maybe this is in some way related to the chromosome that determines sex. And so what he was able to do is say, well, let's just assume that it is. Let's assume that trait, that mutant allele, that mutant variation of the gene for eye color, let's assume it's carried on the X chromosome. And so the genotype for that first mutant fly, that white-eyed male that he found, we could call it, and this is a notation that people typically use, because this is a gene that we're assuming sits on it's sex linked. It sits on a sex chromosome, in this case, the X chromosome. The way that you would specify the genotype of that white-eyed male is, well, on his X chromosome, he had the white variation. He had the white allele, the white variation of that gene. And then on his Y chromosome, he had no variation for that gene. So we assume that it's only contained on the X chromosome. You probably heard of heterozygous or homozygous. Well, this is a case where you're hemizygous. You only have a version of the allele on one of your two chromosomes, one of the two that you've gotten from each of your parents. So this would be the genotype right here of the white-eyed male. The genotype for the red-eyed female is specified by, so it's on the X chromosome.
Yeah, it's right
and
there.
Females have two X chromosomes, just like in the situation for humans. So on each of the X chromosomes, we assume that the females start off with the red allele. And the red allele, the notation is W+. W+. You might say, well, why don't we just use the letter R? Well, we could have, but the general convention in genetics is to use the letter of the first mutant type discovered for that gene, and then to use this little plus type for the wild type. So the wild type is the red eyes, and then W, which is the first mutant discovered for this gene, is the first mutant allele.
Image is modified by the Mad Boy 74.
So we name it after that white. So this is
Mad
white,
Boy
white
74.
allele, and these right here, these represent the red alleles. So these are the genotype of the red-eyed female. And so when you cross that first generation, well, the white-eyed male, he can either, if he's with he'll either produce sperm that have the X chromosome in it, which is going to contain the allele, or sperm that have the Y chromosome in it, which is not going to contain the allele. And the red-eyed female, well, they produce eggs either way, either which of these X chromosomes they contribute, they're both going to have the wild-type allele. And we can see how this crosses. You could get an X from both parents. If you get an X from both parents, you're going to be female, because you're going to be XX. And each of these females, since you got one wild type and one mutant type, and the wild type turns out to be...
So mutation didn't come from the father, so you can only distribute the Y.
Dominant, they still show their phenotype is still red eyes. They still have red eyes, but now they are heterozygous. They're heterozygotes. They are carrying the white allele. Now the male offspring right over here, well, in order to be male, they got the Y chromosome from their dad. So they're not able to get that white allele, and they get the wild type from their mom. And you can see here, this is why all of the males in that first generation were red. They only got one copy of the allele from their wild type mother. But then what was interesting is what the crosses that you see in that next generation. If you took these red-eyed females that we already established, these are all going to be heterozygotes. And so you can see they have the red allele and they have the white allele. And you cross that with red-eyed males. You cross it with red-eyed males. What is going to happen? Well, the females in this generation, in order to be female, you have to get an X from your mom and your dad. And so they get an X from their dad, which has the wild type there, the dominant red allele. And so regardless of which one they got from their mom, they're still going to be red-eyed females. Some of them might be homozygotes, some of them might be heterozygotes. But now we see something interesting happening in the males. You could have heterozygote male flies here, where they got the X from, where they got the red X from their mom, or you could get the hemizygous white-eyed males, where they got the white allele, the white X from their mom. And this is the exact the exact observation that Morgan made. So it's a very interesting thing that he was able to see. He started breeding these in 1908. He started breeding these flies in 1908. It wasn't until a couple of years that he finally found that first mutant white-eyed male, and it was in 1910 and 1911 that he publishes these discoveries in Nature. And the reason why this is a big deal is he says, look, my observations are completely consistent with this I trait, this gene, being on the X chromosome. So he's able to show a direct linkage between, in this case, sex chromosomes and these heritable factors that Mendel first talked about. And he would go on, and his students that he worked with would go on to study this for many, 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 many years. And he actually ends up getting a Nobel Prize for this work. And this is a big deal because he's finally able to draw pretty substantive connections between these heritable factors of Mendel, this theory of Bovary and Sutton, that maybe chromosomes have something to do with these inheritable factors. And he's showing that this is actually the case. These sex chromosomes seem to carry the trait, or in this particular case, the X sex chromosome seems to carry the gene for eye color in these fruit flies.
Let's take a 5 minute. We're nearly there, but I gotta read this article and then I gotta read a couple more. Right? Well, I think I think that's just. Let's go. Next ones are just all videos. Whew, not bad. When I was gonna skip that one, right? Oh no, yeah, after that's that one. And then evolution and population. And then the principles of bioenergetics, which is super short. We can do it. We can do it today. We have to fight on. But let's take a 5 minute. I'm just gonna take a 5 minute. You guys didn't hear anything I said. What the heck? That's fine. That's okay. Yeah, dude. The goal is to get as big as humanly possible by the end of this year. So let's see if we can hit like 230 pounds. I don't know if it's impossible. We'll try. We'll try. I just have to be able to pass PMT, so we'll see. What if I can gain like 30 more pounds? Like 30 more pounds? Like 10 or 15. Maybe like 10 or 15 at least before I leave. Lose a little bit of weight, and then when I get out, then gain as much as I can. That's probably the way. That is the way. This is the way. Excuse me. Wait, A, going for the same gene, different allele. Right? B, little B, same gene, different alleles. So different chromosomes, gametes made AB, 25% across all. Genes far apart on the same chromosome, A B, A, D, A, B, A, B. Wait, is it the same? Okay. Okay. Dad, mom, DNA replication for meiosis. Right. A, little a. First stage, which side the dad and mom go each pair go to is random. Frequency genes. Frequency. Okay, two different genes, different chromosomes, 4 different alleles. DNA replication. 2 arrangements possible. Right. Right. Yes, sir. Yes, sir. It also ends up being 20%. Okay. Crossing over. Mm. 2%. Mm-hmm. Generations of alleles. Right. Few events will take place as the ones that are further apart genes. Not very often in this case. Okay. Feel very closely the juice will and be tightly linked. More recombination events happening and be less tightly linked. Corrected recombination frequencies. Purple gene wild type dominant. Wild type dominant. Normal red eyes and recessive purple eyes. Vestigial gene. Normal long wings, short vestigial wings. So we want homozygous. So we want a BB flag. BB flag. PR plus, VG plus, both dominant. All right, PR plus, VG plus, both dominant. Yeah, red eyes on. So then VG plus is more. Says this. All right, PR, VG purple. Double heterozygote. It's a double heterozygote. Oh, because the genes are linked really close together, huh? And VG. Which we know, which really is really good. Sure. Okay. Okay. Sure. Well, so heterofly with a tester. With a tester. Homozygous recessive for all the genes of interest. AGR and VG alleles. You got purple, short. Okay. What do we got? What do we got? It's going to be PGR plus, VG plus, PRVG, 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 right? They're not producing equal numbers. Comments divided by total screen times 100. Okay. 151. Oh. One 54. 139. Okay. Larger or farther apart. Be closer apart. Maxes out at 50%. Maxes out at 50%. Which corresponds to genes being unlinked. It's larger than whatever directly measured some gene. It's further apart than the results of it. Or another order on the chromosome ABC, ACB, CAB. Must flank the third gene. AC is the largest, so B must be in between. Or the outermost genes from the trio. Gotcha. Okay. It's refreshing. Not you yet. Over here. Over here. I'm gonna get some water before we officially start. Yeah, I want some more water. We'll start.
fat? How much fat can I get?
All right, let's finish this one. No more going out.
Just in some depth. So let's start with a germ cell. As we mentioned already, a germ cell is a cell that it can either go through mitosis to produce other germ cells, or it can undergo meiosis in order to produce gametes. So this is a germ cell right over here. Let me draw the nuclear membrane. So let me draw the nucleus a little bit larger, because that's where we care a lot about the chromosomes in it. And let me draw a centrosome, which will play a role later on. I want to do that in, let's see, I'll do that in this blue color. Each centrosome has 2 centrioles in it, but I just clarify some of the terminology. And in the mitosis videos, I focused on cells of an organism, I just kind of made it up, that had 2 chromosomes, that had a diploid number of 2, that had 1 homologous pair, that had 1 chromosome from each of its parents. For this video, I'm going to focus on a species, not human beings, that would have 23 pairs or 46 chromosomes. I'm going to focus on a species that's diploid number is 4. And so let's say it has 2 chromosomes from the father. Let me do that. I'll do that in this orange color. I'll do it in the chromatin, or I'll kind of depict the chromatin state. It's kind of unwound. So maybe it has a long one from the father, and it has a short one from the father. And then it has homologous chromosomes from the mother. So it would have the long one from the mother, and it would have the short one from the mother, just like that. And obviously this is a huge simplification, but hopefully this gets the point across. So here it has a diploid number of chromosomes. So this is, let me write this down. This is diploid number is 2. We have 4 chromosomes. And then this germ cell,
Yes,
this is a germ cell right over here. It will go through interphase. So let me draw that. So it will go through interphase, in which it grows, and it can replicate its DNA and its centrosome. And so let me draw that. So after it goes through interphase, I want to use my space carefully because I have a lot of steps to go through. After it goes through interphase, I am going to have in my nucleus here, my DNA will have replicated. So this long chromosome from my father, it will now, all of the DNA will have replicated. So it might look something like that. And it's attached at a centromere. All these centro words. At a centromere right here. But I'm still trying to draw in kind of the chromatin state. It's actually all spread out. It's not bunched up, so you can see it very clearly as these X's in kind of in a simple microscope. So it's just replicated. And after replicating, it is still 1 chromosome. It has twice the genetic material, but it is still 1 chromosome. That 1 chromosome is now made up of 2 sister chromatids. 2 sister chromatids. We talk a lot about that in the mitosis video, but it doesn't hurt to reinforce because it can get a little bit confusing. And then you have that shorter chromosome from the father, and then that also replicates into 2 sister chromatids attached at a centromere. So these are still 2 chromosomes from the father. It has twice the amount of DNA, but it's containing the same information, just duplicate versions of that same information. And the same thing's going to happen from the mother.
Good.
You had that long chromosome from the mother, homologous to this one right over here. It's going to replicate. So it's now going to be 2 sister chromatids. And then you have a short strand from the mother that was homologous to this one from your father. And that's also going to replicate. And so it's like that. And at the end of interphase, it would actually all be spread out. Once again, it won't be bunched up into these clearly discernible X's. I drew them a little bit that way, otherwise, because you would have trouble seeing how it replicated. And we also have replicated our centrosome as we've gone through interphase. Now we are ready. In fact, now we are ready for either mitosis or meiosis. But as I said, the focus of this video is going to be meiosis. So let's do some meiosis. So the first phase, so the first several phases we call meiosis 1, and the beginning of meiosis 1 is prophase 1. So let's see what happens in prophase 1. In order to a germ cell is other germs in some depth. So let's start with the germ cell. As we mentioned already, a germ cell is a cell that it can either go through mitosis to produce other germ cells, or it can undergo meiosis in order to produce gametes. So this is a germ cell right over here. Let me draw the nuclear membrane. So that's, let me draw the nucleus a little bit larger because that's where we care a lot about the chromosomes in it. And let me draw a centrosome, which will play a role later on. I want to do that in, let's see, I'll do that in this blue color. Each centrosome has 2 centrioles in it, but well, I just want to clarify some of the terminology. And in the mitosis videos, I focused on cells of an organism, I just kind of made it up, that had 2 chromosomes, that had a diploid number of 2, that had 1 homologous pair, that had 1 chromosome from each of its parents. For this video, I'm going to focus on a species, not human beings, that would have 23 pairs or 46 chromosomes. I'm going to focus on a species that has, that's diploid number is 4. And so let's the father. Let me do that. I'll do that in this orange color. I'll do it in the chromatin, or I'll kind of depict the chromatin state. It's kind of unwound. So maybe it has a long one from the father, and it has a short one from the father. And then it has homologous chromosomes from the mother. So it would have the long one from the mother, and it would have the short one from the mother, just like that. And obviously this is a huge simplification, but hopefully this gets the point across. So here it has a diploid number of chromosomes. So this is, let me write this down. This is diploid.
Okay.
Diploid number is equal to, we have 4 chromosomes. And then this thing, this germ cell, let me write this down. This is a germ cell right over here. It will go through interphase. So let me draw that. So it will go through interphase in which it grows, and it can replicate its DNA and its centrosome. And so let me draw that. So after it goes through interphase, I want to use my space carefully because I have a lot of steps to go through. After it goes through interphase, I am going to have in my nucleus here, my DNA will have replicated. So this long chromosome from my father, it will now, all of the DNA will have replicated. So it might look something like that. And it's attached at a centromere. All these centro words. At a centromere right here, but I'm still trying to draw it in kind of the chromatin state. It's actually all spread out. It's not bunched up, so you can see it very clearly as these X's in kind of in a simple microscope. So it's just replicated, and after replicating, it is still 1 chromosome. It has twice the genetic material, but it is still 1 chromosome. That 1 chromosome is now made up of 2 sister chromatids. 2 sister chromatids. We talk a lot about that in the mitosis video, but it doesn't hurt to reinforce because it can get a little bit confusing. And then you have that shorter chromosome from the father, and then that also replicates into 2 sister chromatids attached at a centromere. So these are still 2 chromosomes from the father. It has twice the amount of DNA, but it's containing the same information, just duplicate versions of that same information. Same thing's going to happen from the mother. If you had that long chromosome from the mother, homologous to this one right over here, it's going to replicate.
Okay.
So it's now going to be 2. It's now going to be 2 sister chromatids.
Right.
And then you have a short strand from the mother that was homologous to this one from your father, and that's also going to replicate.
Yeah.
And so it's like that. And at the end of interphase, it would actually all be spread out. Once again, it won't be bunched up into these clearly discernible X's. I drew them a little bit that way, otherwise you would have trouble seeing how it replicated. Right. And we also have replicated our centrosome as we've gone through interphase. Now we are ready. In fact, now we are ready for either mitosis or meiosis. But as I said, the focus of this video is going to be meiosis. So let's do some meiosis. So the first phase, so the first several phases we call meiosis 1, and the beginning of meiosis 1 is prophase 1. So let's see what happens in prophase 1. So prophase 1. So let me draw the cell right over here. So prophase 1, a couple of things happen. The nuclear membrane begins to dissolve. This is very similar to prophase when we're looking at mitosis. So the nuclear envelope begins to dissolve. These things start to maybe migrate a little bit. So these characters are starting to go at different ends. And the DNA starts to bunch up into kind of its condensed form. So now I can draw it. So now I can start to draw it as proper. So this is the one from the father right over here. And this is the one from the mother. And I'm drawing them overlapping on purpose because something very interesting happened, especially in meiosis. So this is the mother right over here. This is, let me see, let's onto the centromere in blue now. That's a centromere. That's the centromere. Now this is the shorter ones from the father. These are the shorter ones from the mother. And actually, let me just draw them on opposite sides just to show that they don't have to, the ones from the father aren't always on the left-hand side. So this is the shorter one from the father. It could be all on the left-hand side. It doesn't necessarily have to be. And then this is the shorter one from the mother. And I won't draw these overlapping, although they could have. Shorter one from the mother. And once again, each of these, this is a homologous pair. That's a homologous pair over there. Now, the DNA has been replicated. So in each of the chromosomes in a homologous pair, you have 2 sister chromatids. And so in this entire homologous pair, you have 4 chromatids. And so this is sometimes called a tetrad. So let me just give ourselves some terminology. So this right over here is called a tetrad, or often called a tetrad. Now, the reason why I drew this overlapping is when we are in prophase 1, in meiosis 1, let me label this. This is prophase 1.
Okay.
You can get some genetic recombination, some homologous recombination. Once again, this is a homologous pair. One chromosome from the father that I've gotten from the father, the species or the cell got from its father cell, and one from the mother. And they're homologous in that they might contain different base pairs, different actual DNA, but they code for the same genes. So, you know, oversimplification, but in a similar place on each of these, it might code for eye color or, I don't know, personality. And, you know, nothing is that simple or how tall you get. It's not that simple in DNA, but just to give you an idea of how it is. And the reason why I overlapped them like this is to show how the recombination can occur. So actually, let me zoom in. So this is the one from the father. Once again, it's in all in the condensed form. This is one chromosome made up of 2 sister chromatids right over here. I drew the centromere, not to be confused with centrosomes. That's where they are. Those 2 sister chromatids are attached. And then I will draw the homologous chromosome from the mother. So the homologous chromosome from the mother, just like that. Homologous chromosome from the mother. And the recombination can occur at a point right over here. So after you're done the recombination, this side might look something more like this. So let me draw it. Let me draw it like this. So they essentially break up and they swap those little sections. Is one way to think about it. So this one will now have a little piece from the mother, and it might code for similar genes, but now contains the mother's genetic information. And then this one over here will now have the piece, you could say even the homologous piece, from the father. Let me do this to 2 centromeres. And this is really interesting. This isn't, you know, all the time there could be recombination, and oftentimes it can lead to kind of non-optimal things, nonsense code in DNA. It might lead to a non-functional organism. But this happens fairly common in meiosis. And it's a way, once again, to get more variation. We've talked about sexual reproduction before, and sexual reproduction introduces variation into a population. And this, obviously, when different sperms find different eggs, that introduces variation. But then even amongst homologous pairs, you can actually have exchange between these chromosomes. And that's interesting because, as we mentioned, each of these chromosomes, they code for a bunch of different genes. And a gene is kind of what can code for a specific or a set of proteins. And so this right over here, let's say, and this is what I'm about to say is going to be a huge oversimplification. Maybe right over here you coded for eye color, or it was related to, or it helped code for eye color, and then you got that from your dad. And here it helped code for eye color, and you got that from your mom. Your mom might have trended you towards a lighter eye color, and your dad might have trended you towards a darker eye color. But now the one from your mom is on this chromosome, this gene, and then the one, or they're both the same gene, they're just different alleles. They're coding for different variants of that gene. And then the allele from your dad is over here. And once again, you know, some people get confused with genes and chromosomes and all this. Each of these chromosomes contain a bunch of genes. These are very long DNA molecules. These code for a bunch of different genes. So a gene will be a little section of here that could code for a particular.
Gotcha. So that kind of changes it, right? What's next? Chromosome inheritance, and then I'm doing DNA tag, and then I'm doing the other 2. Yeah, okay.
Protein. So that's what happens in prophase 1. In prophase 1, you have this condensation of your chromosomes, of your homologous pairs. You can have this recombination, and it's really interesting. This recombination doesn't tend to happen at just random points that would kind of break the genetic information. It tends to happen at fairly clean points, and the places where this breakup is happening, these are called, the plural, if you just talk about one point, it's chiasma, or if you're talking about the plural, it's chiasmata. Sounds like it could be a horror movie. So chiasma. chiasma. And the fact that they tend to happen fairly cleanly. This is, once again, kind of the beauty of the universe, or at least of biology, is that through billions of years of evolution, these things have kind of optimized for more variation and to happen in fairly clean ways. So I'm going to leave this video right there. I know I just got to prophase one, but this was a really, really important idea of this homologous recombination, or this chromosomal crossover that we see right over here. And then from there, we can continue through the rest of meiosis 1 and then meiosis 2.
Dun, dun, dun. Dun, dun, dun.
At this point in the biology playlist, you're probably wondering a very natural question. How is gender determined in an organism? And it's not an obvious answer, because throughout the animal kingdom, it's actually determined in different ways. In some creatures, especially some types of reptiles, it's environmental. Not all reptiles, but certain cases of it. It could be maybe the temperature in which the embryo develops will dictate whether it turns into a male or a female, or other environmental factors. And in other types of animals, especially mammals, of which we are one example, it's genetic basis. It's a genetic basis. And so your next question is, hey Sal, so let me write this down. In mammals, it's genetic. It's genetic. So like, hey, maybe there are different alleles, a male or a female allele. Then you're like, hey, there's so many different characteristics that make that differentiate a man from a woman. Maybe it would have to be a whole set of genes that have to work together. And to some degree, your second answer would be more correct. So let me just draw it's even more than just a set of genes. It's actually whole chromosomes determine it. So let me draw a nucleus. That's going to be my nucleus, and this is going to be the nucleus for a man. So 22 of the pairs of chromosomes are just regular non-sex determining chromosomes. So I could just do, that's one of the homologous, 2, 4, 6, 8, 10, 12, 14. I can just keep going, and you eventually have 22 pairs. So these 22 pairs right there, they're called autosomal. And those are just our standard pairs of chromosomes that code for different things. Each of these right here is a homologous pair. Homologous, which we learned before. You get one from each of your parents. They don't necessarily code for the same thing, for the same versions of the genes, but they code for the same genes. If eye color is on...
Serious. Why does it have to freeze up now?
This gene is also on that gene. And the other gene, the homologous pair, you might have different versions of eye color on either one. That determines what you display. But these are just kind of the standard genes that have nothing to do with our gender. And then you have these two other special chromosomes. I'll do this one. It'll be a long brown one, and then I'll do a short blue one. And the first thing you'll notice is that they don't look homologous. How could they code for the same thing when the blue one's short and the brown one's long? And that's true. They aren't homologous. And these we'll call our sex-determining chromosomes. Sex-determining chromosomes. And the long one right here, it's been the convention is to call that the X chromosome. Let me scroll down a little bit. And the blue one right there, for that as the Y chromosome. And to figure out whether something is a male or a female, it's a pretty simple system. If you got a Y chromosome, you are a male. So let me write that down. So this nucleus that I drew just here, obviously you have a whole broader cell all around here. This is the nucleus for a man. So if you have an X chromosome, and we'll talk about in a second why you can only get that from your mom, an X chromosome from your mom, and a Y chromosome from your dad, you will be a male. If you get an X chromosome from your mom and an X chromosome from your dad, you're going to be a female. And so we can actually even draw a Punnett square. There's almost a trivially easy Punnett square, but it kind of shows what all the different possibilities are. So let's say this is your mom's genotype for her sex-determining chromosome. She's got 2 X's. That's what makes her your mom and not your dad. And then your dad has an X and a Y, and a Y chromosome, and has a Y chromosome. If you do a Punnett square, what are all the different combinations of offspring? Well, your mom could give this X chromosome, and in conjunction with this X chromosome from your dad, this would produce a female. Your mom could give this other X chromosome with that X chromosome. That would be a female as well. Or, well, your mom's always going to be donating an X chromosome, and then your dad is going to donate either the X or the Y. So in this case, it'll be the Y chromosome. So these would be female, and those would be male, and it works out nicely that half are female and half are male. But a very interesting and somewhat ironic fact might pop out of you when you see this. What determines whether someone is, or who determines whether someone is, whether their offspring are male or female? Is it the mom or the dad? Well, the mom always donates an X chromosome. So in no way does a mom, what the haploid genetic makeup of the mom's egg, of the gamete from the female, in no way does that determine the gender of the offspring. It's all determined by whether, let's say these are all of, let me just draw a bunch of, dad's got a lot of sperm, and they're all racing towards the egg, and some of them have an X chromosome in them, and some of them have a Y chromosome in them, and obviously they have others. And obviously the one, if this guy up here wins the race, or maybe I should say this girl, if she wins the race, then the fertilized egg will develop into a female. If this sperm wins the race, then the fertilized egg will develop into a male. So and the reason why I said it's ironic is throughout history, and probably the most famous example of this is Henry VIII. Henry VIII. You have these, it's not just the case with kings, it's probably true because most of our civilization is male dominated, that you've had these men who are obsessed with producing a male heir to kind of take over the family name, and in the case of Henry VIII, take over a country. And they become very disappointed, and they tend to blame their wives when the wives keep producing females, but it's all their fault. Henry VIII, the most famous case was with Anne Boleyn. She's some, I'm not an expert here, but the general notion is that, look, he became upset with her that she wasn't producing a male heir, and then he found a reason to get her essentially decapitated, even though it was all his fault. His fault. He was maybe producing a lot more sperm that looked like that than was looking like this. He eventually does produce a male heir, so he was, and if we assume that it was his child, then obviously he was producing some of these, but for the most part, it was all Henry VIII's fault. So that's why I say there's a little bit of irony here, is that the people doing the blame are the people to blame for the lack of a male heir. Now, one question that might immediately pop up in your head is, Sal, is everything on these chromosomes related to just our, maybe sex-determining traits, or are there other stuff on them? So let me draw some chromosomes. So let's say that's an X chromosome, and this is a Y chromosome. Now the X chromosome, it does code for a lot of more things, although it is kind of famously gene poor. It codes for on the order of 1,500 genes. And the Y chromosome is pretty, it's the most gene poor of all the chromosomes. It only codes for on the order of 78 genes. I just looked this up, and who knows if it's exactly 78. What it tells you is it does very little other than determining what the gender is. And the way it determines that, it does have one gene to just our, maybe sex determining traits, or are there other stuff on them? So let me draw like that.
One's the small one?
Even though you have someone is male, whether their offspring are male or female, is it the mom or the dad? One question that, it's the most gene poor of all the chromosomes. It only codes for on the order of 78 genes. I just looked this up, and who knows if it's exactly 78. What it tells you is it does very little other than determining what the gender is. And the way it determines that, it does have one gene on it called the SRY gene. You don't have to know that. SRY, that plays a role in the development of testes, or the male sexual organ. So if you have this around, this gene right here can start coding for things that will eventually lead to the development of the testicles. And if you don't have that around, that won't happen. So you'll end up with a female. And I'm making gross oversimplifications here. But everything I've dealt with so far is this is clearly plays a role in determining sex. But you do have other traits on these genes. For example, and the famous cases all deal with specific disorders. So for example, color blindness, the genes, or the mutations, I should say. So the mutations that cause color blindness, red-green color blindness,
Are they on
which
there?
I did in green, which is maybe a little bit maybe inappropriate. Color blindness, and also hemophilia. Hemophilia. This is an inability of your blood to clot. Actually, there's several types of hemophilia, but hemophilia is an inability for your blood to clot properly. And both of these are mutations on the X chromosome. Mutations on the X chromosome. And they're recessive mutations. They're recessive. So what does that mean? It means both of your X chromosomes have to have, let's take the case for hemophilia, both of your X chromosomes have to have a hemophilia mutation in order for you to show the phenotype of having hemophilia. So for example, if there's a woman, and let's say this is her genotype, she has one regular X chromosome, and then she has one X chromosome that has I'll put a little superscript there for hemophilia. She has the hemophilia mutation. She's just going to be a carrier. Her phenotype right here, her phenotype is going to be no hemophilia. No hemophilia. She'll have no problem clotting her blood. The only way that a woman could be a hemophiliac is if she gets two versions of these, because this is a recessive mutation. Two versions. Now this individual will have hemophilia. Now men, they only have one X chromosome. They only have one X chromosome. So for a man to exhibit hemophilia, to have this phenotype, he just needs it only on the one X chromosome he has.
Oh, never.
So, and then the other one's a Y chromosome. So a man will have, so this man will have hemophilia. So a natural question should be arising is, hey, this guy, let's just say that this is a relatively infrequent mutation that arises on X chromosome. The question is, who's more likely to have hemophilia, a male or female? All else equal, who's more likely to have it? Well, if this is a relatively infrequent allele, a female, in order to display it, has to get 2 versions of it. So if the frequency of it, let's say that the frequency of it, and I looked it up before this video, roughly they say between 1 in 5 to 10,000 men exhibit hemophilia. So let's say that the allele frequency of this is 1 in 7,000 is the frequency of XH, right? The hemophilia version of the X chromosome. And that's why 1 in 7,000 men display it, because it's completely determined whether, there's a 1 in 7,000 chance that this X chromosome they get is the hemophilia version. Who cares what the Y chromosome they get is, because that essentially doesn't code at all for the blood clotting factors and all the things that drive hemophilia. Now, for a woman to get hemophilia, what has to happen? She has to have 2 X chromosomes with the mutation. Well, the probability of each of them having the mutation is 1 in 7,000. So the probability of her having hemophilia is 1 in 7,000 times 1 in 7,000, or that's 1 in 49 million.
It's quite a lot.
So as you can imagine, the incidence of hemophilia in women is much lower than the incidence of hemophilia in men. And in general, for any sex-linked trait, if it's recessive, if it's a recessive sex-linked trait, which means men will, if they have it, they're going to show it because they don't have another X chromosome to dominate it. Or for women to show it, she's have to have both versions of it. The incidence in men is going to be I guess we could say it's, so let's say that P is, or let me write, M is the incidence in men, incidence, spelling badly, incidence in men. Then the incidence in women will be what? You could view this as the allele frequency of that mutation on the X chromosome. So women have to get 2 versions of it. So the woman's frequency is M squared. And you might say, hey, that looks like a bigger number, I'm squaring it. But you have to remember that these numbers, the frequency is less than 1. So in the case of hemophilia, that was 1 in 7,000. So if you square 1 in 7,000, you get 1 in 49 million. Anyway, hopefully you found that interesting, and now you know how we all become men and women, and even better, you know whom to blame when some of these, I guess, male-focused parents are having trouble getting their son.
What the heck?
Let's take a look at the nucleus of a cell that's just
Finally, we
starting to
got
undergo.
Ephrat. I like this girl. It's way better than Sal.
Meiosis. So we have these 46
We gotta
chromosomes,
slow her down.
and beforehand each chromosome probably looks something. Let's take a look at the nucleus of a cell that's just starting to undergo meiosis. So we have these 46 chromosomes, and beforehand each chromosome probably looks something like that. That's the centromere in the middle. And then as meiosis is beginning,
Chromosomes do not duplicate during meiosis. They duplicate before meiosis during the S phase of interphase.
they will duplicate into, each chromosome will duplicate into something like that. That's again that centromere in the middle. So we have, they will duplicate. And then we're going to say that the blue ones are the paternal chromosomes. They come in homologous pairs. And the nucleus that we're looking at must belong to the cell of a male, because right over here you can see that is the Y chromosome. It's a bit smaller than most of the other chromosomes. I'm going to digress just for a moment to clarify a very common point of confusion, and that is that if you look over here at these 2 chromosomes, well, this is considered one chromosome, but so is this called one chromosome. And this can be confusing because this is only one chromatid and this is 2 chromatids. So why are we calling them each one chromosome? And the answer to that question is because we count chromosomes by the number of centromeres. So this single chromatid has 1 centromere, but these 2 chromatids are attached in the middle by 1 centromere. So we also call that one chromosome.
Okay.
Just keep that in mind. It's just a technical point to know that even though they're different, they're still both considered one chromosome.
Sure.
And in this chromosome, the 2 chromatids are duplicates of each other. So it's just a copy of itself. Anyway, back to our nucleus. So we have these 46 chromosomes, 23 homologous pairs, and they're not, the chromosomes are not necessarily arranged in the way that I drew them. I just drew it that way for the sake of organization. But we have these pairs, and we're going to focus on one pair of homologous chromosomes. But I want you to keep in mind throughout this video that whatever we're describing that's happening to this pair of homologous chromosomes is also happening to the other 22 pairs of homologous chromosomes. It's just that it would be too hard to depict in a video how that's happening, but keep in mind it's not just happening to the pair that we're talking about, but what we're just going to be describing is happening to all of the pairs of chromosomes in the nucleus. So here we have our pair of homologous chromosomes. And during prophase 1 of meiosis, the homologous chromosomes pair up with each other and form a unit called a tetrad. And it's called a tetrad because, well, tetra means 4, and this unit has 4 chromatids, right? 1, 2, 3, and 4. And the process during which the homologous chromosomes pair up with each other is called synapsis. So during synapsis, the homologous chromosomes will get a little bit closer to each other, something like that. And at a certain spot, they might actually cross over or overlap. So I'm going to circle that spot, and that's called the chiasma. And in some cases, another thing happens. This protein complex that resembles something like a railroad track forms. We'll see in a minute why. And this is called railroad track forms. Protein complex that resembles something like a railroad track forms. We'll see in a minute why. And this is called the synaptonemal complex.
Okay.
You can actually see the word synapse in there because this happens during synapsis. So we form the synaptonemal complex, and with the help of the synaptonemal complex, these 2 chromatids, the ones that are crossing over, will actually swap material downward of that point. So we're going to get something that looks like that. Look at how the purple chromosome now has some blue over there, and look at how the blue chromosome now has some purple over there. And the way that happened was that the DNA in the chromosome, actually some bonds in that DNA broke, and the DNAs just kind of swapped places. So what we just described, this process by which the 2 chromosomes swap information, is called crossing over. Or another way to say this is genetic recombination.
Genetic recombination.
And let's see why this is called genetic recombination. So we're going to fast forward to the end of meiosis 2, where the chromosomes get split into 2, and all the chromatids get separated into.
What is this guy saying so much stuff? What the heck? What is YouTube viewers? What is going on? Okay, here's the video.
Different gametes, 2, where the chromosomes get split into 2, and all the chromatids get separated into different gametes, and I want a recombination. And let's see why this is called genetic recombination. So we're going to fast forward to the end of meiosis 2, where the chromosomes get split into 2, and all the chromatids get separated into different gametes. And I want to pause and remind you that everything we're describing that's happening to this pair of chromosomes is also happening to all the other 22 pairs of homologous chromosomes. But anyway, so now let's put each one of the chromatids in a different gamete and look at how we get 4 different gametes. And we can call these 2 gametes recombinant. And we're calling them recombinant because they have a combination of alleles that's new. We haven't had this combination of alleles even in a parent. And just to clarify things, let's see what the gametes would look like if crossing over did not happen. So let's go back to our original chromosomes and let's split them.
We don't have
And let's put them into 4 different gametes. And we are going to get that. And you can see that in this case, we only have 2 different types of gametes. So we can see how genetic recombination increases genetic
Diversity.
variability,
Okay. Right.
which is usually a good thing.
Yep. Which is usually a good thing. Yep. Yep.
One of the very interesting things about genetic recombination is that you can actually use genetic recombination to figure out the distance between genes on a chromosome. And for you to do this to all the genes on a chromosome, you could actually map out the chromosome and figure out exactly where the genes are. And we're going to explore that concept. So we're looking at a pair of homologous chromosomes. Now let's just say that the orange chromosome is the paternal chromosome, so it comes from the father. And let's say that the yellow one is the maternal chromosome. And just to remind ourselves, so these are sister chromatids.
Right. Yes, ma'am. Whole thing.
That means that they're identical chromatids. They have identical genes on them.
Right.
So those colored circles that I drew represent just some genes that I randomly picked.
Okay.
And you can see that on the 2 sister chromatids, I drew them in the same color because they're the same. And for that matter, these are also sister chromatids. These 2 yellow chromatids are also sister chromatids. And I'm just going to review a bit to give us some context. So normally in the cell, the paternal chromosome would just look something like that with the centromere middle, and the maternal chromosome would also look something like that. But during meiosis or mitosis, when they replicate, this chromosome will turn into that. So it's going to duplicate itself, and each one of those strands is a sister chromatid. And so will the maternal chromosome turn into something that looks like that. But this is still considered one chromosome, and this is considered one chromosome. And that can be a little bit confusing, because how could this be one chromosome and this also be one chromosome if this X has double the material as the original strand? And the answer to that question is because we count chromosomes by the amount of centromeres that are present. So this has 1 centromere, it's considered 1 chromosome. This X has 1 centromere, so it's considered 1 chromosome. However, they're not quite the same. This on top, it's just 1 chromatid, and the bottom chromosome is made up of 2 chromatids. So try not to get confused. Even though we're calling this 1 chromosome, it's really made up of 2 identical sister chromatids. But anyway, that was a bit of a digression. Let's focus again on our homologous chromosomes. So I picked a couple of genes that I just, put on, and we're going to focus on 3 genes in particular. And to make this a bit more real or relevant, let's just say that the green genes represent, I don't know, maybe complexion, maybe like dark complexion versus a lighter complexion. And on the maternal chromosome, I also drew it in green to remind us that these are homologous chromosomes or homologous alleles. In other words, these are both alleles that code for complexion, but different shades of green because they're probably different versions. So these green genes also code for complexion. And let's say that this darker purple or magenta gene codes for, let's say, hair color. So that means that the lighter purple on the maternal chromosome also code for hair color.
Right.
And then let's say that the blue genes code for eye color. So we have dark blue on the paternal chromosome and light blue on the maternal chromosome.
Sure.
And let's then focus on the sister chromatids where genetic recombination occurs. So these 2 strands are going to swap genetic information between them. Maybe like a chunk on bottom will swap, maybe something in middle will swap, maybe something on top will swap. But actually, genetic recombination also occurs between sister chromatids. However, sister chromatids are identical, so it would be of no consequence. Anyway, so let's look at the 2 chromatids where genetic recombination is happening, the 2 that I circled. Let's take a closer look at those. So here are the 2 chromatids that are going to exchange genetic information and undergo recombination. And I want to ask you a question. So I'm going to give you 2 choices, and I want you to try to figure out which one is more likely. So for the first choice, let's look at the purple and green genes. And the question is, what's more likely? is it more likely that the purple and green genes undergo recombination?
Yes.
And when I say that, I mean that the genes that are originally on one chromosome separate. So I mean to say that, for example, this purple gene will get separated from that gene, and this lighter purple gene will get separated from that gene. So is it more likely that the purple and green genes recombine?
Recombine.
Or the way I like to view it, gets separated?
It's separated.
Or is it more likely that the blue and purple genes recombine or get separated?
No. It would be one.
And the way to think about this is, well, look at the distance between them. So the distance between the purple and green genes is something like that. So in order for recombination to occur with respect to the purple and green genes, it would have to happen somewhere over here. Or on the other side, it doesn't matter, it doesn't make a difference. So you have this whole distance. Whereas if you wanted recombination to occur with respect to the purple and blue genes, you have a very small area to work with. It would have to happen somewhere here. So somewhere along, this line of the chromosome. So that means that it's much more likely for recombination to happen with respect to the purple and green genes.
Oh, there you go.
That is more likely because you have this entire distance to work with. If these two chromatids swap genetic information anywhere along this stretch of the chromosome, so the green and purple genes will separate or recombine. And the blue and purple genes are less likely to recombine because recombination would have to occur only in this little sliver of chromosome, and that's just smaller than the other part of the chromosome that we were looking at. So we just learned a very important concept, and that is that the further apart two genes are, the more likely it is that they will recombine. I'm actually not sure if that's the proper way to use the word, but I just use it that way. So I'm going to put it in quotes. And again, when I say recombine, I mean that two genes that were originally on the same chromosome get separated. And then the next thing we learned is that the closer two genes are to each other, I'll abbreviate each other just EO, the less likely it is that they will recombine. And now I'm going to introduce you to some terms. The centimorgan is the unit of measurement that we use to measure distance on a chromosome. And another way to say centimorgan is a genetic map unit, or M.U., which stands for map unit. And I'll give you the official definition of a centimorgan because I think it's important. It kind of ties in distance to what it has to do with recombination.
Okay.
And so a centimorgan is the distance between genes, just going to abbreviate between like that, for which one product of meiosis in 100 is recombinant. And to put that in simpler terms, it means that if two genes are 1 centimorgan apart, it means that 1 out of 100,
What did you say?
two genes are 1 centimorgan apart. It means that 1 out of 100 times, or 1% of the time that meiosis happens, those two genes will be recombinant or
Those two genes will get separated.
separate or recombine. And we'll actually do an example of this to illustrate what this means. So here we have our two chromatids again. And let's just say that the distance between the purple and green genes is 25 map units. Remember, a map unit is the same thing as a centimorgan. And
25%.
let's say that the distance between the blue and purple genes is 6 map units. So this is clearly not drawn to scale very well, but it's just an estimate. So let's first focus on the purple and green genes. So if they're 25 map units apart, so remember, if two genes are 1 map unit apart, that means that 1% of the time they'll recombine.
Right.
So these are 25 map units apart. So that means that 25% of the time that meiosis happens, recombination will occur with respect
Which
to
is half,
purple
because
and green
50%
genes.
is already max.
So let's see what that looks like. So we're going to see that in this spot right over here, the chromatids just swap information. So let's draw what that would look like. So let's draw our paternal chromosome, or at least part of it. And then we'll draw our, actually I'm going to draw that a little bit higher because we need more room. So here's our paternal chromosome, and then our maternal chromosome, or chromatid. And then since they kind of swapped in this spot, so I'm going to draw yellow right over here. That's the part that came from the maternal chromosome. And then I'm going to draw orange over here. That's the part from the paternal chromosome. And now let's fill in our genes. So the blue genes will just stay where they were. So we just leave them put. And the same goes for the purple genes. But then we have that piece of maternal chromosome. So we get that hunter green gene over here. And then we get the lime green gene right over here. So this is what I mean by recombination being a separation of genes. So this purple gene and that lime green gene were on the same chromosome before, but they got separated. They're not on the same chromosome anymore. And the same applies to these two genes. Now let's look at the blue and purple genes. So they're 6 map units apart. So that means that 6% of the time that meiosis happens, the purple and blue genes will get separated. So we're going to say that upwards of this spot, the chromatids swap information. So let's draw that. So here we'll draw a part of our paternal. That's the paternal chromosome and the maternal one. And they swapped somewhere like over here. So let's fill that in. So that's the part of the maternal chromosome that lands up, sorry, that's the part of the maternal chromosome that ends up on the paternal one. And
Wait, what? I thought it wasn't going to do
orange,
it.
purple and blue gene, and that end. And then I'm going to draw. And then since they kind of swapped in this spot, so I'm going to draw yellow right over here. That's the part that came from the maternal chromosome. And then I'm going to draw orange over here. That's the part from the paternal chromosome. And now let's fill in our genes. So the blue genes will just stay where they were. So we just leave them put. And the same goes for the purple genes. But then we have that piece of maternal chromosome. So we get that hunter green gene over here. And then we get the lime green gene right over here. So this is what I mean by recombination being a separation of genes. So this purple gene and that lime green gene were on the same chromosome before, but they got separated. They're not on the same chromosome anymore. And the same applies to these two genes. Now let's look at the blue and purple genes. So they're 6 map units apart. So that means that 6% of the time that meiosis happens, the purple and blue genes will get separated. So we're going to say that upwards of this spot, the chromatids swap information. So let's draw that. So here we'll draw a part of our paternal. That's the paternal chromosome and the maternal one. and they swapped somewhere like over here. So let's fill that in. So that's the part of the maternal chromosome that lands up. Sorry, that's the part of the maternal chromosome that ends up on the paternal one, and the orange over here. And now let's fill in our genes. So let's first fill in the ones that just stay put. So we have that lime green gene, then we have that hunter green gene, and our purples also stay put. But the blue genes swap chromosomes. So we have our dark blue gene over here and our light blue gene over here. And again, take note of how they swap places or how they separate. So these two genes were together on the same chromosome before, but not anymore. And the same for these two genes. So let's just tie this back into the bigger concept going on. If we were to do a statistical analysis of how often certain recombinations happen, that can help us map out the genes on a chromosome.
Okay. Oh, wow, that's it. Wow, that was fast.
Genotypes, that right here is going to be white. So you have blood types recessive. So this is also going to be an A, E, homologous pair 2, right? Hair to combination 16 eyes. So brown eyes and big teeth. Eyes, parents for both teeth, brown big teeth. So there's 3 combination teeth that express, that show dominance, but they sort independently because they're on different chromosomes. You could use it, where did I do it over here? You could use it to explore incomplete dominance when there's blending where red and white made pink genes, or you could even use it.
Normally, when we think about DNA, we think about the nucleus of a cell. And that's because a cell's DNA is contained in its nucleus. But there are actually a few exceptions to this general rule. So there are certain organelles that actually have their own DNA. And 2 very famous examples of this are the mitochondria and chloroplasts. So mitochondria and chloroplasts have their own.
What did you say in the meeting?
Normally, when we think about DNA, we think about the nucleus of a cell. And that's because a cell's DNA is contained in its nucleus. But there are actually a few exceptions to this general rule. So there are certain organelles that actually have their own DNA. And 2 very famous examples of this
Mitochondria.
are the mitochondria
And the chloroplasts.
and
Oh.
chloroplasts.
Oh, yeah, chloroplasts.
So mitochondria and chloroplasts have their own DNA. I'm just going to scribble in here in blue. And not only do they have their own DNA, but they can actually replicate their DNA and replicate themselves independently of the nucleus of the cell in which they are. So let's just talk briefly about mitochondria. So mitochondria are these organelles found in eukaryotic cells, and they're sometimes referred to as the powerhouse of the cell because they break down glucose to make this high energy molecule called ATP, and then the cell takes this ATP and uses it for all sorts of cellular processes. And the mitochondrial DNA, written like that, mtDNA, has about 37 genes in it, and found in eukaryotic cells. And they're sometimes referred to as the powerhouse of a cell because they break down glucose to make this high energy molecule called ATP, and then the cell takes this ATP and uses it for all sorts of cellular processes. And the mitochondrial DNA, written like that, mtDNA, has about 37 genes in it. And these genes, most of them have to do with the cellular respiration that's going on in the mitochondria. Let's talk a bit about chloroplasts. So chloroplasts are these organelles that are found in plant cells. They're also found in algae cells. And chloroplasts are the site of photosynthesis. We wanted to be more specific, so you have these stacks called granum. Well, in singular it's granum, plural it's grana, and those granum are made up of these, that's an M over there. And those granum are made up of these little circles called thylakoids. And photosynthesis happens within these thylakoids. So during photosynthesis, sunlight is harnessed, of course, with a bunch of other steps to make glucose. So this is where the concept of making its own food comes from. It's actually making glucose, it's making its own food. And then that glucose goes to the mitochondria of that cell and gets broken down, make ATP, and then the cell uses that ATP for whatever it needs to do. The DNA in chloroplasts, sometimes written cpDNA, has about 100 genes. And these genes also, most of them have to do with proteins or things that are involved in photosynthesis. And the reason that this is interesting is, well, Let's take a look at how sexual reproduction normally takes place. We have an egg cell, and the nucleus of this egg cell has only half the amount of DNA that a normal cell in that organism would have, so we call that N. And then we have a sperm cell. Remember, the sperm cell is really much, much smaller than an egg cell, so this is in no way drawn to scale. And the sperm cell also has in its nucleus only half the amount of DNA that cells in this organism normally have, so that's also N. But then they fuse to make a zygote. And this zygote is 2N. It has the normal amount of DNA that a cell in this organism would have. Half of it comes from the egg cell and half of it comes from the sperm cell. And then this zygote is going to divide into 2 cells, and then those 2 cells, of course, divide further, and so that's also N. Remember, the sperm cell is really much, much smaller than an egg cell, so this is in no way drawn to scale. And the sperm cell also has in its nucleus only half the amount of DNA that cells in this organism normally have, so that's also N. But then they fuse to make a zygote.
That's
And
2N.
this zygote is 2N. It has the normal amount of DNA that a cell in this organism would have. Half of it comes from the egg cell and half of it comes from the sperm cell. And then this zygote is going to divide into 2 cells, and then those 2 cells, of course, divide further. And this goes on until, until they are enough cells to put together an organism. But this egg cell, well, it's a fully developed cell, and it not only has genetic information, but it has organelles in the cytoplasm. So it has these mitochondria in its cytoplasm, and those mitochondria have DNA in it, which I'm just going to scribble some glue inside. And the zygote also has those mitochondria. Because remember, the zygote is, well, practically an egg cell, but with the only difference being that its nucleus has the additional DNA of the sperm cell. And remember, the sperm cell does not donate anything to the egg cell except for half of the DNA in the nucleus. It does not give the egg cell and the zygote anything else. So you have a zygote with those mitochondria, and of course they have their DNA in it. And then when this zygote replicates itself, so it replicates the nucleus, but it also replicates the mitochondria in the cytoplasm. And these cells will, I'm going to skip out the nucleus, I'm just drawing the mitochondria. So they have these mitochondria, but these mitochondria came only from the egg cell. None of those mitochondria came from the sperm cell. And so this brings us to the concept of maternal inheritance. And maternal inheritance, well, it's basically like exactly the way it sounds. It's inheritance that happens only from the maternal end or only from the egg cell. So right here, we're showing that the mitochondria that this organism will eventually have originates from the mitochondria that came only from the egg cell and not from the sperm cell, and therefore it exhibits a maternal inheritance. So both mitochondria and chloroplasts exhibit maternal inheritance because they are in the egg cell that eventually becomes an organism. And maternal inheritance, it's interesting to note, is contrary to Mendelian genetics. So maternal inheritance is contrary to Mendelian genetics because Mendelian genetics assumes that half of the DNA comes from the egg cell, half from the sperm cell. It does not take into account any sort of genetic information that comes from only one of the gametes, for example, just from the egg cell. And in fact, everything we just described here can be referred to as extranuclear inheritance. So extranuclear inheritance would refer to any genes that are passed on from structures that are not in the nucleus. So extranuclear meaning outside of the nucleus. Mitochondria and chloroplasts are outside of the nucleus, so they when they are inherited, we refer to it as extranuclear inheritance. So now that we've introduced extranuclear inheritance, let's actually take a look at one of the early experiments that helped to discover extranuclear inheritance.
I think we'll replay this. I need to go to the bathroom. Come back, we're almost done with it. And then what's next? Oh, then DNA tech is next. Hopefully we can fly through it. I don't know. Maybe. We can try. I mean, we can try. Is this a question as well? Yeah, that's a question. So we only have, that one seems super short. These are just a bunch of videos. Oh, no, wait. Super short. Well, then this one's kind of pretty long. Yeah, we'll see. We'll see. Let's watch this again in 2x
Normally,
speed.
when we think about DNA, we think about the nucleus of a cell. And that's because a cell's DNA is contained in its nucleus. But there are actually a few exceptions to this general rule. So there are certain organelles that actually have their own DNA. And two very famous examples of this are the mitochondria and chloroplasts. So mitochondria and chloroplasts have their own DNA. I'm just going to scribble in here in blue. And not only do they have their own DNA, but they can actually replicate their DNA and replicate themselves independently of the nucleus of the cell in which they are, Na. We think about the nucleus of a cell, and that's because a cell's DNA is contained in its nucleus. But there are actually a few exceptions to this general rule. So there are certain organelles that actually have their own DNA. And two very famous examples of this are the mitochondria and chloroplasts. So mitochondria and chloroplasts have their own DNA. I'm just going to scribble in here in blue. And not only do they have their own DNA, but they can actually replicate their DNA and replicate themselves independently of the nucleus of the cell in which they are. So let's just talk briefly about mitochondria. So mitochondria are these organelles found in eukaryotic cells, and they're sometimes referred to as the powerhouse of the cell because they break down glucose to make this high energy molecule called ATP, and then the cell takes this ATP and uses it for all sorts of cellular processes. And the mitochondrial DNA, written like that, mtDNA, has about 37 genes in it. And these genes, most of them have to do with the cellular respiration that's going on in the mitochondria.
Okay.
Let's talk a bit about chloroplasts. So chloroplasts are these organelles that are found in plant cells. They're also found in algae cells. And chloroplasts are the site of photosynthesis. We want to be more specific, so you have these stacks called granum, well, in singular granum, plural grana, and those granum are made up of these, that's an M over here. And those granum are made up of these little circles called thylakoids. And photosynthesis happens within these thylakoids. So during photosynthesis, sunlight is harnessed, of course, with a bunch of other steps to make glucose. So this is where the concept of making its own food comes from. It's actually making glucose, it's making its own food. And then that glucose goes to the mitochondria of that cell and gets broken down, make ATP, and then the cell uses that ATP for whatever it needs to do. The DNA in chloroplasts, in cpDNA, has about 100 genes. And these genes also, most of them have to do with proteins or things that are involved in photosynthesis. And the reason that this is interesting is, well, let's take a look at how sexual production normally takes place. We have an egg cell, and the nucleus of this egg cell has only half the amount of DNA that a normal cell in that organism would have, so we call that N. And then we have a sperm cell, how sexual production. And all these genes also, most of them have to do with proteins or that cell and gets broken down, make ATP, and then the cell uses that ATP for whatever it needs to do. The DNA in chloroplasts, sometimes written cpDNA, has about 100 genes. And these genes also, most of them have to do with proteins or things that are involved in photosynthesis. And the reason that this is interesting is, well, let's take a look at how sexual production normally takes place. We have an egg cell, and the nucleus of this egg cell has only half the amount of DNA that a normal cell in that organism would have, so we call that N. And then we have a sperm cell. Remember, the sperm cell is really much, much smaller than an egg cell, so this is no way to scale. And the sperm cell also has in its nucleus only half the amount of DNA that cells in this organism normally have, so that's also N. But then they fuse to make a zygote. And this zygote is 2N. It has the normal amount of DNA that a cell in this organism would have. Half of it comes from the egg cell, and half of it comes from the sperm cell. And then this zygote is going to divide into 2 cells, and then those 2 cells, of course, divide further. And this goes on and on until, until there are enough cells to put together an organism. But this egg cell, well, it's a fully developed cell, and it not only has genetic information, but it has organelles in the cytoplasm. So it has these mitochondria in the cytoplasm, and those mitochondria have DNA in it, which I'm just going to scribble some blue inside. And the zygote also has those mitochondria. Because remember, the zygote is, well, practically an egg cell, but with the only difference being that its nucleus has the additional DNA of the sperm cell. And remember, the sperm cell does not donate anything to the egg cell except for half of the DNA in the nucleus. It does not give the egg cell and the zygote anything else. So you have a zygote with those mitochondria, and of course they have their DNA in it. And then when this zygote replicates itself, so it replicates the nucleus, but it also replicates the mitochondria in the cytoplasm. And these cells will, I'm going to say about the nucleus, just from the mitochondria. So you have these mitochondria, but these mitochondria came only from the egg cell. None of those mitochondria came from the sperm cell. And so this brings us to the concept of maternal inheritance. And maternal inheritance, well, it's basically like exactly the way it sounds. It's inheritance that happens only from the maternal line or only from the egg cell. So right here, we're showing that the mitochondria that this organism will eventually have originates from the mitochondria that came only from the egg cell and not from the sperm cell, and therefore exhibits maternal inheritance. So both mitochondria and chloroplasts exhibit maternal inheritance because they are in the egg cell that eventually becomes the organism.
Mm.
And maternal inheritance, it's interesting to note, is contrary to Mendelian genetics. So maternal inheritance is contrary to Mendelian genetics because Mendelian genetics assumes that half of the DNA comes from the egg cell, half from the sperm cell. It does not take into account any sort of genetic information that comes from only one of the gametes, for example, just from the egg cell. And in fact, everything we just described here can be referred to as extranuclear inheritance. So extranuclear inheritance would refer to any genes that are passed on from structures that are not in the nucleus. So extranuclear meaning outside of the nucleus. Mitochondria and chloroplasts are outside of the nucleus, so they when they are inherited, we refer to it as extranuclear inheritance. There was a scientist by the name of Carl Correns, and he was somewhat of a contemporary of Gregor Mendel. He lived from 1864 to 1933, and Gregor Mendel lived from 1822 to 1884. So they were contemporaries, but Correns was younger than Mendel, and we will soon see that Carl Correns helped discover some things that do not fall into the category of Mendelian genetics. So Carl Correns did a lot of experiments with this plant called the 4 o'clock plant. That's how we know it, which is what you're looking at. I know it looks a little bit like a tree, but it's supposed to be a plant. And actually, the scientific name for the 4 o'clock plant is Mirabilis jalapa. Not exactly sure how to pronounce that. But anyway, so he did a lot of experiments with the 4 o'clock plant. And one very interesting thing about this plant is that you can have within the same plant leaves that
I have to
are.
look at them. I have to look at how this plant looks like. 4 o'clock. Oh, I mean, it doesn't really. Oh, okay, sure. Oh, how does a pea plant look like? Oh, okay. I mean, I guess. Sure. Go back.
A lot of different colors. So let's say that this is the main branch of the plant. And we're going to say that the leaves that come off anywhere on this branch are going to be white. So there are our white leaves. And we're going to say that any leaves that come off of this branch are going to be green. So those are our green leaves. And then the leaves that come off of this branch are variegated. And that means that they have a pattern of green and white mixed. So there are our variegated leaves. And why is it that there are all these different colors within the plant? So let's take a look at some of the cells. Let's take a look first at a cell that's from a leaf that's green. So this is in a leaf that's green, so that's our cell. Here's our nucleus. And in case you're wondering why I'm drawing the cell as a square, it's because plant cells have this cell wall that give it a more rigid shape and make it a bit more squarish, closer to a square than a circle. But anyway, that's our cell. And this cell will have chloroplasts in it, which I'm drawing as these green little circles. And remember, chloroplasts have their own DNA, sometimes referred to as cpDNA. And chloroplast DNA has in it basically the stuff that the chloroplast needs to carry out photosynthesis. And one of the genes in the chloroplast DNA is a gene that makes chlorophyll, which is a pigment that's involved in the photosynthesis. And chlorophyll is what makes the leaf green. It's a pigment that turns the leaf green, or if you want to be more specific, chlorophyll absorbs all of the colors in sunlight, except for green, so green is really reflected. But anyway, the point is that chlorophyll makes the leaf green. So now let's take a look at a cell that comes from a white leaf. So this cell comes from a white leaf. So again, we'll draw our nucleus, and then it has chloroplasts. But the chloroplasts in this cell, the DNA in those chloroplasts have a mutation. So the cpDNA has a mutation that does not allow it to produce chlorophyll, or allows it to produce only a very, very tiny amount of chlorophyll because it needs chlorophyll to survive, but it's not enough that the leaf will be green. So I'm just going to write very, very tiny amount of chlorophyll, and that's why it appears white. And then the variegated leaves, well, they have some cells that have regular chloroplasts that make chlorophyll, and then they have some cells with the mutated chloroplasts, or chloroplasts with mutated DNA that do not make chlorophyll, or rather make a very small amount. And then they actually have a third type of cell, which, maybe you're guessing it already, have both types of chloroplasts. They have chloroplasts that do make chlorophyll, and then they have chloroplasts that have mutated DNA and do not make much chlorophyll.
That's the one you just said is very good?
And what Correns noticed when he crossed a whole bunch of these plants together, he noticed that the progeny had nothing to do with the sperm cell or the pollen cell. It had only to do with the egg cell. So wherever he would take the seed from, the seed is where the egg cell is located. If he took it from a branch that only had white leaves, all the progeny had only white leaves. It didn't matter what the pollen cell was. And the same if he took a seed from a branch that had only green leaves, so all the progeny had all green leaves, no matter where the pollen cell came from. And this is because the trait that we're looking at, the color of the leaf, well, that's determined by the DNA in the chloroplast, and the chloroplast exhibits maternal inheritance. it is going to be inherited only through the egg cell, through the maternal line, or another way to say this, it exhibits extranuclear inheritance. Because a chloroplast has DNA that's outside of the nucleus. Let's take a closer look at what Carl Correns did. So we have this chart to help us out. In the first column, we have the egg cell of the female, that's the seed. And then we're going to cross it with what we have in the second column, which is the pollen cell, that's the male gamete. And they all look the same because it doesn't make any difference in our case. And then we have in the third column our zygote, or the result. So let's look at our first row. So we have this egg cell that came from
White
a
leaves?
branch that had leaves that were only white. So it came from a flower plant with only white leaves.
Okay.
And when we cross it with a pollen cell, no matter what that pollen cell is, no matter where it came from, we always get the same result. We will always get a plant with only white leaves.
Okay.
Let's look at our second row. So we have this egg cell that came from a branch that had only green leaves. And again, no matter what we cross it with, no matter where the pollen cell came from, a white leaf, a variegated leaf, a green leaf, or a flower of a green leaf, so we always get the same result. We get a plant that has only green leaves.
Sure.
It gets a little bit more interesting when we look at the egg cells from the variegated parts of the plant, because there are three different types of egg cells we could have. We could have one that resembles the cell you see if it came from a flower with only white leaves.
Go back.
At the egg cells from the variegated
back.
leaf, a variegated leaf, a green leaf, or a flower of a green leaf. So we always get the same result. We get a plant that has only green leaves. It gets a little bit more interesting when we look at the egg cells from the variegated parts of the plant, because there are three different types of egg cells we could have. We could have one that resembles the cell you see if it came from a flower with only white leaves. Then we have another egg cell that looks like that. It looks like the egg cell you'd find in a plant that had only green leaves. And then we have this third type of interesting cell that has a combination of the quote-unquote normal chloroplasts that are green, and then it has some chloroplasts that have that mutation that don't allow it to make chlorophyll or make a very small amount of chlorophyll.
Sure.
So it's kind of mixed. Anyway, let's look at egg type 1. We cross it with a pollen cell. We always get the same result. We get a plant with only white leaves. And then we look at egg cell type 2. Whatever we cross it with, we get only green leaves. And then if the egg cell type 3, so the zygote, of course, will have both types of chloroplast. But remember, this zygote is going to divide further. And it's going to divide into, if it divides randomly, it'll divide into three different types of cells. Some of the cells will look like this, with the chloroplasts with the mutated DNA. Some of them are going to look like that with the regular chloroplasts, and then some of them are going to be mixed. And this will give you a variegated plant. Some of the leaves are going to have that mixed pattern. You might have some leaves that are white, you might have some leaves that are green. And so the bottom line take-home message is, as I explained before, because the particular trait we're looking at, leaf color, because the gene for that trait is in the chloroplast, it exhibits maternal inheritance. Maternal inheritance is a type of extranuclear inheritance. I'm just going to write that in parentheses.
Oh, okay.
Because this inheritance has to do with DNA that's outside of the nucleus. But anyway, this exhibits maternal inheritance because it has nothing to do, this particular trait is not passed down through the male at all. It's only passed down through the female.
To the mama.
That is, as we explained before, because the chloroplasts are coming only from the egg cell. The sperm cell does not contribute any chloroplast to the zygote. It only contributes DNA that's in the nucleus. So therefore, the leaf color in this chloroplast plant exhibits maternal inheritance. And the same concept would apply to the mitochondria. So we explained mitochondria also has its own DNA. And so if a person were to have a disease that had to do with the DNA inside of the mitochondria,
It's going to be passed down from the mama.
we would know that that person got it from his or her mother and not from his or her father, because the mitochondria also exhibits maternal inheritance. There's one more thing about extranuclear inheritance that I want to mention, and that is, why is it that mitochondria and chloroplasts have their own DNA? Is there something that can explain that? And there is the symbiotic theory seeks to explain why mitochondria and chloroplasts have their own DNA. And this theory tells us that mitochondria and chloroplasts were once independent prokaryotes. So they lived independently. So of course, if they're independent, they need to have their own DNA.
Right.
But eventually, they joined what I'm going to call
The
an
ancestral cell.
ancestral eukaryotic cell. And in case you're wondering, like when this happened, we'll say about 1.5 billion years ago.
Long time ago. Long time ago.
So what happened is that the mitochondria and chloroplasts joined an ancestral eukaryotic cell, and I'm going to call it ancestral because it's not exactly a eukaryotic cell that we'd see today, but it's a cell that would eventually become a eukaryotic cell. We could also call it a host cell because it's going to host mitochondria and chloroplasts. So let's put them inside. So now they're living in this host cell. And why do you think they would want to do this? So they're going to live together in symbiosis. And symbiosis is when organisms live together and each one kind of gives the other something and everybody gains something. So an example of this that you might have heard of is in our intestine, in our gut, we have bacteria E. coli. And at first glance, that might seem like it's not such a good thing, but it's actually a really good thing because we give the E. coli a warm and cozy place to live. They get some nutrients from us, and in exchange, they make for us vitamin K, which is something very useful for us. So that's an example of symbiosis. Each one of the organisms kind of tries to give something, and everybody's happy. So what's going on in the host cell? So the host cell gives the mitochondria and chloroplasts a nice place to live and gives them nutrients. And in exchange, the chloroplast makes glucose through photosynthesis. And then the mitochondria takes that glucose and produces ATP. And then that ATP is used as energy. The mitochondria uses some of the ATP. The chloroplast uses some of the ATP. And of course, the host cell uses some of the ATP. So everybody's happy. Everybody's getting something. And then eventually, this cell, evolved in many different ways. And of course, it became the eukaryotic cell that we know of as today. But not exactly in the way it was because chloroplasts are not found in all eukaryotic cells. It's basically found in plants as an algae, but that's the basic idea. So let's go back to the term endosymbiotic theory. So endo just means inside because the mitochondria and chloroplasts began to live inside a host cell. And symbiotic simply refers to symbiosis.
Reminds you of Groot or
Just
something.
arrow down here refers to symbiosis because each part, the mitochondria, chloroplast, and host cell, everybody's giving something. And so this would explain why mitochondria and chloroplasts have their own DNA because once upon a long time ago, they were independent prokaryotes and they lived on their own.
All right. Well, thank you. Thank you kindly. 8 questions, buddy. Penetrance. Where's penetrance? X-linked coming from the mama. Well, not necessarily, but X-linked recessive trait. 5%, the mother who's a carrier and the father who's unaffected. It doesn't really matter because it's independent. We'll say X-linked, I don't know, we'll say color blindness will stand for CP. I don't know, let's just put E. X-linked recessive. So the mother's a carrier, so she was XXB. Father is unaffected, so he's XY. So we have X, XB, X1. But it's recessive. XC is recessive. The male? Well, if they're both male, then recessive trait, who's a carrier and father who's unaffected. The heck? Dude. It's XB. Oh, but then this becomes a dominant because it can't become recessive. There's no other trait for it to dominate. I mean, there's no other dominant trait for it to dominate, so it's just recessive. Recessive will show, so that's 1/4. Huh? 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 Oh, but there's 2 males here. Oh, it's 2 males. Is what is 1/16? No, actually, it's 1/4 times 1/4 because will be both male and colorblind. So we have to multiply those probabilities. I am independent, and multiply. Anyways, try it this way. There's a 50-50 chance 2 males is 25%. Each male receives only 1. Well, yeah, 25%. Sure. I mean, I got 1/4 times 1/4 is 1/16. But okay, buddy. In what order are they found? Well, A to D is 8%. A to D is 8%. B to C is 8%. A to D is 8%. Okay, so I think A to D are going to be pretty kind of far apart. B to C are going to be far apart as well. the same distance as A to C is, which is 2 units after, and B to C and B to C. So this makes sense. And then what happens? And then C to D are 3%. A to B is 3%. No, this doesn't make sense. I think it really is just A to D, A to D. What? D, A to D. C to A is 5%. And B to C, and then C to D. Maybe C or A. I mean, it's like C might be like in the middle, so right, A to C, A to D, A to D, A to D. If A to D is 2 letters, that means B to C must also be 2 letters, but that's not true. Here. And B to C here is true, B to C here is true, but A to D is not true. So it's either C or A, and I'm thinking since A or B are close, A and B are close, it has to be this one. Decab. Let's go! Let's go. Let's go. What is the genotype of the parent minds? Every mice. When the litter was born, every mice had black eyes, and half had brown fur. Every mice had black eyes, and only half of them had brown fur. Or every mouse had black eyes. Well, let's just see. With RRBB. We are. So in this one we have BR BR black eyes, brown fur. We want black eyes, all brown fur. No. So it can't be it can't be B, E, E, or it's not A. And then R, B, B. It's the same thing, isn't it? So I don't think it can be B either. I don't know. I'm just trying to trick to this one. Let me see if what I'm doing if it makes sense. No, not this one either. I'm gonna get half. E R E R E R. This one makes sense. I guess not. Is this what I did? I don't know. Unless we sort of trick. Maybe C. What the heck? Maybe B. What's the trick here? No minus is BB. None of the offspring inherited BB. If you get BB, you get a 1. 25% would have bright eyes. So one parent is BB. And the other could be either BB or BB. Little b. Big B is mommy. This another one has that BB capital. Which leaves, well, leaves B, C, and D. But then for brown, the only way to get RR is from R times R. Count 1 is RBB. Count 2, RRBB. That's not even true. What the heck? Ah. Oh my goodness, this was wrong. All the others from black eyes. Half are brown, half are fur. Oh, okay, so from this we can eliminate BB together. So eliminate D, ABC. At least one parent. We had 25% would have brown eyes. So one parent must be homozygous. Oh, sorry, one parent. Yeah, one parent must be homozygous, which leaves this one here. At least one parent. At least one parent must be, which leaves B and C. B and C. Okay. Then we do the fur color. One parent is heterozygous. The other must be homozygous. The other must be homozygous precisely. So that's 50% brown, 50% brown. Oh, okay. So at least with this one, we'll just be. Gotcha. Okay. Oh, what happened? Recombination event. What the heck? What are we talking about now? Oh, what happened? Two strand single. Two strand single. Three strand single. Two strand single. The number of crossover events. 1, 2, 3. Three strand double crossover. Only two strand revolving crossover at a time. Three strand double crossover. Three strands double crossover. I'm confused about this one. Quite big. I'm not on the right one. Uh-oh. Uh-oh. Okay, maybe that's why. Whoops. Yeah, maybe we can. I was supposed to ask on this one. Oh well. Come on, buddy. Refresh. Double crossover. Three strand double crossover. Double crossover. Is that what it is? Okay, I think so. I guess. Not to come with this is not true. Before synapses. That's not true. What do you think? Okay. What's over here? The effect on the phenotype? Probably. No effect. No effect. Mm. Mm. I need to delete it. I should delete it. Maybe deletion? Maybe makes sense. Yeah. Sure. Five questions. P. Who is Sandy? Oh my god. Sandy and Danny. I'm gonna say this is Sandy's family. What are we talking about here, huh? Dude. Because I gotta figure out. Let me try to figure this out. both her brothers are very tall and each had so I think Sandy is 6. I think. So circle is Sandy, which is female, and square is what's her name? What's his name? Danny, and he's the male. Because both her brothers and each had it, so I'm thinking this. And then let's see if I'm correct. Her mother, uh, her mother was tall, though she had no. Her mother was tall and had long fingers. Okay. And her father? That would correlate. Sandy's sister had a perfectly healthy son. Okay, yeah. Okay, Sandy is 6. Yeah. Sister to sister, let's go. What are we talking about here? What type of disease is Marfan syndrome? What type of disease is it? Male, daughter, male, daughter. Four children affected the male. Well, let me see. It looks like the female did not have it. The male had it. I'm gonna say it's most likely, since it's passed down from mama to son, I'm gonna say it's on the X. So let's say the father was the carrier of the Marfans. We got X, X, M, X, Y, X, N, X, X, Y, which would make sense why it only affected the daughter here. Sure. Then the daughter was the carrier, so she was XMX. She had XY, which was XM. X, 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 and Y, carrier male, or actually not carrier male. Right? It's then they gave it to the males. And then he gave it to the daughter. And how did he give it to the daughter? Oh, right. Okay. Yeah, then he gave it to the daughter. Sure. So looks like it's Definitely X-linked. Recessive? I don't think it's recessive though. Is it recessive? No, it's not recessive. I think it's autosomal dominant. Autosomal dominant. I think it's dominant. Let's go. Come on. What are you talking about? Incomplete dominance. Incomplete dominance? I think it was an incomplete dominance. Only what? Two dominant alleles for a full. Two dominant and two needs two dominant alleles. That's not true. I'm gonna say B. I'm gonna say, is it incomplete? Needs two dominant alleles. That's not true. I'm gonna say D then. What the heck? Is it C? No. Oh, it's A then. Okay. Well, yeah. Sure. I have a 50% chance as well. No? Penetrance? Oh, penetrance. Well, yeah, I mean, I'm having the next child. Oh, okay. Tight joints, right? I said it loosens. Regain their shape. So I'll say tight joints. Yeah, okay. Yeah, okay, buddy. Anyways, let's continue. What's the next one? DNA tech. Wow, DNA tech has a lot, huh? And this one has kind of a lot as well. This one's pretty short. Alright, let's get on with it. Get on with it.
And that we use to define One criterion that we use to define biological life is that the organism must have the ability to reproduce. And the parent organism must have the ability to pass on to its offspring some sort of material that contains the instructions for living. And we call this material genetic material.
Right.
What is genetic material? Well, today we know that it's DNA, or deoxyribonucleic acid, but this wasn't always so obvious. We're going to go through a couple of earlier.
Maybe I'll just 2x it all the videos and see if that speeds things up.
Experiments that help prove to us that DNA is genetic material. The first experiment we're going to discuss was one that was conducted by a scientist by the name of Friedrich Miescher. And Miescher lived from 1844 to...
So we run through it, and then I'll do the questions for all of them last, because that's the part that takes me the most time, because I'm actually trying to do it. And because we also have a couple of articles, we have to read for each one. So there's quite a little bit. There's quite a lot of articles, to be honest. Even in in this one there's one, but yeah, I think it's best to just 2x it. See if I can make it, because I need to hit 4K.
To 1895. And Miescher worked with cells that he took out of pus. And today we know that these are lymphocytes. And lymphocytes happen to have very large nuclei. So if that's the cell, the nucleus might look something like that. And of course, there's this material inside the nucleus. And because the nuclei are so large, Miescher was able to isolate the material inside the nucleus. And he analyzed this material and came to the conclusion that it was made up of two components. The first was protein. And the second was this substance that acted like an acid. And so we called it nucleic acid. Nucleic because it came from the nucleus. And so Miescher was the first one to isolate and identify nucleic acid. The next experiment we're going to talk about was one conducted by a scientist who lived at about the same time. His name was Wilhelm Roux. And he lived from 1850 to 1924. And Roux worked with cells that were divided. And he analyzed these cells. So I drew a cell here, and I drew a couple of organelles also.
4 years, huh?
Lived from 1850 to 1924. And Roux worked with cells that were divided. And he analyzed these cells. So I drew a cell here, and I drew a couple of organelles also. Let's just label them. We have the endoplasmic reticulum here. We have a couple of mitochondria. In singular, it's mitochondrion. And then we have a couple of ribosomes. I'm just going to abbreviate it like that. And I also drew some vesicles. And of course, in the center, we have the nucleus. And we're going to say that this cell is undergoing mitosis.
What's the next thing? I don't know how this would have to do with DNA technology. I think it's fine to do DNA technology now. Actually, we kind of skipped DNA tech. That's fine. That's okay.
Mitosis right now, so I'm going to draw the chromosomes like that in the center. I'm going to draw 4 chromosomes, and I'm also going to draw the mitotic spindle, which will help to separate the chromosomes into the 2 cells that are being formed. And what Roux noticed was that when the cell divided, so the organelles were not necessarily distributed in a very organized and even fashion. You can see the cell on the left got 3 mitochondria, the cell on the right only got 2, the cell on the left got 3 ribosomes, the one right only got...
Hold on, lady.
Going mitosis right now, so I'm going to draw the chromosomes like that in the center. I'm going to draw 4 chromosomes, and I'm also going to draw the mitotic spindle, which will help to separate the chromosomes into the 2 cells that are being formed. And what Roux noticed was that when the cell divided, so the organelles were not necessarily distributed in a very organized and even fashion. You can see the cell on the left got 3 mitochondria, the cell on the right only got 2, the cell on the left got 3 ribosomes, the one right only got 2. But he noticed that the material in the nucleus divided in an extremely organized fashion. So each nucleus got exactly 4 chromosomes. And so Roux concluded that because the material in the nucleus was divided in a very orderly and even fashion, that must be the genetic material. And this makes sense because the genetic material or instructions for living is the most important thing that the parent cell has to pass on to the daughter cell. If one of the daughter cells didn't get enough mitochondria, well, that's not so terrible. As long as it has the correct instructions for living, it can make more mitochondria. If one of the cells accidentally didn't get a ribosome, well, if it has the right instructions for living, it can make ribosomes. So the conclusion here was that genetic material is made up of protein and or nucleic acid. This was the conclusion that Friedrich Miescher's experiment and Roux's experiment helped prove.
How will we do it?
One criterion that we use to define biological life is that the organism must have the ability to reproduce. And the parent organism must have the ability to pass on to its offspring some sort of material that contains the instructions for living. And we call this material genetic material. What is genetic material? Well, today we know that it's DNA, or deoxyribonucleic acid, but this wasn't always so obvious. We're going to go through a couple of earlier experiments that help prove to us that DNA is genetic material. The first experiment we're going to discuss was one that was conducted by a scientist by the name of Friedrich Miescher. And Miescher lived from 1844 to 1895. And Miescher worked with cells that he took out of pus. And today we know that these are lymphocytes. And lymphocytes happen to have very large nuclei. So if that's the cell, the nucleus might look something like that. And of course, there's this material inside the nucleus. And because the nuclei are so large, Miescher was able to isolate the material inside the nucleus. And he analyzed this material and came to the conclusion that it was made up of two components. The first was protein, and the second was this substance that acted like an acid, and so we called it nucleic acid. Nucleic because it came from the nucleus. And so Miescher was the first one to isolate and identify nucleic acid. The next experiment we're going to talk about was one conducted by a scientist who lived at about the same time. His name was Wilhelm Roux, and he lived from 1850 to 1924. And the second was this substance that acted like an acid, and so we called it nucleic acid. And he lived from 1850 to 1924. And Roux worked with cells that were divided, and he analyzed these cells. So I drew a cell here, and I drew a couple of organelles also. Let's just label them. We have chromosomes into not necessarily distributed in a very organized and extremely organized fashion. Genetic material or instructions for.
I get it, so that's fine.
We spoke about the experiments of Friedrich Miescher and Wilhelm Roux, and how their experiments helped show that genetic material is made up of either protein or nucleic acid, or perhaps both. So the next logical question to ask is, well, which one is it? Is it protein, is it nucleic acid, or perhaps it's both? So this question was answered with the experiment of two scientists, Alfred Hershey and Martha Chase. And in their famous Hershey-Chase experiment that was published in 1952, they showed that it's nucleic acid that's genetic material and not protein. So Hershey and Chase worked with bacteriophages. And a bacteriophage is a virus that specifically infects bacterial cells, and it can also be referred to simply as a phage. So what is a bacteriophage? Well, it has nucleic acid, which can be either DNA or RNA. In this video, I'm just going to refer to all nucleic acid as DNA, but keep in mind that when I say that, it can also mean RNA, because in some viruses, nucleic acid is RNA. And that nucleic acid, or DNA, is surrounded by a protein coat. And how do bacteriophages infect bacterial cells? Well, they get kind of close to the bacterial cell and sort of sit on top of it and inject their DNA into the bacterial cell. Then their DNA gets integrated into the bacterial cell's DNA, which I'm going to draw right here. So here's the bacterial cell's DNA. So the virus's DNA gets somehow integrated, and now this bacterial cell is going to produce a whole bunch of viruses. So this is some background information. Now let's talk about Hershey and Chase's actual experiment. Hershey and Chase took some phages, and they put these phages in a medium, which means a broth that has a lot of nutrients, so that these phages can now multiply and reproduce and make a lot more of themselves. But they wanted to label the protein coat of the new generation of viruses. And how are they going to do that? So they made sure that all the amino acids that were in this broth had in them radioactive sulfur. So they were labeled with sulfur 35, which is one of the radioactive isotopes of sulfur. And in this way, they can kind of keep track of where the protein is going and what's happening with the protein. And the reason that they chose sulfur for this part of the experiment is because they wanted to label the protein in particular. And sulfur is found in amino acids, which means they're found in proteins, but sulfur is not found in DNA. So this is a good way to make sure that they label the protein, but not to label the protein in particular. And sulfur is found in amino acids, which means they're found in proteins, but sulfur is not found in DNA. So this is a good way to make sure that they label the protein, but not the DNA. So now when this phage, or the phages that are put into the medium, reproduce, they're going to take nutrients from this broth, and among those nutrients are amino acids, and they're going to incorporate those labeled amino acids into their protein coats. So they produced a generation of viruses that have radioactively labeled protein coats. And now they allow this generation of viruses to infect a bacterial cell. So you can see they climb on top of the bacterial cell and inject their DNA into the cell. So remember, their DNA gets incorporated into the DNA of the bacterial cell, and the bacterial cell will produce a whole bunch of viruses. Now take note that the original protein coats, of course, remain outside of the cell. And Hershey and Chase now wanted to separate the protein coats from the bacterial cells. So they
The protein coat from the bacterial cell.
centrifuge this mixture to kind of get rid of the protein coats. So let's get rid of them. And notice how the phages inside this bacterial cell, they just have a regular protein coat that's not radioactively labeled with S35. That's why they're drawn in green. And so now what they did was they took the bacterial cells, lysed them, which means they made them burst, and they analyzed the viruses. And they saw that the viruses were not at all radioactively labeled. So they concluded that the protein coat must have remained outside the cell, outside the bacterial cell. And if the protein coat remained outside of the bacterial cell, then it must be that that is not genetic material, because in order for this bacterial cell to produce viruses, it had to have contained the genetic material of the virus. And if the protein coat remained outside, it must be that that is not the genetic material. Let's talk about the second part of Hershey and Chase's experiment. So again, they took a phage or a couple of phages and put it in a medium with a lot of nutrients so that it can reproduce and make lots of viruses. But this time, they wanted to label not the protein coat, but the nucleic acid inside. And again, I'm just going to refer to nucleic acid as DNA, but keep in mind that it can also be RNA. So they want to label the DNA. And how are they going to do that? So they made sure that all of the nucleotides that were in the broth, and the viruses are going to need nucleotides to make their DNA, so all of them were radioactively labeled with phosphorus 32. So P32 is radioactive isotope of phosphorus, and this is how they're going to label the DNA. And the reason they chose phosphorus 32 is because phosphorus is found in DNA nucleotides, but it is not found in amino acids, so it will not get integrated into the protein coat. So it's a great way to differentiate between the two. So they put them in the broth and allow them to reproduce, and they produce a generation of viruses that have this radioactively labeled DNA, which I drew in that magenta. And of course, the protein coat is in green because it's not labeled in any way. So they allow these viruses, these phages, to infect the bacterial cell. The protein coat remains outside, and it injects the DNA into the bacterial cell. And the bacterial cell is going to produce a whole bunch of phages, and the protein coats, of course, remain outside. And now they're going to centrifuge this mixture of protein coats and bacterial cells because they want to get rid of the protein coats. So they spin it in a centrifuge, and the protein coats, which are less dense, will end up in the supernatant, and the bacterial cells, which are heavier, end up in the pellet. So we're going to get rid, sorry about that. So we're going to get rid of these protein coats. The protein coats. So is going to produce a whole bunch of phages, and the protein coats, of course, remain outside. And now they're going to centrifuge this mixture of protein coats and bacterial cells because they want to get rid of the protein coats. So they spin it in a centrifuge, and the protein coats, which are less dense,
Less dense,
will end up in the supernatant,
supernatant,
and the bacterial cells, which are heavier, end up in the pellet.
and the bacteria what?
Centrifuge and the protein coats, which are less dense, will end up in the supernatant, and the bacterial cells, which are heavier,
Bacteria cells. Oh, okay.
end up in the pellet. So we're gonna get rid. Sorry about that. So we're gonna get rid of these protein coats. Now, Hershey and Chase lyse these bacterial cells and make them burst, and they analyze the viruses inside. And the viruses have a lot of nucleotides with this P32, and so there's a lot of radioactively labeled DNA. Maybe not all the DNA have radioactively labeled phosphorus because some of the nutrients came from the bacterial cell, but a fair number of the viruses had radioactively labeled DNA inside of them. And so they concluded that since the DNA entered the cell, it must be DNA that's genetic material. Or, should really be more specific, it's nucleic acid. That's genetic material. In order for the bacterial cell to produce viruses, it had to have inside the genetic material. And since there's this radioactively labeled DNA within the viruses, they concluded that nucleic acid is genetic material. We mentioned that Hershey and Chase published the findings of their experiment in 1952, and a very, very short while later, in 1953, James Watson and Francis Crick published their famous paper in which they actually identified the structure of DNA or nucleic acids. So they put together a tremendous amount of research that was happening during their time and before their time, and they identified the structure of DNA, and they told us that DNA is a double-stranded helix with a sugar phosphate backbone, the sugar in this case being deoxyribose. And then what you see on the inside, kind of what looks like the rungs of a ladder, those are nitrogen bases. And there are 4 of them: adenine, thymine, guanine, and cytosine. And adenine and thymine pair up with each other, guanine and cytosine pair up with each other. So let's just recap the 4 experiments that we discussed. We spoke about Friedrich Miescher, and Miescher was the first one to isolate and identify nucleic acids. Then we spoke about Wilhelm Roux, and Roux's experiments helped show that it was the material in the nucleus that was genetic material. But still at that point, people weren't quite sure.
Oh, what was it, the guy's experiment? Jesus, I'm just going through it a little bit fast. I just want to make sure.
Was divided in a very orderly and even fashion.
Oh, Rue. So you said it's in the nucleus. This guy said it's nucleic acid.
Miescher was the first one to isolate and identify nucleic acid.
Isolate and identify nucleic acid.
And so Roux concludes because the genetic material or instructions for living is and so that it was made up of 2 components. The first was protein. If one of the daughter cells didn't get enough as the right instructions for living of protein and or nucleic acid. This was the conclusion that it's on the left got from if one
Okay.
or if it's protein or DNA, and many people thought it was protein.
Rue says,
The cells, ribosomes, is or this was the conclusion because proteins are more complex than nucleic acid. Then we spoke about Hershey and Chase and how they helped prove that it was nucleic acid that's genetic material and not protein. And then Watson and Crick. And Miescher was genetic or DNA, and many people thought it was protein because proteins are more complex than nucleic acids. Then we spoke about Hershey and Chase and how they helped prove that it was nucleic acid that's genetic material and not protein. And then Watson and Crick actually identified the structure of nucleic acids.
Let's say
we got Sal again. I like the girl better. That's fine. Let's go bathroom real quick. We might make it though. I mean, if I 2x it, I don't know. All right, let's go, Sal. I'm gonna 2x you though.
That you have some vials here, and you know that in the solution you have fragments.
Too loud.
And what you're curious about, well, let's say that you have some vials here, and you know that in the solution you have fragments of DNA in each of these. And what you're curious about, well, what about the DNA fragments in this first vial, vial number 1? How long are those fragments? How many base pairs? How long are they? Well, you might say, well, why don't I just take them out and count them? Except for the fact that they're incredibly small and incredibly hard to handle. Even a fairly large fragment of DNA, let's say we're talking about something that's on the order of 5,000 base pairs, well, that's going to be approximately 1 to 2 micrometers long. If you were to completely stretch it out, and we can't even start to think about how thin the actual diameter is. We just lengthwise the long way, it's only going to be 1 to 2 micrometers, which is super duper small. This is 1 to 2 thousandths of a millimeter. So that's not going to help us to somehow try to manipulate it physically with our hands or with kind of rough tools. So how do we do that? And we can have other vials there. How do we see how long the DNA strands that are sitting in those vials actually are? And the technique we're going to use, gel electrophoresis, it actually could be used for DNA strands, could be used for RNA, could also be used for proteins, any of these macromolecules, to see how long are those fragments. And so let me write this down. Gel electrophoresis. Electrophoresis. And it's called gel electrophoresis because it involves a gel, it involves electric charge, and phoresis is just referring to the fact that we're going to cause the DNA fragments to migrate through a gel because of the charge. So phoresis is referring to the migration or the movement of the actual DNA. So how do we do this? Well, here's our setup right over here. We have our gel that's inside of that's embedded in a buffer solution. So this gel, the most typical one is agarose gel, that's a polysaccharide that we get from seaweed, and it's literally a gel. It's a gelatinous material. And what we're going to do is we're going to take samples. So we might take a little sample from this one right over here, and we'll put it in this well right here. And you can do these wells as little divots in the gel. You can take a little sample from here and put it into this well, and then you can put a sample from here and you can put it in that well. And it's going to be made inside this buffer. So you can see the buffer right in this fluid. And that's really just water with some salt in it. And the buffer is going to keep the pH from going too far out of bounds as we place a charge across this entire thing, because if the pH gets too far in the basic or acidic side, it might actually affect the DNA or affect the charge on the DNA. And what we're going to do is we're going to put a charge across this whole setup, where the side where the wells are, where we're going to place the DNA, that's going to be where we're going to put the negative electrode. So that's our negative electrode there. And the other end is going to be our positive electrode. And we're going to use the fact that DNA has a negative charge at the typical pHs, or the pHs that we are going to be dealing with. And we can go back into previous videos, and we can see it right over here. You see these negative charges on our phosphate backbone. And so what is going to happen? What is going to happen once we connect both of these to a power source, and then this side is negative and this side is positive? Well, the DNA is going to want to migrate. Now, let's think about what will happen. Will shorter things migrate further, or will longer things migrate further?
Shorter things.
Well, you might say, well, longer things are going to have more negative charge, so maybe they go farther away. But then you also have to remember that they're also moving more mass. So their charge per mass is going to be the same regardless of length. And so what determines how far something gets, how much it migrates over a certain amount of time, is how small it is. Remember, we have this agarose gel, and people are still studying the exact mechanism of how this DNA or these macromolecules actually migrate through the polysaccharide. But if you imagine this polysaccharide is kind of this mesh, this net, this sieve, well, smaller things are going to be able to go through the gaps easier than the larger things. And so if you let some time pass, some of the DNA, let's say this DNA gets around there, let's say, and I'm just coloring you actually when you see these colors, let's say this DNA gets around that far, so it doesn't get as far. Let's say this DNA gets around there, let's say, and I'm just coloring you actually when you see these colors, let's say this DNA gets around that far, so it doesn't get as far. Let's say that this DNA doesn't migrate. Let's say it has some that migrates that far. Let's say it has some that migrates that far. And so if you just saw this, you wait some amount of time, and you were to come back, and you were to see this migration. You were to see this migration occur. And the longer you wait, the further these things are going to get. In fact, if you wait too long, you're going to fall off all the way over the other edge. Is if you just saw this, you say, okay, well, this strand right over here, these must be smaller DNA molecules. They must be shorter. These must be a little bit longer, and these must be even longer than that. And this grouping right over here is going to be the longest of all. So this was a mixture of some longer strands and still longer ones, but not quite as long. And for example, maybe there are some really short strands. Maybe there are some really short strands in that what I'm drawing as that orange group right here. So what I just did right here, this could tell you the relative length of these strands, but how would you actually measure them? Well, that's where you can go find standardized solutions, which we call a DNA ladder. So let's say you go get the DNA ladder. I'm going to draw it in pink. So you literally can buy this. You can buy it online. And the standard solution, let's say it separates like this. So it separates, that goes there. Let's say some of it goes like there, and some of it goes like there. Well, you would be able to know from the labeling, or whichever one you choose to buy, that this grouping here, this is all of the DNA that is 5,000 base pairs. Let's say this right over here is 1,500 base pairs. And let's say this over here is 500 base pairs long. And so now you can use this DNA ladder, these standardized ones, to gauge how many base pairs these are. So you say, okay, this blue one here, this is a bunch of DNA that's a little bit longer than 500 base pairs, but shorter than 1,500 base pairs. You can see this green one here, well, it's a little bit longer than 1,500 base pairs. It didn't migrate quite as far as this big bundle of 1,500 base pairs that did. And so then you can get a better approximation, and you can choose your ladder based on what you think you are going to find there, what you are actually going to look for. Now, the other thing to appreciate is when you see the DNA having migrated this far, you might say, okay, is this one DNA strand? Is that one DNA strand that I'm looking at? And just going back to the measurements, no, that is many DNAs that you're looking at. They're not all stretched out like that. Remember, even something that is 5,000 base pairs long is only going to be 1 to 2 micrometers if you stretch it out. So you wouldn't even be able to see it. It's a thousandth of a millimeter. You wouldn't even be able to see it. So this is many molecules of DNA is migrating that far. They would have to be that small to be able to migrate through that polysaccharide gel. Now, the last thing you're probably saying is, okay, wait, but how am I even seeing it over here? How do I actually see this DNA, especially if they're these super small molecules? And the answer is you put some type of marker on the DNA that will make them visible, some type of dye or something that might become fluorescent. And one of the typical things that people often use is ethidium bromide. And ethidium bromide is called an intercalating agent. And it's a molecule. You can see the ethidium right over here. These are two backbones of DNA. You can see the base pairs bonded here. And then this right over here, that is ethidium that has fit itself. That's why we call it intercalating. It has fit itself in between the rungs of the ladder. And when it does so inside of DNA, it actually becomes fluorescent when you apply UV light to it. So if you put this ethidium bromide into all of your DNA right over here, and then as it migrates, and then if you were trying to turn on a UV light, it would become fluorescent, and you would actually see these things. And so if you wanted to see what it actually would look like in real life, well, this is what it would look like when if you were to look at it straight on, where this would have been a well. Let me make it a bit easier to read. So right over here would have been the well where you would put the DNA ladder, and it would come up with standardized measurements. Maybe that's our 5,000 base pairs. This right over here is our 1,500 base pairs, and this right over here is our 500 base pairs. And then let's say you get some solution of some other DNA, and you wait a little while, and you see, look, it migrated not quite as far as the 500 base pairs. So it must be a little bit. This must be a bundle of things a little bit longer than 500 base pairs, but for sure a lot shorter than 1,500 base pairs. And once again, it doesn't have to have just one fragment length. You could have had another group that was maybe right at 1,500 base pairs. pairs. And you've probably seen this whenever you see people talking about genetic analysis and things like this. You're often seeing people look at one of these readouts from gel electrophoresis. So now you know what's actually going on here. This isn't a strand of DNA. There's a bunch of DNA that has been tagged with some type of a dye or lithium bromide or something like that. And it's a bunch of those molecules, and they've migrated based on the charge. They're trying to get away from that negative charge to the positive charge. And the smaller molecules, this is a bunch of small molecules right over here, are able to get further because they're able to get through the mesh of the agarose gel, creating that far. And they would have to be that small to be able to migrate. And the technique we're going to use, gel electrophoresis, it actually could be used for DNA strands, could be used for RNA, it can also be used for protein. It's embedded in a buffer solution. So this gel, the most typical one is agarose gel. That's a polysaccharide that you get from seaweed. And it's literally a gel. It's a gelatinous material. And what we're going to do is we're going to take samples. So we're going to take a little sample from this one right over here, and we'll put it in this well right here. You can do these wells as little divots in gel. You can take a little sample from here and put it into this well. And then you can put a sample from here, and you can put it in that well. And it's going to be made inside this buffer. So you can see the buffer right here in this fluid. And that's really just water with some salt in it. And the buffer is going to keep the pH from going too far out of bounds as we place a charge across this entire thing. Because if the pH gets too far on the basic or acidic side, it might actually affect the DNA or affect the charge on the DNA. And what we're going to do is we're going to put a charge across this whole setup, where the side where the wells are, where we're going to place the DNA, that's going to be where we're going to put the negative electrode. So that's our negative electrode there. And the other end is going to be our positive electrode. And we're going to use the fact that DNA has a negative charge at the typical pHs, with the pHs that we are going to be dealing with. And we can go back into previous videos, and we can see it right over here. You see these negative charges on our phosphate backbone. And so what is going to happen? What is going to happen once we connect both of these to a power source, and then this side is negative and this side is positive? Well, the DNA is going to who want to migrate. Now let's think about what will happen. Will shorter things migrate further, or will longer things migrate further? Well, you might say, well, longer things are going to have more negative charge, so maybe they go farther away. But then you also have to remember that they're also moving more mass. So their charge per mass is going to be the same regardless of length. And so what determines how far something gets, how much it migrates over a certain amount of time, is how small it is. Remember, we have this agarose gel, and people are still studying the exact mechanism of how this DNA or these macromolecules actually migrate through the polysaccharide. But if you imagine this polysaccharide is kind of this mesh, this net, this sieve, well, smaller things are going to be able to go through the gaps easier than the larger things. And so if you let some time pass, some of the DNA, let's say this DNA gets around there, let's say, and I'm just color you what you when you see these colors, let's say this DNA gets around that far, so it doesn't get as far. Let's say that this DNA doesn't migrate. Let's say it has some that migrates that far. Let's say it has some that migrates that far. And so if you just saw this, you wait some amount of time, and you were to come back, and you were to see this migration. You were to see this migration occur. And the longer you wait, the further these things are going to get. If you wait too long, you're going to fall off all the way over the other edge. Is if you just saw this, you say, okay, well, this strand right over here, these must be smaller DNA molecules. They must be shorter. These must be a little bit longer, and these must be even longer than that. And this grouping right over here is going to be the longest of all. So this was a mixture of some longer strands and still longer ones, but not quite as long. And for example, maybe there's some really short strands. Maybe there are some really short strands in that what I'm drawing as that orange group right over here. So what I just did right here, this could tell you the relative length of these strands, but how would you actually measure them? Well, that's where you can go find standardized solutions, which we call a DNA ladder. So let's say there, and some of it goes like there. Well, you would be able to know from the labeling or whichever one you choose to buy that this grouping here, this is all of the DNA that is 5,000 base pairs. Let's say this right over here is 1,500 base pairs, and let's say this over here is 500 base pairs long, longer than 500 base pairs, but shorter than 1,500 base pairs. You can see this green one here. You might say, okay, is this one DNA strand? Is that one DNA strand that I'm looking at? And just going back to the measurements, no, that is many DNAs that you're looking at. They're not all stretched out like that. Remember, even something that is 5,000 base pairs long is only going to be 1 to 2 micrometers if you stretch it out. So you wouldn't even be able to see it. It's a thousandth of a millimeter. You wouldn't even be able to see it. So this is many molecules of DNA is migrating that far. They would have to be that small to be able to migrate through that polysaccharide gel. Now, the last thing you're probably saying is, okay, wait, but how am I even seeing it over here? How do I actually see this DNA, especially if they're these super small molecules? And the answer is you put some type of intercalating agent, and it fit itself. That's why we call it inside of DNA, and you would actually see these things. And so if you wanted to see what it actually would look like in real life, well, this is what it would look like if you were to look at it straight on, where this would have been a well. Let me make it a bit easier to read. So right over here would have been the well where you would put the DNA ladder, and it would come up with standardized measurements. Maybe that's our 5,000 base pairs. This right over here is our 1,500 base pairs, and this right over here is our 500 base pairs. And then let's say you have some solution of some other DNA, and you wait a little while, and you see, look, it migrated not quite as far as the 500 base pairs. So this must be a bundle of things a little bit longer than 500 base pairs, but for sure a lot shorter than 1,500 base pairs. And once again, it doesn't have to have just one fragment length. You could have had another group that was maybe right. At 1,500 base pairs. And you probably see this whenever you see people talking about genetic analysis and things like this. You're often seeing people look at one of these readouts from gel electrophoresis. So now you know what's actually going on here. This isn't a strand of DNA. There's a bunch of DNA that has been tagged with some type of a dye or lithium bromide or something like that. And it's a bunch of those molecules, and they've migrated based on the charge. They're trying to get away from the negative charge to the positive charge. And the smaller molecules, this is a bunch of small molecules right over here, are able to get further because they're able to get through the mesh of the agarose gel.
Extract this from the fragment.
I'm here with Emily, our biology content fellow, to talk about PCR, or polymerase chain reaction, which you've actually done a lot of. Why have you done PCR?
PCR was kind of the mainstay of my graduate project.
It's quite a lot here, but let's try to 2x speed. I need to pee again, dude.
Where I built all sorts of different recombinant DNA molecules and use them to learn things about plants.
And so what does PCR in particular do?
PCR basically makes you a lot of copies of a particular fragment of DNA that you're interested in.
And why? And so how does that? Like, why would you need to make a lot of copies of a particular fragment of DNA?
So you might want to be making lots of copies so that you can clone it into a plasmid and then do some other experiments with it. That's a big use.
So when we talked about cloning, and we're talking about sticking a fragment of DNA inside of a plasmid. It's not like you're just taking one fragment into one plasmid. You're doing that with many. So you need a lot of fragments of DNA.
Exactly. That is exactly it.
And you might start with a very small sample of DNA. So you just really do that. Where else would you have to do PCR?
PCR is used a lot in forensics. It's also used a lot in medical diagnostics. So this could actually be your DNA that was being checked to see if you have a gene that would predispose you to a particular condition. All sorts of really practical applications.
Because it's hard to identify just one fragment of that gene. So you want to make copies, or as they say, amplify it, so that you can write it in gels and stuff and see how all of those molecules, how big they are, or something like that.
Exactly. If you were just looking in your DNA pulled out of your cell, that would be a needle in a haystack. So this is how you can really zoom in and look at just the thing you need to see.
Okay, so you've drawn some diagrams here, and I actually have never done PCR, but you have. So I'm going to tell you how I understand it happening, and then you tell me if this makes sense. So what you drew over here, this is double-stranded DNA, just and it's going to be from a sample of someone's hair or whatever else. And let's say we want to replicate or make many copies of a fragment of this. And so let's say the fragment that we really care about is the fragment roughly from there to there. This part is what we want to make. We want to make multiple copies of. And so this first step, denaturation.
It's a weird word.
It's a weird word. You have 96 degrees Celsius, so this is almost at the boiling point. So it's quite hot, and that separates the two strands.
Precisely.
And so once they're separated, then you can cool things down. Although this still isn't that cool. 55 degrees Celsius would be very uncomfortable. But you would cool it down to this, and then these primers show up. And so, one thing to remind ourselves is this process is happening inside of a test tube or in a big solution. So you heat it up, the DNA, the two strands separate, and you just have this primer lying around.
So the primer is something that you've ordered from a company, and you've ordered a lot of it. So you put in a ton of primer in your reaction so that there's a really good chance that when you get to this step here called annealing, that a primer is going to bind to many of your pieces of DNA.
So this is our solution. Is this all happening in water?
Water with some salts and stuff floating around, yeah.
Okay, so we have our solution right over here. You'd put whatever your initial DNA sample is in there, and once again, it's a very small amount. You put a lot of that primer, so you want to put that in a lot of surplus when you do that in some Technicolor. You obviously would see it in real life, just all dissolve.
It would just look like a drop of liquid.
It looked like, but for visualization, you put a lot of primer, and so you heat it up. The DNA strands separate, and then when you cool it back down, this primer is going to be specific to the ends of the region that you want to copy.
Exactly.
And so when you go to just
look like a drop of liquid.
It looked like, but for visualization, you put a lot of primer, and so you heat it up. The DNA strands separate, and then when you cool it back down, this primer is going to be specific to the ends of the region that you want to copy.
Exactly.
And so when you order online or wherever that you want a certain primer, you're going to pick the sequence of that primer to be specific to the regions you want.
Exactly. That's super important.
Okay. And so when you cool it back down, the primer attaches, and then you heat it back not quite to the 96 degrees Celsius, but to 72 degrees Celsius.
Exactly.
Go to
a drop of liquid. So there's a really good chance that
But you denaturation.
It's a weird word.
It's a weird word. You have 96 degrees Celsius, so this is almost at the boiling point. So it's quite hot, and that separates the two strands.
Precisely.
And so once they're separated, then you can cool things down, although this still isn't that cool. 55 degrees Celsius would be very uncomfortable. But you would cool it down to this, and then these primers show up. And so, one thing to remind ourselves is this process is happening inside of a test tube or in a big solution. So you heat it up, the DNA, the two strands separate, and you just have this primer lying around.
So the primer is something that you've ordered from a company and you've ordered a lot of it. So you put in a ton of primer in your reaction so that there's a really good chance that when you get to this step here called annealing, that a primer is going to bind to many of your pieces of DNA.
So this is our solution. Is this all happening in water?
Water with some salts and stuff floating around, yeah.
Okay, so we have our solution right over here. You put whatever your initial DNA sample is in there, and once again, it's a very small amount. You put a lot of that primer, so you want to put that in a lot of surplus and do that. You obviously can see it in real life, you just don't solve.
It would just look like a drop of liquid.
It looked like, but for visualization, you put a lot of primer, and so you heat it up. The DNA strands separate, and then when you cool it back down, the primer is going to be specific to the ends of the region that you want to copy.
Exactly.
And so when you order online or wherever that you want a certain primer, you're going to pick the sequence of that primer to be specific to the region you want to copy.
Exactly. That's super important.
Okay, and so when you cool it back down, the primer attaches, and then you heat it back up, not quite to the 96 degrees Celsius, but to the 72 degrees Celsius, where you extend those. And I'm assuming since it's called polymerase chain reaction, that this is where the polymerase is involved.
That is exactly where the polymerase comes in.
So the polymerase is what is actually extending this. And is it, so I'll just draw a polymerase enzyme right over there, doing the extending. And is it any type of polymerase enzyme? Can I just take like the polymerase from my cells and throw it in there?
So you actually need a special polymerase because you need one that is going to be pretty heat resistant. So as you were mentioning, even the cool step of this process is not something that your body would want to be hanging out in. So the polymerase is actually from a really heat tolerant microorganism.
And what is that? It's called a TAC polymerase?
Thermophilus aquaticus, I think.
Thermus aquaticus.
Makes quite a mouthful. And
they found it at heated vents, this organism that is able to stand high temperatures. But that I guess leads to another question: why do you have to heat it up to begin with? I guess just to separate the two strands.
That's really the key reason. You just have to get them apart. You don't have an enzyme to do it the way you might in a cell. So heat does the trick.
Okay, so I get it. So this is one step. I'm getting at least the polymerase part of the PCR, where you heat it up, the strands separate. Then you have all of this extra primer there. The primer, because there's so much primer, the primer is much more likely to bind to at least at this part of the sequence than for these two strands to get back together at this point. And then you have the polymerase, the TAC polymerase in particular. And you would have added that at the beginning. The TAC polymerase, I'll do it in yellow color. So you would also put all that TAC polymerase in there. And once again, these things are robots. They don't know exactly what they do. They just bump into things in the right way and react in the right way. And then you also have to add a bunch of nucleotides.
Yes, absolutely. Your reaction is not going to work if you forget the nucleotides.
So the TAC polymerase, when you heat it back up again after the primers have been attached, is going to start adding all of these nucleotides. And what, you just wait a certain amount of time? Will it just keep going on forever?
It'll keep going on for a while. Usually you do pick the length of that step to match how much time you expect the polymerase to need to complete your fragment. But it kind of will stop. Either it'll fall off or it'll stop and you go on to the next step.
Okay, so I get this so far. So far we have, after one cycle, after one cycle we would have doubled at least that part of the sequence that we care about. Although we might have copied even beyond that sequence. So where's the chain reaction come to this?
So I guess you can interpret chain reaction in two ways, and one is that's sort of what the polymerase does, is it adds things to make a chain. But there's actually even more of a chain reaction to mention here, and that's that we're actually getting this kind of exponential process going on.
So you do it one cycle, you get to this situation right here. You heat it up, the strands separate, you cool it down, the primers attach, then you heat it up again. The TAC polymerase does its job, and like all polymerase, it goes from the 5' to the 3' direction. We talked about that in the application. So now you have two strands, but now since all that stuff is in that solution, you can heat it up again. Now these two strands can turn into, or these two double strands can now turn into 4 single strands. Then you can cool down again. Now they get primers attached to them, and they're still the same primers. We still care about the same sequence. And then that can keep, and so now you go from 1 to 2 to 4, and so you keep repeating this. So how many times would be typical for you to repeat the cycle?
So like 35 might be a pretty typical number of cycles to do. Depends a little what you're doing, but you're going to do it a lot of times.
And so if you do this 35 times, I mean, each time you're multiplying by 2, so it's going to be 2 to the 35th power, which is well over 1 billion times. So, and how would that take? You've done it before.
Depends on the length of your fragment, but usually like 2 to 3 hours.
So in 2 of the hours, you can start with one fragment and get into the billions.
If it's perfectly efficient, which I wish it always were, but you usually get quite a few pieces made.
And one thing that I've always wondered when I first learned about this, and I'd like to go to the lab and do this with you, is okay, I get that you have a primer, and then the polymerase is just going to extend it like that. But I was like, well, it's going to be, how does it know where to stop? And you explain, well, on that first pass, it might not know where to stop. But then when you start going in the other direction, it's over here. And when it goes in the other direction, it's not going to have anything else to copy.
Exactly.
And so then so most of the billions of molecules that you produce are going to be both ends, kind of a nice cut.
The vast majority, exactly.
Fascinating.
All right, so let's say that you've got this little guy over here, and he's got his shoes, and he's just happy. All right, so let's say that you've got this little guy over here, and he's got his shoes, and he's just happy smiling. So this guy right here is our protein. So let's look at how this protein was created. So in order to make protein, we have to start out with our base, and in this case, our base is DNA. DNA. From DNA, we generate messenger RNA. And then that messenger RNA eventually leads to the formation of a protein. The protein is this happy guy over here. This is pretty straightforward, but what if we wanted to go in reverse? What if we started out with the protein and we wanted to figure out what its DNA sequence was? So we wanted to go in this direction. So let's look at how this is done. Now, scientists thought that it would be nice to basically be able to type in the name of any protein that they're interested in, and automatically it would pop up with the DNA sequence of that protein. Now, that is known as a DNA library. DNA library. And a DNA library would be beneficial for researchers and scientists and clinicians. So let's look at how this is done into that protein. Now, that is known. So let's look at how this is done. Now, scientists thought that it would be nice to basically be able to type in the name of any protein that they're interested in, and automatically it would pop up with the DNA sequence of that protein. Now, that is known as a DNA library. DNA library. And a DNA library would be beneficial for researchers and scientists and clinicians. So let's look at how this is done. So we'll start out with our protein. And our protein is basically a chain of amino acids. So amino acids basically are formed from messenger RNA. So if we know the amino acid sequence of our protein, we know what the messenger RNA sequence is based on the.
It's not possible to infer origin from both the sequence because multiple codons can code, and mRNA sequence can be inferred from DNA sequence.
Codon table that we all are too familiar with. So if we have the messenger RNA sequence, what we do is we add an enzyme known as reverse transcriptase. Reverse transcriptase. And when we add reverse transcriptase, basically takes this messenger RNA and makes a complementary DNA sequence to the messenger RNA, and that's known as cDNA. The C stands for complement. DNA polymerase. So now that we have and then you can take that cloning vector and add it to some bacteria, and it'll basically infect the bacteria, and the bacteria will basically produce lots and lots of this DNA, this double-stranded DNA. And basically, once we have that sequence, we'll put the sequence into a large database that's readily accessible online, and that database will basically populate the DNA library. So now anybody that is interested in the DNA sequence of a particular protein can just go into this library and pull up the genetic sequence of the protein of interest.
What? I thought I finished the video.
And that database will basically populate the DNA. You can just go into this, pull up the genetic sequence of the protein of interest.
There we go.
Let's talk a little bit about DNA cloning, which is all about making identical copies of a piece of DNA. And usually it's a piece of DNA that codes for something we care about. It is a gene that will express itself as a protein that we think is useful in some way. Now, you might have also heard the term cloning in terms of the Clone Wars and Star Wars, or Dolly the sheep. And that is a related idea. If you're cloning an animal or an organism, like a sheep, well, then you are creating an animal that has the exact genetic material as the original animal. But when we talk about cloning and DNA cloning, we're talking about something a little bit similar. Although we'll see it's still quite fascinating. It's identical copies of a piece of DNA. So how do we do that? Well, let's say that this is a strand of DNA right over here, and I'm just drawing it as a line, but this is a double-stranded. I'll just write it down. This is double-stranded. I don't have to take the trouble of keep drawing multiple strands. Actually, let me just try to draw the two strands just so we remind ourselves. So there we go. This is the double-stranded DNA. And let's say that this part of this DNA has a gene that we want to clone. We want to make copies of this right over here. So gene to clone, gene to clone. Well, the first thing we want to do is we want to cut this gene out somehow. And the way we do that is using restriction enzymes. And there's a bunch of different restriction enzymes. And I personally find it fascinating that we as a civilization have gotten to the point that we can find and identify these enzymes, and we know at what points of DNA that they can cut, they recognize specific sequences, and then we can figure out, well, which restriction enzyme should we use to cut out different pieces of DNA? Well, we have gotten to that point as a civilization. So we use restriction enzymes. We might use one restriction enzyme, use a different color here, that latches on right over here and identifies the genetic sequence right over here and cuts right in the right place. So that might be a restriction enzyme right over there. And then you might use another restriction enzyme that identifies the sequence at the other side that we want to cut. So let me label those things right over there. Those are restriction enzymes. Restriction enzymes. And so now you would have, after you apply the restriction enzymes, you will have just that gene. You might have a little bit left over on either side, but essentially you have cut out the gene. You've used restriction enzymes to cut out your gene. And then what you want to do is you want to paste it into what we'll call a plasmid. And a plasmid is a piece of genetic material that sits outside of chromosomes, but they can reproduce along, or that could actually, they can replicate along with the machinery of the genetic machinery of the organism, or can express itself just like the genes of the organism that are in the chromosomes express themselves. So this is where we cut. We write this. We cut out the gene, and then we want to paste it. Then we want to paste it into a plasmid. And plasmids tend to be circular DNA, so we will paste it into a plasmid. And in order for them to fit, there's oftentimes these overhangs over here. You might have an overhang over there. You might have an overhang there. And so the plasmid that we're placing in might have complementary base pairs over the overhangs, which will allow it will become easier for them to react with each other if they have these overhangs. So we're pasting it into a plasmid. And this is amazing because obviously DNA, this isn't stuff we can manipulate with our hands the way that we would copy and paste things with tape. You're making these solutions and you're applying the restriction enzymes. The restriction enzymes are just in mass cutting these things. They're bumping it just the right way to cause this reaction to happen. Then you're taking those genes and then you're putting them with the plasmids that happen to have the right sequences at their ends so that they match up. And you also put in a bunch of DNA ligase to connect the backbones right over here. And we also saw DNA ligase when we studied replication. So that is DNA ligase, which you can think of it as helping to do the pasting. And so now we have this plasmid, and we want to insert it into an organism.
Did he say the plasmid was?
Is a piece of genetic material that sits outside of chromosomes, but that can reproduce.
Sits outside of chromosomes.
Along with that, they can replicate along with the machinery of the genetic machinery of the organism, or it can express itself just like the genes of the organism that are in the chromosomes express themselves. So this or a type of organism in that solution, and some to connect the backbones right over here. And we also
Okay.
saw DNA ligase when we studied replication. So that is DNA ligase, which you can think of it as helping to do the pasting. And so now we have this plasmid, and we want to insert it into an organism that can make the copies for us. And an organism that's typically used is, or a type of organism is bacteria, and E. coli in particular. And so what we could do is we could, let's say that we have a bunch of, let's say you have a vial right over here. You have a vial, and it has a solution in it with a bunch of E. coli. A bunch of E. coli. You actually would be able to see it visually, but there's E. coli in that solution. And then you would put your plasmids, which would be harder to see, in that solution. And somehow we want the E. coli, we want the bacteria to take up the plasmid. And the technique that's typically done is giving some type of a shock to the system that makes the bacteria take up the plasmid. And the typical shock is a heat shock. And this isn't fully understood how the heat shock works, but it does. And so people have been using this for some time. So if you have a bacteria, right over here. It has its existing DNA. So this is its existing genetic material right over there. Let me label this. This is the bacteria. You put it in the presence of our plasmids. So you put it in the presence of our plasmid, and you apply the heat shock. And some of that bacteria is going to take in the plasmid. It's going to take in the plasmid. And so just like that, it's going to take it in. And so what you then do is you place the solution that has your bacteria, some of which will have taken up the plasmid, and you put it, and then you try to grow the bacteria on a plate. So let me draw that. So here we have a plate to grow our bacteria on. And it has nutrients right over here that bacteria can grow on. It has nutrients. And so you could say, okay, we'll put this here, and then a bunch of bacteria will just grow like that in the plasmid. It's going to take in the plasmid. And so just like that, it's going to take it in. And so what you then do is you place the solution that has your bacteria, some of which will have taken up the plasmid, and you put it, and then you try to grow the bacteria on a plate. So let me draw. So here we have a plate to grow our bacteria on. And it has nutrients right over here that bacteria can grow on. It has nutrients. And so you could say, okay, we'll put this here, and then a bunch of bacteria will just grow. So you'll see things like this, which would be many cells of bacteria. There would be colonies of bacteria. You could just let them grow. But there's a problem here, because I mentioned some of the bacteria will take up the plasmids and some won't. So you won't know, hey, when this bacteria, when it keeps replicating, it might form one of these colonies. So this is a colony that you like. So this one is a good colony, a check mark there. But maybe this colony is formed by an initial bacteria or set of bacteria that did not take up the plasmid. So it won't contain the actual gene in question. So you don't want that one. So how do you select for the bacteria that actually took up the plasmid? Well, what you do is
You gotta mark it, right? Tag it.
besides the gene
You gotta be able to see it.
that you care about, that you want to make copies of, you also place a gene for antibiotic resistance in your plasmid. So
Antibiotic resistance.
now you have a gene for antibiotic resistance here. And so only the bacteria, I think it's amazing that we as humanity are able to do these types of things, but now only the bacteria that have taken up the plasmid will have that antibiotic resistance. And so what you do is, in your nutrients, you do nutrients plus antibiotics. What's an antibiotic? This bacteria, when it's here, then a bunch of bacteria, some of which will have taken up the bacteria here, then a bunch of bacteria will just grow. You can just let them grow. And so you won't know, so this is a colony, initial, or the bacteria, the bacteria, I think, that actually took up the plasmid. Well, what you do is, besides the gene that you care about, that you want to make copies of, you also place a gene for antibiotic resistance in your plasmid. So now you have a gene for antibiotic resistance here. And so only the bacteria, I think it's amazing that we as humanity are able to do these types of things, but now only the bacteria that have taken up the plasmid will have that antibiotic resistance. And so what you do is, in your nutrients, you do nutrients plus antibiotics. What's an antibiotic? Antibiotic. And so this one will survive because it has that resistance, it has that gene that allows it to not be susceptible to the antibiotics, but these are not going to survive. They're not even going to happen. They're not even going to grow because there's antibiotics mixed in with those nutrients. And so this is a pretty cool thing. You started with the gene that you cared about. You cut and pasted it into our plasmid. Write the labels down. Into our plasmid. That also contained a gene that gave antibiotic resistance to any bacteria that takes up the plasmid. You put these plasmids in the presence of the bacteria. You provide some type of a shock, maybe a heat shock, so that some of the bacteria takes it up. And then the bacteria starts reproducing. And as it reproduces, it also is reproducing the plasmids. And because it has this antibiotic resistance, it is going to grow on this nutrient antibiotic mixture. And the other bacteria that did not take up the plasmid are not going to grow. And so just like that, you can take this. You can take this colony right over here and put it into another solution or continue to grow it. And you will have multiple copies of that gene that are inside of that bacteria. Now, the next question, and I'm oversimplifying things fairly dramatically, is, well, how do you now have a bunch of bacteria that have a bunch of copies of that gene? How do you make use of it? Well, the bacteria themselves, let's say that gene is for something you want to manufacture, say insulin for diabetics. Well, you can actually use that bacteria's machinery, use its reproductive machinery to keep replicating the genetic information. But you can also use its productive machinery, as you could say. So it's going to express its existing DNA, but it can also express the genes that are on the plasmid. In fact, that's what would give the bacteria its antibiotic resistance. But if this gene was, say, for insulin, well, then the bacteria will produce a bunch of insulin molecules, which you might be able to use in some way. I'm not going to go into all the details of how you will get the insulin out and how you can make use of it, but needless to say, it's pretty cool that we can even get to this point.
So in this video, we're going to be talking about something known as DNA hybridization. DNA hybridization. All right, so in this video, we're going to be talking about something known as DNA hybridization. DNA hybridization.
He's saying like 10 times. Okay, DNA hybridization.
What is DNA hybridization? Well, basically what it So let's work through an example to try and explain what DNA hybridization is. So let's imagine that we have two cells. So over here we have
There's 3 more after restriction enzyme, an article and 2 videos. Okay.
a cell A, and over here we have cell B. Now let's imagine that cell A is a cancer cell. So this is a cancer cell, and cell B over here is a normal cell. This is normal. Now, cancer cells basically have the ability to proliferate and grow and metastasize and move throughout the body. So basically they have this unregulated cell growth. And the reason that the cell growth is unregulated is because there are various mutations that cause changes in the proteins that are expressed and changes in the regulation of the cell cycle. And there are hundreds and hundreds of different mutations, hundreds of different proteins that can be affected, and all of them can lead to cancer.
All right. I'm gonna just quickly. I mean, I think we can maybe get through the next ones in like maybe an hour. Evolution and dynamics. There's quite a lot, but maybe 2. So I'm looking at 10 already, and then principle of bioenergetics, depending on what it is. Maybe we can get it done in like 30 minutes. Maybe we'll start to 12 today just to see. Yeah. Something like that. Something like that. We have to hit 4. We have to hit 4. For sure, we have to hit 4 today and tomorrow. Okay, let me just quickly get something. I don't know how the other ones are gonna look like. Hold on. Anyways, I don't know how the other ones are gonna look like, but I really don't think that the next ones for the one I was supposed to do this week is gonna take me too long. I haven't looked at it though. But I'm not gonna spend time right now. Anyways, let's continue this one. I think maybe this one I can do in like an hour. Finish it 9, take a 10-15 minute break, work out. Then we're on this one. Then I think maybe 2-ish or something. And this one, since it seems really short, unless these videos are like 30 minutes long, I hope not. We can finish that. Try and do it before golf. I don't know. And then we add Anki. We could add Anki tomorrow. Well, then that means we'd be late with it. I think, right? Yeah, maybe. Yeah, maybe tomorrow then. Oh, I don't know. Oh, we'll see when we get there. Yeah, let's continue.
Different amounts. Now, what are kind of the two options that we have for certain genes? Let's imagine that we have gene A over here. So if this is gene A, what are the two options? Either gene A can be upregulated or it can be downregulated. So if it's upregulated, then what we have is we have the gene products, which is mRNA and eventually protein. We have a lot more of the mRNA and the protein that gene A encodes for. And what that basically means is that let's imagine that gene A encodes for a protein that will induce cellular proliferation and will allow that cell to go and metastasize throughout the body. Well, if we have a lot more of gene A being expressed, either because the promoter is upregulated or for whatever reason, now we have lots and lots of this protein that basically allows cellular proliferation to occur. We have lots of this protein floating around the cell, and we have this cancer cell proliferating uncontrollably. So another option is if we have gene B. So if we have gene B, gene B could be downregulated. And that basically means that that gene B isn't producing its gene product. And what if that gene product were something that basically stopped the cell from proliferating? Well, if we have less inhibition, then we basically have more proliferation. And the third option for any specific gene in a cancer cell, so let's say gene C, is that there's no change. So there's just no change. So what we want to do is use DNA hybridization technology in order to assay the gene transcription profiles of a cancer cell compared to a normal cell. And in order to do that, we need to use something known as a microarray. So a microarray. Now, what is a microarray? Well, array basically means that we're assaying a whole bunch of different things. And in this case, we're assaying the transcription profiles of a bunch of different genes. And micro just means that it's small. So this could be as small as a chip. So let's imagine that we have a microarray chip. So let's say that we've got this chip, and it's basically just this square. And this chip has a lot of different holes in it. So let's imagine that we've got just lots and lots of holes. So we have just hundreds of these holes. And I'll just draw a few for simplicity's sake. So we have a bunch of these holes on the microarray chip.
Yeah, that was so much.
Now, these holes are actually microarray chips. So let's say that we've got this chip, and it's basically just this square. And this chip has a lot of different holes in it. So let's imagine that we've got just lots and lots of holes. So we have just hundreds of these holes. And I'll just draw a few for simplicity's sake. So we have a bunch of these holes on the microarray chip. Now, these holes are actually little tiny wells. They're microscopic wells. So if we actually look at this from the side, so here's the chip. We're looking at it from the top. So it's lying on the table. We're just looking down at it. If we looked at it from the side, one of these wells would look like this. And inside the well would be a complementary mRNA strand. So we've got just lots and lots of these little complementary mRNA strands. And what are they complementary to? Well, they're complementary to a specific gene. So let's say that inside one of these wells, let's draw another well over here. Let's say that inside one of these wells, we have the complementary mRNA to gene A. So we've got the complementary mRNA to gene A. Now let's imagine that in this cancer cell, gene A is upregulated for whatever reason. And if gene A is upregulated, it's being overtranscribed, and that means that there's lots and lots of the gene A mRNA floating around in the cell. So there's just a bunch of the gene A mRNA. And this is in comparison to the normal amount of gene A products, which might just be a few gene A mRNAs. Now, what we can do is we can take this cell and we can break it apart. And we can label the mRNA with a certain color. So let's say that I label each one of these mRNAs with a yellow fluorescent label.
Mm-hmm.
So let's imagine that I labeled every single one of these mRNAs with a yellow fluorescent label. And let's imagine that I labeled the mRNA in the normal cell with a blue fluorescent label. Now, what I can do is I can break these cells apart, and I can basically add the intracellular contents to this well. So I can add it to this well. And since I have lots and lots of this mRNA that's labeled yellow, what I'm going to have a lot of the mRNAs binding to the complementary strands. And so I'm going to have a really bright yellow well. And when I add the normal cell intracellular contents, I'm going to have some blue. So I'm going to have lots of yellow and a little bit of blue. And what that'll basically look like is it'll really just, you won't be able to see the blue. It'll really look like just a bright yellow dot. So let's imagine that this is the well. It'll look like a bright yellow dot. And a computer can scan every single one of these wells and basically decide, okay, is it a brighter yellow or is it a brighter blue? If it's a brighter yellow color, if you see mainly yellow, that means that you have a lot more of that specific gene's products being expressed in the cancer cell compared to the normal cell. Now, let's imagine that we look at a downregulated gene. So if we look at a downregulated gene, let's just draw another well. So let's draw a well over here. Now, if we look at a downregulated gene, we've got lots of the gene B mRNA, the complementary strands inside this well. And we're going to have very few of the gene B mRNA in the cancer cell and then a lot more in the normal cell. And once again, we'll label the gene B mRNA with a yellow fluorescent label. And over here, again, over there, we're going to label it blue. So we'll label it with this blue fluorescent label. And once again, we're going to lyse the cells and we're going to expose the intracellular contents to this well. And what we're going to have is we're going to have very few of the gene B products binding. And we're going to have a lot of the normal cell, of the gene B byproducts in the normal cell binding. So when you look at this well, it's going to pop up as mainly blue. And once again, the computer is going to read this and it's going to notice, oh, well, this well has mainly a blue fluorescent label, which means that the normal cell is expressing a normal amount and there's a lot less of that gene being expressed in the cancer cell. So this is kind of the idea of a microarray chip in assaying the gene expression profile in a cancer cell versus a normal cell. It's able to tell you whether a specific gene is upregulated or downregulated. And you're also able to see if a specific gene has no change. And if there's no change, then instead of seeing either a yellow or a blue dot, you would see something kind of in the middle. So maybe you'd see a green dot. And that's basically a quick way in order to look at a whole bunch of different genes on a single chip and try and quickly determine which gene is upregulated or downregulated in the cancer cell compared to a normal cell. And this can help you tailor your therapy. So let's say that you know that this well right here is for a specific protein and you have a drug that's able to target that protein. Well, now you're able to tailor your therapy for that individual patient using this microarray technology.
Okay. I might just read a little bit for this because people are saying you didn't really do that well. I mean, I kind of understood it, but if you understood it, doesn't mean you really learned it. Mm. Okay.
So here we have gene array. Well, let's imagine that we've got just our microscopic wells. So if we actually looked at this from the side. So here's the chip. We're looking at it from the top. So it's lying on the table. We're just looking down at it. If you looked at it from the side, one of these wells would look like this. And inside the well would be a mRNA, a complementary mRNA strand. So we've got just lots and lots of these little complementary mRNA strands. And what are they complementary to?
Gotcha. Which genes are active, mutated, or present in the sample? It's just complementary DNA. Like the mRNA from both samples, reverse transcribe into cDNA. Gotcha.
Well, they're complementary to a specific gene. So let's say that inside one of these wells, let's no reason, the mRNA with a certain color. So let's say that I labeled really just your specific gene.
Yeah, I mean, he explained pretty well. I mean,
Normally when we think about cloning, we think about cloning in this sense. So let's say that we've got a baby and it's just so cute that we want 2 of them. So we can go and clone a baby. Well, this isn't exactly what we're talking about when we talk about cloning in the scientific sense. Normally what that means scientifically is, let's say that I've got a cell and there's a certain gene, let's call it gene A for now, and I want to clone that gene. So how do I go about cloning the gene A? Well, the first step is to isolate the messenger RNA from the gene of interest. So if we have the messenger RNA, what we need to do is we need to convert it into DNA. So if we add reverse transcriptase, messenger RNA from the first step is to isolate the messenger RNA gene, let's call it DNA. Isolate the messenger RNA from the gene of interest. So if we have the messenger RNA, what we need to do is we need to convert it into DNA. So if we add reverse transcriptase, which is an enzyme that will go and actually create DNA from RNA, we'll end up with something known as complementary DNA. Now, complementary DNA, and that's what the C stands for, basically the complementary DNA sequence to the mRNA sequence. Now, keep in mind that the complementary DNA only contains exons, so the introns have already been spliced out.
Oh.
And we talked about introns and exons in an earlier video. So next, what we have to do is we take this cDNA, this complementary DNA, and now what we want to do is we want to amplify it. We want to create lots and lots of the complementary DNA so that we can have plenty of cDNA to work with. So in order to do that, we have to take this complementary cDNA and transform it into a plasmid. Now, what does transform mean? Well, what it basically means is that we're taking the complementary DNA and we're basically putting it into a plasmid. So we're going to transform it into a plasmid. And this plasmid is going to contain antibiotic resistant genes. So antibiotic resistant genes. And we'll talk about why that's important in just a second. So now that we have this plasmid, we want to actually infect bacteria with the plasmid. So we want to put it into bacteria. And once it's in the bacteria, what we want to do is we want to actually add antibiotics to the culture of bacteria. So we're going to add antibiotics. And since the plasmid contained antibiotic-resistant genes, the bacteria that were successfully transfected will survive, and all the other ones that didn't have the plasmid inserted will die. So now we've got pure culture of bacteria that all contain the plasmid, which contains the gene of interest, the DNA of interest. And now the bacteria just does its thing. So it's just going to start to replicate. The bacteria is going to replicate, and as it's replicating, it's going to produce lots and lots of the gene of interest. It's going to produce lots of the mRNA of interest. And basically what we just did through this process is we just cloned the gene of interest. And that's what we're talking about when we're talking about cloning, at least from a scientific point of view.
Okay. Okay. Thanks, buddy. Thank you.
So in this video, I'm going to be talking about something known as a southern blot. So a southern blot basically allows you to visualize a specific piece of DNA that you're interested in. So let's imagine that we have a cup and it's filled with DNA. So it's got just a whole bunch of DNA inside. And there's just lots and lots of this DNA.
What?
So in this video, I'm going to be talking about something known as a southern blot. So a southern blot basically allows you to visualize a specific piece of DNA that you're interested in.
Okay.
So let's imagine that we have a cup and it's filled with DNA. So it's got just a whole bunch of DNA inside. And there's just lots and lots of this DNA. And let's imagine that I'm specifically interested in one gene. So let's imagine that I'm interested in gene A. And I want to see if gene A is inside of this cup, if it's inside of this long piece of DNA. Now, in order to figure out whether or not gene A is inside this cup, basically we have to do this process known as a southern blot.
Okay.
And we'll break it up into a couple of different steps.
Sure.
So step one, what we're going to do is we're going to take this DNA and we're going to cleave it. So take the DNA and cleave it.
But why?
So let me draw that out. So we're going to take this big old strand, we're going to remove it outside of the cup over here. So we've got this big strand, and we're going to cut it up. We're going to expose it to enzymes that will basically cleave the DNA in a whole bunch of different parts.
Sure.
And that'll result in lots of these smaller pieces of DNA. And so that's basically the first step. So we've got a bunch of small little pieces of DNA. Now, step two, what do we do? Well, what we're going to do is we're going to take all of these tiny little DNA fragments and we're going to run them on the gel. So specifically, we're going to do a gel electrophoresis on these DNA fragments. And I made a video on gel electrophoresis, so if you want to refresh, you can watch that video. But basically, the gel electrophoresis will help us separate these DNA fragments based on size and based on charge. So let's just diagram that out. So we're going to take these DNA fragments and we're going to run them on a gel. So let's imagine that this is the gel, and we add the DNA fragments to different wells. So the fragments are going to move down the gel, and they're going to basically be separated based on size and based on charge. So we're going to have these fragments separated like so. So now we've got this gel, and we've got the DNA fragments separated by size on this gel. So the next step, step number 3, is basically we're going to take this gel and we're going to transfer it to a filter.
We got to make DNA to the DNA, make it single-stranded by making a pH basic.
The gel onto a filter. And what the filter will basically allow us to do is it'll allow us to visualize, because this gel is very flimsy, so we want to transfer it onto a filter. So what we'll do is we'll take a filter that's basically the same size as the gel, and we're going to basically just put it right on top of the gel for a little bit, and the fragments will basically transfer onto the filter. So now we're going to have a filter with these fragments, and the filter is a lot sturdier than the gel. So this is the filter, and I'll just write that down over here, and this over here is the gel. Okay, so the next step, step number 4, is we're going to take the filter, and we're going to expose it to a radio-labeled D piece of DNA. So expose to radio-labeled DNA. Now, this radio-labeled DNA is going to be the complement to our gene of interest. So we're interested in finding out if gene A is present in this massive DNA over here. So what we do is we're going to take the complementary sequence to gene A and radio-label it and expose it to this filter. So let's imagine that the radio-labeled piece of DNA is this pink piece of DNA. And let's imagine that we do have gene A. So let's imagine that this piece of this DNA fragment was actually gene A, or gene of interest. So So what's going to happen is when we expose the radiolabeled DNA to this filter paper, it's going to anneal to our gene of interest. So we're going to have this radiolabeled piece of DNA stuck to this DNA fragment, which is its complement. So in order to visualize it, in order to visualize this radiolabeled piece of DNA, we have to do the fifth and final step, which is expose the filter to an X-ray film in order to visualize the radiolabeled probe. So expose to X-ray. And the X-ray, basically, it'll shoot a bunch of
The radiation in a probe itself exposes the film.
X-rays instantly. Since this piece of DNA is radiolabeled, it will pop up on the X-ray film. So we're going to have a film, and we'll draw that film over here. So we'll have this film. And basically, the only thing that'll pop up is this fragment over here, and that fragment will have a control, and we'll be able to say, okay, well, since we have this fragment, it's basically the radiolabeled piece of DNA, and since we see the radiolabeled DNA, it means that it was bound to this gene A, which means that gene A was in this cup of DNA.
So it's to identify a specific sequence of DNA, a gene from DNA. What is it? Is there a wash that? Signal show up no matter what. Well, I guess that would make sense, huh? The one sounded is 1,000 times better.
Have you ever wondered how we sequence DNA? Well, let's just take a quick look at DNA sequencing. So we're going to break down DNA sequencing into 3 different steps. So the first step is you take the sample of DNA that you're interested in sequencing, and you basically use PCR to amplify the sample. So by using PCR to amplify the sample, you're able to generate lots and lots of DNA fragments. So the next thing that you do is normally in PCR, you have to add nucleotides. You have to give the growing strand the substrate from which it can grow. So normally you add in regular deoxynucleotides, and those look something like this. You got an OH group here, you got an H group here, you have a base, and you've got a carbon group, oxygen, hydrogen. So this is what a normal nucleotide looks like. But interspersed in the PCR, what you also want to add is you want to add in something known as a dideoxy.
Oh, okay. Reading the exact order of nucleotides in the DNA lookup.
Nucleotide. So a dideoxynucleotide looks something like this. It's basically exactly the same thing, but it only has a hydrogen here. So this oxygen is removed. And what that basically does is if this dideoxynucleotide, we can abbreviate it NTP, if this incorporates into the growing strand, since there's no oxygen group here, the strand can no longer elongate. So you basically have termination of strand elongation as soon as this DDNTP incorporates. So what you can do is you can actually fluorescently label the different dideoxynucleotides. So for example, we have 4 different options. So we can label all the G's blue, we can label all the A's.
What genes are present? What mutations are rare, or whether something was a certain leader. What's the DNA sequence is at a certain location? Sanger sequencing. PCR, add normal nucleotides, DATP, DTTP, DGTP, DCTP, DDNTPs, each one labeled with a fluorescent tag. DDNTP stop extension, no phosphatides or bonds.
Red, all the T's green, and all the C's orange. And so basically what you have is you have these dideoxynucleotides with different fluorescent labels getting incorporated into the growing strand. And since
Mm-hmm.
PCR is able to amplify and create millions and millions of DNA fragments, you can basically, what you can do is you'll have strands of different lengths. So we'll just kind of look at an example. So let's imagine that we've got nucleotide being incorporated here, a regular nucleotide, and then another one incorporated here, and then another one, and then just randomly, all of a sudden, we have a dideoxynucleotide being incorporated here. And this would stop elongation of the strand. So you can have a DNA strand, that's just 4 nucleotides long. And after another round of PCR, what we might have is we might have 1, 2, 3, 4, 5, 6, just growing, and all of a sudden, whoa, what happened? You got a dideoxynucleotide being incorporated. And so basically you just do this. And after you've got millions of samples, you'll eventually be able to have something that looks like this. You'll have maybe just one regular nucleotide and you've got a dideoxynucleotide incorporated. Or you might have maybe, let's say, 2. So you'll have 2 and you've got a, so what you can basically do is you can see that you have strands that are elongated and different strands are terminated at different points by a dideoxynucleotide. And so basically the next step is you use gel electrophoresis in order to separate the strands by size. So when you run all the different fragments on a gel, it'll separate them by size. And then you can just have a computer go in and analyze all the fluorescent labels. So if it sees here that you've got this blue fluorescent light, then it knows that the second nucleotide in the sequence is a G. So it'll say G. And then it'll look here, and it'll say, okay, well, this is a C. It'll look here, so we have another G, and so on and so forth. And basically, the computer is able to, by reading these fluorescent labels, these fluorescent tags, it's able to give you a DNA sequence. And so this is basically an overview of how DNA sequencing works.
Jeez. That's pretty cool. All right, next one.
So what is gene expression? Well, it's basically the process where a gene is used
We
to
might be
synthesize
early on
some
this
sort of
one.
product. So you go from a gene to a product. And normally this product is a protein, but sometimes you can have non-protein coding genes. You can create things like ribosomal RNA, so I'm going to choose this out. So you can either have a protein, you can have ribosomal RNA, shortened to rRNA, you can have tRNA, tRNA. You can also have something known as small nuclear RNA. So basically you go from a gene to a product. Now, how do we determine what the function of a gene is? How do we determine if a specific gene is exactly what does it do? Well, let's imagine a scenario where there's a cell, and normally it's able to be able, if you give it milk, so let's imagine that we give it a bottle of milk. Just drop a little bottle of milk. Not the greatest bottle in the world, but let's imagine this is a bottle of milk. So, milk. Okay, so if you give the cell milk, and normally it's able to take the milk and digest it, and it's able to use the milk for energy. Well, what if we wanted to figure out, okay, what gene is responsible for being able to digest milk? Well, one thing that we can do is if we have an idea of maybe what gene it might be, we can just knock out that gene. So let's just imagine that there's a gene here, and we imagine that this has something to do with the digestion of milk. Well, if we knock it out, and then we give milk to the cell, and if it's still able to digest milk, then we know that this gene didn't really have much to do with digestion of milk. But if we knock it out and the cell is no longer able to digest milk, then we know that this gene A had something to do with the digestion of milk. So this process is known as a knockout. Gene is responsible for being able to the greatest bottle in the world, but let's imagine this is a bottle of milk. Okay, so if you give the cell milk, and normally it's able to take the milk and digest it, and it's able to use the milk for energy.
Okay, so maybe you don't see so much like ATP within the cell or something.
Well, what if we wanted to figure out, okay, what gene is responsible for being able to digest milk? Well, one thing that we can do is if we have an idea of maybe what gene it might be, we can
Does milk contain glucose?
just knock out that gene. So let's just imagine that there's a gene here, and we imagine that this has something to do with the digestion of milk. Well, if we knock it out, and then we give milk to the cell, and if it's still able to digest milk, then we know that this gene didn't really have much to do with digestion of milk. But if we knock it out and the cell no longer able to digest milk, then we know that this gene A had something to do with the digestion of milk. So this process is known as a knockout. So basically, you're knocking out a gene and trying to figure out what the function is of the gene. So if you knock out a gene, what happens to the organism? So you basically create a knockout loop and study its effects. So another thing you can do is something known as reverse genetics. Reverse genetics. So here what you do is first you start with a gene, and then you sequence it. So you figure out what is the sequence of the gene. And then what you can do is you can look for other gene sequences somewhere else in the genome that share a similar sequence. So you sequence it, and then you look for a homologous sequence somewhere else in the genome. And if you know what that homologous sequence does, then you have a pretty good idea of what that gene might do. So if you know that there's this homologous sequence somewhere else in the genome, and it goes for a specific protein, then you know that the function of that protein, then you know that the gene of interest might create a protein that has a similar function.
Oh, okay. Yeah, that makes sense. Gotcha. Thanks, buddy. My eyes are starting to burn. Gotcha. It's going to produce blunt ends. What does that mean? What does that mean? Blunt end. Does that mean it's not staggered? Like it doesn't have overhangs? Sticky tails that can basically become no overhangs. Both ends are even and flat. Oh, okay. Gotcha. Ooh, smart. Gotcha. Mm. Ikorai? So that's pretty cool. Gap remains, and voila, it's okay now. Doesn't need to be. Eco right site. Take that out, and then put the target gene inside. Get inside. Next, the gene target and linearize open the plasmid. Combine them along with DNA ligands. Stick together, complementary base pairings. Yada yada. Once they are joined by ligands, fragments become a single. Working DNA, vertical gene, and the gene pattern, making a recombinant. Gotcha. Oh, we got one more article after this, and then the rest are videos. And we move on. Let's go. Let's go. And we might be able to like 2 hours, which we'll see if that's enough time. Friction, digest, and ligations involve many molecules. An example above is recircularize. Right? Yeah. You could just close back up. But flip backwards. Ooh, how do you know in that case if it's backwards? Well, I guess you know because then the gene is not, well, because then when you try to identify it, it's not there, right? So we'll get all three at the same time. Gotcha. Heck yeah, I want to check out the simulations. Heck yeah, I want to learn about the simulations. Show me how the simulations. Oh, it's an endonuclease. DNA. Oh, it's dead. It's going. It's okay. I can cut. Here, cutting real good, huh? Nah, it dies. You kill me. Back to the pH. Come back. I came back. Too acidic. Adrenaline is dead. Too basic, it's dead. I got too many hydroxyl group. Find that right balance, which is 7. Oh, it's 7. Okay. Wow, there's so many of them. Right. Oh, restriction enzyme. So do they all have BAM-he? Oh no, BAM-he. Where's the HI? BAM-he. Prime 3. Okay. Oh, what did it say? Flows over. Oh man. Oh really? We gotta get in here with this one, huh? Nutrition science, DNA fragments. Okay. Ligating DNA fragments. Oh. Based on your answer, we gotta go back. It's fine. I don't have time. I don't have enough time for this.
All right, so in this video, we're talking about something known as restriction enzymes. Restriction enzymes. Now, what are restriction enzymes? Well, let's go through an example. Hopefully that'll help answer that question. So let's imagine that we've got a bacteria and it's floating around doing this thing, and out of nowhere comes this virus. And the virus is just kind of upset with it, so it decides that it wants to infect this bacteria. So what the virus does is it goes over to the bacteria and it basically attaches to it, and it injects viral DNA into the bacteria. So this is the viral DNA. I'll just label that here. So viral DNA. And the bacteria, being a living creature, also has its own DNA. So this right here in blue is the bacterial DNA. I'll just label that here. So bacterial DNA. So basically the virus infected the bacteria. But the bacteria wants to figure out some way to destroy the viral DNA. And one way that it can do that is by labeling its own DNA. So let's imagine that the bacteria labels its DNA with these purple dots. All right. So what these purple dots actually are is a methyl group. So we'll say that these purple dots are methyl groups. In order for the bacterial DNA to be methylated, there's an enzyme known as methylase. So methylase is an enzyme that basically floats around. And as bacterial DNA is synthesized, this enzyme methylase goes around and methylates bacterial DNA. Now, what this basically does is it allows the bacteria to recognize its own DNA and recognize any DNA that's not methylated as foreign DNA. So what we now have is another enzyme that's floating around in the cytoplasm of the bacteria. And that enzyme is known as a restriction enzyme. So this restriction enzyme. So this restriction enzyme is kind of floating around, doing this thing, and it recognizes the methylated bacterial DNA, but then it sees this foreign unmethylated DNA, and it goes and destroys it. So this is basically a way for bacteria to protect itself from being infected by viruses, and it basically does so by methylating its own DNA and destroying any other foreign DNA that is unmethylated. So the reason that this is called a restriction enzyme is because researchers were noticing that certain bacteria were actually they were resistant to being infected by viruses, so they were basically restricting viral growth. Thus, when they figured out what enzyme was causing that, they just called it a restriction enzyme. They restricted the growth of viruses. Now, in order for the restriction enzyme to work, it needs to recognize a specific sequence, a specific bacterial sequence. So let's go ahead and write out an example sequence. So imagine that if we zoom down to the nucleotide sequence of the bacterial DNA, let's imagine that maybe the sequence was GAATTC. And of course, if this was one strand, then the sister strand would be GAATTC. Now, I don't know if you noticed, but there's something unique about this specific sequence. So if you read this top line from left to right, GAATTC, and you read this bottom strand from right to left, GAATTC, they're basically identical. And this is known as a palindromic sequence. So palindromic sequence. Now, basically what palindromic sequence is exactly this, and it's what restriction enzymes recognize. So let's imagine that we have a restriction enzyme. We'll just give it a name. Let's imagine that its name is EcoR1. So EcoR1, that's the name of one type of restriction enzyme. EcoR1 is actually able to recognize this palindromic sequence. So as EcoR1 is floating around the bacterial cell, it'll recognize this sequence, and if it's methylated, it won't touch it. But if it's unmethylated, as in the viral DNA, it'll actually cleave it. So it'll come here and it'll cleave it. We'll choose a different color. So it'll cleave the DNA, and it'll cut it like so. And what that'll do is it'll result in two strands, because it just cut the DNA. So now what we're going to have one strand, it's going to be G, and this bottom strand, TTAA, and then we're going to have this top part over here. So we're going to have AATTC, and then G down here. So basically, now these two strands are just floating around, and these ends down here, so this end kind of hanging over the edge, they're known as sticky ends. So these are known as sticky ends. And sticky ends basically will float around, and if they contact one another, since they're complementary strands, they'll just reanneal. So normally, if this gets cut, then we'll have two sticky ends, they'll float around, and they'll reanneal. And we can actually take advantage of the fact that we can have the strands reanneal, and we'll talk about different ways that we can actually use that for medicinal purposes. So one way that we can use this to our advantage would be, let's say that we want to synthesize human insulin. So let's say that I want to make human insulin. So how can I make human insulin using the restriction enzyme technology? Well, let's imagine that we have a bacteria over here. And this bacteria has a bacterial DNA inside. And what we can basically do is take this bacterial DNA, so extract it out of the cell, so move it out here. So here's the bacterial DNA. What we'll do is we'll take EcoR1, and we'll basically, and this bacteria has a bacterial DNA inside. And what we can do is take this bacterial DNA, so let's extract it out of the cell, so we'll move it out here. So here's the bacterial DNA. What we'll do is we'll take EcoR1, and we'll basically cleave the bacterial DNA at this site. So let's say that we cut it right here. So if we cut it right there, and then we kind of unroll the bacterial DNA, now we have this space, right? And this space basically has these sticky ends here. So I'll just fill those in. So let's say that there's a G.
But to cut it, we have to unmethylate it, right? They won't cut. Others are mentalists. I guess others don't really care. So I guess if we're dealing with E. coli plasmid, then maybe. Damn DCN. They already used demethylated E. coli. Methylated deficient strain. Gotcha. There you go.
CTTAA, and then over here on the side on the bottom space, we'll have a AATTC and a G. So now we've got these sticky ends over here, and what we can go ahead and do is we can take the human insulin gene. So we'll take human insulin, this is the insulin gene, and we'll have exposed it to EcoR1 already. So now it's got the sticky ends over here. So now on this side we've got an AATTC and a G, and then over here on the side we've got a G and a C and a T and a K. So now we can take this whole thing and just plop it right in here. And as you can see, this part of the insulin gene will reanneal with this right here, and then this part of the insulin gene will reanneal with this part of the bacterial. And what that will basically allow the bacteria to do is allow it to simply pull a bunch of insulin. So that's what can be used in the pharmaceutical and biotechnological world.
Thanks, buddy. No post. No post yet. Oh, okay. Tip screens. Thank you. Fantastic. You go away. Fantastic. Here we go. Keeping it some things.
So where's the applications?
Buddy, I can't hear you. Hello?
All right, well, let's first look. So what are some applications of DNA technology?
There we go.
Applications of DNA technology. All right, well, let's first look at medicine. So what are some applications of DNA technology in medicine? Well, one of the two big things where recombinant DNA technology was first used was to create insulin and human growth hormone. So before the advent of recombinant DNA technology, it was really hard to manufacture. You basically had to isolate it from another. Human, purify it, and then give it to patients. But with recombinant DNA technology, you can create insulin and human growth hormone. So before the advent of recombinant DNA technology, insulin and growth hormone were really hard to manufacture. You basically had to isolate it from another human, purify it, and then give it to patients. But with recombinant DNA technology, you can basically just grow these proteins in E. coli. You can grow them and culture them in E. coli bacteria. So this really has changed the way that medicine is practiced, and it's really helped a whole bunch of people. So vaccines is another application of DNA technology. A while ago, vaccines were made by first denaturing the disease, and then after the disease had been weakened, they would inject it into a human, and they would hope that their immune system would be able to put up a fight against a weakened virus. And that way, in the future, if they were infected with that virus, they would at least have some kind of immune response towards the virus. The problem with this was that the patient would sometimes get the disease because you're injecting a weak virus, but sometimes it would weaken up. So with DNA technology, they can actually recreate the outer shell of the virus and inject that. So it's a lot more cost effective, and it doesn't have the risk of actually causing the disease in the host. So this is much safer, and it is cheaper, and it's very it produces a better immune response. And so some vaccines that we actually use recombinant DNA technology to create include the Hep B virus and Hep B virus and the herpes virus and malaria. So these are some applications of DNA technology in medicine. Another cool application of DNA technology is in solving crimes. So forensics. So there are parts of the genome known as non-coding regions of the genome, and these regions can actually help forensic scientists identify specific individuals. So they can look at things like short tandem repeats, STRs, and these are basically short sequences of DNA, like 2 to 6 base pairs long, and they're normally found in really high amounts. They're just these short repeats that are found in really high amounts and to varying degrees between different individuals. So if they actually sequence these short tandem repeats, they can identify specific individuals given the DNA sample. They can also look at mitochondrial DNA. So mitochondrial DNA is inherited from your mother, and it's found in really high amounts within an individual cell. So even if there's very little sample available, forensic scientists can analyze mitochondrial DNA in order to identify a potential suspect. Another technology that is used in forensic science is Y chromosome typing. So that's basically YSTR, and this is looking at short tandem repeats found on the Y chromosome. And so DNA technology has helped scientists pick out individuals that have committed various crimes based on DNA samples that they were able to find. So agriculture is another field that has greatly benefited from recombinant DNA technology. So for example, scientists can now create plants crops that are resistant to insects and that are resistant to herbicides, and can also delay ripening of the crop so that you can transport the crop from the farm to the store. So by doing this, you're basically able to create more crops to feed a growing population of individuals. And it also helps with the economy because then you've got farmers that are growing all their crops, and if there was some sort of bacteria or virus that was able to destroy their entire crop, then that farmer would not get paid for that season. So by transgenically modifying the crop so that it's resistant to specific things, then they're able to grow the crop, sell it, and feed individuals.
Gotcha.
So DNA technologies are really cool, and they've provided us with a lot of really good things. However, there are some safety and ethical issues that surround DNA technology. So back in 1975, there was a conference on recombinant DNA, and they concluded that recombinant DNA used for research purposes can be particularly risky. And so they implemented a guidelines to try and minimize that risk. The NIH later issued formal guidelines for recombinant DNA work, and now they're very well regulated, and there are lots of laboratory safety procedures to try and regulate the use of recombinant DNA in the lab. So one example of a safety concern would be, what if we transferred cancer genes? So we took cancer genes and then we put them into a bacterial genome. Then that bacteria, if it infected someone, then it could potentially transfect cancer genes into an individual. So that would be pretty bad. So that's one example of a safety concern using DNA technology. Another safety concern is, how do we protect researchers that are working with recombinant DNA from being took cancer genes and then we put them into a bacterial genome? Then that bacteria, if it infected someone, then it could potentially transfect cancer genes into an individual. So that would be pretty bad. So that's one example of a safety concern using DNA technology. Another safety concern is how do we protect researchers that are working with recombinant DNA from being affected by the technology? Well, there are a lot of safety guidelines in place to try and minimize any exposure risk that researchers have when working with these recombinant DNA technologies. So some ethical issues that come up include, if we're able to modify the genome, then if imagine that there's a pregnant woman and we're able to sequence the baby's genome, and let's say that we notice that there's some kind of defect. What are the ethics surrounding the correction of that defect? Is it ethical to fix a mutation that might cause a cancer, for example? How do we know that fixing a mutation isn't going to cause some other cancer? How do we know what the long-term effects of genetically modifying an infant's genome are? So these are all ethical questions that kind of surround that. And if we kind of drag it out, what if an individual is perfectly normal genetically? Is it okay if we put in genes that make them smarter or faster? What are the ethics surrounding that? So those are some ethical issues. So that would be genetic modification. So another ethical concern that has been brought up is we're able to genetically fingerprint individuals. So forensic scientists are able to pinpoint a suspect's DNA. They're able to figure out they're able to figure out what individual left a DNA sample at a crime scene, for example. So we're getting better at genetic fingerprinting. But what are the ethics around that? What if the government was able to track every single person that opened a certain door based on the DNA that was left behind? Or what if someone took a piece of gum that you spit out on the sidewalk and isolated DNA and was able to track it back to you? So there are some problems around privacy issues. So I'll write that down here, privacy issues. And with the Human Genome Project, how do we prevent genetic information from being used in a discriminatory manner? So, for example, if we know that someone has the gene for a specific breast cancer, then maybe health insurance companies won't insure that individual, or maybe future employers won't want to offer that individual a job because they know, oh, this individual's going to get breast cancer later on. We don't want her working here. So there are these privacy issues that come up with the ability to be able to sequence and modify a gene.
Right? What the heck? Oh, that's it. Wow, there's only 2 comments. Well, DNA technology is done. I'm just going to leave it here because we still have to go through the question. Like I said, I'm going to skip it for now. So we'll duplicate it. I might think about doing this, to be honest. Yeah, potentially. Maybe not. Maybe a later time. What is this? Riles? What is Riles? What the heck's going on? So let's move on to evolution and population dynamics. And I guess we'll start with the article. Okay. Take like a short. Actually, let's just see how long these videos will be.
Now that we're familiar.
Okay, 8 minutes on you.
So let's
10 minutes on you. Article. Hey, this guy's usually 5 minutes. Oh, this guy's here. Let's go. Oh, that means the rest of these are like 5 minutes. Oh, that's so nice. Is this the same guy? Oh,
we've already made several.
we got Sal again. It's still loud, man. Sal again. 4 minutes. Oh, this guy's 4 minutes. 4 minutes. And then a couple articles. Okay. Yeah. Maybe like an hour and a half, I think. This one, I don't know. Well, let's go ahead and also duplicate the tab. What did I do? Which one did I do today? Oh, I did chromosomal. Who's doing this?
Our bodies are constantly.
Oh, it's the girl. Nice.
So in this video,
Nice. It's the girl.
in regular class, I hear another student.
Let's go. Oh, that's really nice. That's really nice. Cool. Let's see. actually make sure we delete the right side because yeah, let's get it out here. I want to make sure that I'm not. Can we put you inside here? Work. No, we add you inside here. There we go. But actually, this one is technically. I just want to name it. We name it to be. What is it? IFD schedule guide. Yeah, I think that makes sense. What the heck? Oh, I think this one's supposed to be 12. So it's supposed to be 12. Okay, over here. Conditionals done, blah, blah, leave it there. We'll duplicate you. And we did meiosis today, so we'll put you like, I don't know, put you like here or something. I think probably I should, maybe if I ever like have some free time, just go ahead and redo the practice problems, like actually go through them. 10, 11, 12, 13. Yeah. Oh, anyways, rename you to be the schedule image questions, something like that. And we'll go back to the summary. There we go. Nice. Yeah, I don't want to get it confused. All right, let's take a quick 5-minute before we begin. Let's put you on here. And then this one, 12, 13, 12. Make sure you're on the right one. I'll just finish the attack. And yeah, cool. We can do it. We can do 4. We can do it. We can do 4, no questions. Maybe questions can happen tomorrow morning. Maybe. I don't know. Sometime tomorrow morning. Wake up early enough. I think we could do it. I just gotta start waking up a little bit earlier. I don't know. I've just been too tired. We can do it. We can It's let's take 15 minutes actually. Well, let's see. Let's take 10 minutes. We go bathroom. Let me go bathroom. Put some eye drops. Let's work out, and then I'll do it. Let's put some eye drops. That's way too dry. We got. I think I just time it for 50 minutes so that I know when to continue the stream. And then after that, I guess I might take like a 5-minute break. I'm already classically conditioned, right? With the timer sound. Anyways, enough talk. Enough talk. Get to it. I'll put you guys down. Come back here. It's too noisy, bro. Let's go to like middle. Yeah, it's really loud there. Too much noise, dude. Shh. Shh. No, it's too bright. I wonder if I can do it without the glasses. Let's just change the. Too bright, too bright. Oh, Jesus, that's a lot better. And the brightness, let's change it down too. I don't want to hurt my eyes so much. We're like 70. Perfect. Put you over here. Oh, by the way, are you guys seeing it? Oh, yeah, you guys are. Okay. Focus up. Resolution. Jeez, is that a quick recap? Really? No, but it's kind of we'll see. Change in the genetic makeup. Gotcha. Excuse me. Okay, let's go. Macro. Oh, gotcha. Is that true though? An individual? Just that wouldn't make sense because an individual is part of the population. I still want to wear the glasses. Maybe we can switch to the red glasses for a second. right? Maybe let's put the cap on for lesson 12. That way we can get it done a little bit quicker.
build on that to develop the hardy. Use it a new color. Now that we're familiar with the idea of allele frequency, let's build on that to develop the hardy. Use it a new color. Right over here. The hardy Weinberg, which is a really useful principle. Probably have if you found someone of what percentage of a population might be homozygous recessive or homozygous dominant, or might be a heterozygote, and it really builds on the work we've already seen with allele frequency. Now, before we go into that, we're going to make some assumptions, and these are all just assumptions that get us a stable allele frequency in the population from generation to generation. We're going to assume that there's no selection, no natural selection is going, or even unnatural selection is going on that would change the allele frequency. So it's not like people with one of the alleles or another are going to be more or less likely to reproduce and have viable offspring. We're also going to assume no mutation. So we're going to assume that one of these alleles isn't from generation to generation turning into another one, or turning into maybe a different, a new type of trait, whether it's green eyes or whatever else it might be. And we're also going to assume large populations. So that would definitely throw out the example that we looked at in the last video, which I did just to understand the notion of allele frequencies, where we said, hey, look, one out of the four of the gene isn't from generation to generation turning into another one, or turning into maybe a different, a new type of trait, whether it's green eyes or whatever else it might be. And we're also going to assume large populations. So that would definitely throw out the example that we looked at in the last video, which I did just to understand the notion of allele frequencies, where we said, hey, look, one out of the four of the genes in this population, or 1/4 of the alleles in this population, are the dominant brown, while 3/4 or 75% were the recessive blue. We were going to assume large populations, turning into maybe a different, a new type of trait, whether it's green eyes or whatever else it might be. We're also going to assume large populations. So that would definitely throw out the example that we looked at in the last video, which I did just to understand the notion of allele frequencies. Where we say, hey, look, one out of the four of the genes in this population, or 1/4 of the alleles in this population, are the dominant brown, while 3/4 or 75% were the recessive blue. We're going to assume large populations, so many. And so that's so that if you have very small populations, you can imagine that depending on how these reproduce, it's very easy to get to changes in frequencies. With larger populations, that helps us make the assumption that we have stable allele frequencies. So once again, this is also that we have stable allele frequencies. Now, based on that, we've already seen if we can take the frequency of the dominant trait, which we can denote with P, and to that we add the frequency of the recessive trait, allele, we be very careful here. The frequency of the dominant allele, and to that we add the frequency of the recessive allele. What's that going to be? Well, you see in this case, it'll add up to 100% or 1, and it's always going to add up to 100% or 1, because we're assuming that there's only one of two alleles in the population. So you have a 100% chance of getting one of these two. That whatever percentage is going to be, whatever the frequency here is, 100% minus that is going to be whatever Q is. So these two things are going to be equal to 100% or equal to 1. And now we can start to do a little bit of interesting mathematics. It allows us to start thinking about things like homozygotes and heterozygotes. And so to do that, let's square both sides. So let's square both sides of this. A little bit of algebra in biology class. And so when you square the left-hand side, this is just squaring a binomial. You might want to review this. This looks like Latin to you. There's many algebra videos on content that go into this. This is going to be p squared plus 2 times pq plus 2 times pq plus q squared. And of course, 1 squared is still going to be equal to 1. Now, what are each of these terms here? Let's just think about something. P squared is the same thing as p times p. Well, P is the frequency of your dominant allele. So this is the frequency of your dominant allele, the percentage of the allele population, I guess you could say, the allele frequency.
So P squared is the heterozygous genotype.
Let's see, that is dominant, and you multiply that times again. Well, another way to think about P is this is the probability if you were to randomly pick one of these genes. So if you were randomly to pick one of these 4 genes, and here I'm just using my oversimplified population, of course, for the truths that we're about to surface to be true, you're going to assume a large population. But in this one right over here, one way to view P is what's the probability if I were to pick a gene at random? What's the probability that it is the variant, or it is the dominant allele? What is the probability that it represents the brown variant? So that's one way to view P. So the probability of getting let's just write it that way, a capital B, a dominant brown allele. So what's P times P? That's the probability of getting 2 dominant alleles. Or another way of thinking about it, this is the probability for someone in the population to be homozygous dominant. So it's the probability of someone being capital B and capital B. And so what is, by the same logic, what is Q squared? Well, Q squared, that's just Q times Q. Q is the probability of getting 1 recessive allele. So this is the probability of getting 2 recessive alleles, 1 from your mother and 1 from your father. So this is the probability of if you were kind of randomly born into this population of getting two recessive alleles. Now, what is this middle term right over here? Well, P times Q. So PQ. So one way to think about it, if you said, what's the probability that from your mother you get the random, you know nothing about them, or if you pick a random mother and a random father, what's the probability? I have to be careful here. So if you're just randomly getting alleles, what's the probability that from one side you're going to get the dominant, and from the second side you're going to get the recessive? So that would be PQ. That would be P times Q. P times Q. So that's getting it from, say, from one parent, and then that's from the second parent. What about the other way around? From the first parent, you have a Q probability of getting the recessive one, and from the second parent, you have a P probability of getting the dominant one. So there's two ways of becoming a heterozygote. And so if you add these two probabilities, what do you get? These are both PQ. I'm just changing the order of multiplication. You sum these two, you get 2 PQ. So this is the probability. This is the probability of being a heterozygote. This is the probability of being a heterozygote. So this is a pretty neat result. Just by making a few assumptions and reasoning through the solution of allele frequency, we're able to come up with this expression that actually is fairly powerful in thinking about allele frequency in a population, and actually the different genotype frequencies in a population. You see, it all makes sense. These all add up to one. The probability of someone being homozygous dominant plus the probability of someone being a heterozygote plus the probability of someone being a homozygous recessive, they're going to add up to 100% because someone's going to have to be one of these three things. I'm going to leave you there in this video. In the next video, we're actually going to use this Hardy-Weinberg equation to actually come up with some very interesting results about the population.
So that's genotype frequency.
So let's stick with this idea, the simplification, that there's a gene for eye color. It only comes with two variants. It has the dominant variant, which codes for brown eye color, and it has the recessive variant, which codes for blue eye color. Okay. So if you have if either one of your alleles is capital B, you're going to have brown eyes. The only way to have blue eyes is to have lowercase. Is to be homozygous for the recessive allele. Now let's say that in a population, it's a large population, one that meets all the Hardy-Weinberg equilibrium assumptions. Let's say that you were to observe that 9% of this population has blue eyes. So now we're talking about the phenotype. You can actually observe that they have blue eyes. So based on this, can we figure out P, which is the frequency of the dominant allele? Can we figure this out? And can we figure out Q, which is the frequency of the recessive allele? Can we figure that out as well? And I encourage you to pause this video and based on what we saw the Hardy-Weinberg equation, can we figure these things out given this information? Let's revisit the Hardy-Weinberg equation.
Well, possibly, if blue is recessive, then that's BB, and then we can just work the math, right?
We've worked it out in the previous video, but I'll rewrite it right now. It says the allele frequency for the dominant allele frequency squared plus 2 times the dominant allele frequency times the recessive allele frequency plus the recessive allele frequency squared is equal to 1. And we saw that this just comes from the idea that P plus Q is going to be equal to 1. There's a 100% chance if you were randomly pick a gene that it's one of these two variants. Now, when you say 9% has blue eyes, what does that mean? Well, the only way to have blue eyes is if your genotype is homozygous recessive. If you have capital B in here, then you're going to have brown eyes. So we can say that 9% also has this genotype. Or you can say that the frequency in the population of this genotype is 9%. But we've already seen that's exactly what this term right over here is. That's this Q squared term. This is the probability, one way to think about it, this is the Q, of course, is the frequency of the recessive allele. Now, you could view this as the probability of getting 2 of the recessive alleles is going to be, if you're in your population, it's going to be 9%. So we could say Q squared is equal to 9%. Another way to think about it, this term right over here is 9%.
So there you go. Now we can just plug it in. P is equal to, P plus Q squared. So 9% squared is equal to 1. We got P plus 81% equal to 1. P is equal to minus 81, which is P is equal to 19. Where P is what again? Frequency of A.
0.09. That's what this 9% has this genotype. That's what this tells us right over here. So then we can solve for Q. If Q squared, I'll write it as a decimal, 0.09. That means that Q is going to be the square root of 0.09, which is equal to 0.3.
Whoops. What? I thought we squared Q. Oh yeah. Whoops. Whoopsie daisy. Well, whatever. Well, yeah, because P plus Q squared is 9. Well, it's 0.09. Square root of 0.09, which is 0.3. which is
So just like that, we were able to figure out the allele frequency of the recessive allele. 30%, I can write that as a percentage, 0.3 or 30%. If you look at the genes in the population, 30% express
Q is equal to
our
30%.
code for the recessive allele, are the recessive variant.
Yeah, so P plus Q. So we got P is equal to 70%.
And so based on that, we can figure out what percentage code for the dominant variant. The rest of the genes must code for the dominant. We're assuming there's only 2 of them. P plus Q equal 100%, or P plus Q is equal to 1. So this must be 70%. So just based on that, we can kind of dig a little bit deeper here. So what is P squared? P squared is going to be 70% squared, or 0.7 squared. So this right over here is 0.7 squared, which is 0.49. So one way to think about it is, based on this, and once again, a simple equation, but these really neat ideas are starting to pop out of it. Based on just this information, we're now able to say that 49% of the population is going to have a genotype of capital B, capital B. They're going to be homozygous dominant. And then we can figure out this right over here. 2 times P times Q, that's going to be 2 times 0.7 times 0.7 times 0.3 times 0.3. So let's see, that's going to be 2 times 0.21. So this is going to be, this right over here is going to be 0.42. Another way to think about it is 42% of this population is going to have the genotype uppercase B and lowercase b. And you see they all add up. 49% plus 42% is 91%, plus 9% all adds up to 1, all adds up to 100%. So you get a little bit of information here. Based on what we know about allele frequencies, making a few assumptions, we're able to get a lot more knowledge about this population. And this is actually very useful in real life. When people think about, say, a recessive allele that might cause some type of a disease, based on the incidence of that disease, people can start to think about, well, what percentage of the population is a carrier, say that they're heterozygote for that disease. This is actually very useful in real life.
You go whooshing. Yip, yip. Yip, yip. Yip, yip. Videos. Up until article.
So today we're going to talk about a topic that's very central to the idea of evolution, and that's natural selection. But before we get into that, I want to talk about what evolution isn't. So evolution isn't when some organism, like this monkey, magically transforms into a human. And it's also not when an organism changes in some way when it's in trouble, like a person growing wings after jumping off a building. And I want to clarify that this is not what we're referring to when we think of evolution. And evolution is a process that occurs to populations of an organism, not individual members. And it occurs over huge amounts of time, and we're talking millions and millions of years for even small changes. So natural selection is one of the forces that ultimately drives evolution. But what is natural selection exactly?